A very big hello to all my wonderful PUC champions. I hope all my dear children are doing great. So my champs were asking about the 5-marker question. So there goes this particular video. And I have also included the HOTS question, high order thinking question. So if at all you were someone who are facing difficulty while solving questions from the question bank, most probably you might see those questions solved over here. So do not be worried. And a disclaimer, I might have a bit low energy today. I have little fever and cold. So please try to be self-motivated, yeah? And if at all you think that this video is doing great, it's helping you well, what do you have to do? Click on the subscribe button at that point itself. If you feel, you can click on the like button and comment down in the comment section as well. Also, if you have any doubts, please be free to comment it down. I will be taking that into consideration and giving you the answers. Cool. So let's go ahead with the first chapter, sexual reproduction flowering plants. I've already made a video on all 1-marker, already made a video on all 2 and 3-marker, and now it's time for the 5-marker questions out there. And here goes the first question. Plan an experiment and prepare a flow chart of the steps that you would follow to ensure that seeds are formed only from the desired sets of pollen grains. So basically, I am going to make a seed with the desired sets of my pollen grains, whatever pollen grains that I would like to, I am going to make it go land on the stigma, and I'll make seeds depending on what plants I want to fuse it. So what do you mean by that? Basically, I am going to do artificial hybridization. So this is the first thing that you need to crack it, right? So you have to plan an experiment for that, as in you have to basically explain all the steps of artificial hybridization. Now, name the type of experiment that you carried out. Baba, what is the type of the experiment that we carried out? It is artificial hybridization. And write the importance of such experiments. Importance will be what? Basically, suppose I want some desired characters in one particular plant. I can get that using what? Artificial hybridization technique. That is a desirable character, right? It can be useful for crop improvement programs. I want varieties of different crops which will sustain really well in drought condition, in high salinity. All of it, I will be easily able to get it. Now, the thing is, you have to explain about all of it. So how do you explain it? This is the flowchart that you need to follow. You need to write about all of this. What's the first thing that you need to do is that you have to do the selection of the parents. Choose a desired female parent and the male parent. Okay, the female parent could be there and a male parent. Suppose there's a female parent that I have taken, and over here, my dear children, there's the female part as well as the male part. Okay, and there is another plant where basically I have, so this is my male parent, and this I want to be the female parent. The problem over here is that this female parent is also having pollen grains, or basically is having the male reproductive part. So that means the pollen grains can land on the stigma and fertilization can happen this way. But actually, what I want to do is I want to collect the pollen grains from here and I want to put it over there. That's my logic out there, because I want both of their characters to come in the next generation. So the first thing that I have to do is that whenever a flower is in bisexual condition, that means it has both the sexes, male and the female sexes, I would have to remove the male part from here. If only the plant that I am taking over here is bisexual, the first thing that I would have to do is removal of the male part, which is known as emasculation. Emasculation. Okay, I am removing the masculine part. So emasculation is being done. I have removed it. Now what do I do after this? So selection of parents is one thing. Suppose this female parent is bisexual, I do emasculation. Apart from that, the next step that we would have to follow is that first one, select the parents. Second one, emasculation. Now the third step is going to be bagging. Okay, what do you mean by that? So suppose over here, if I remove this, there are multiple other flowers. The pollen grains can land from here and come on this particular plant, right? Is it okay? No, because I just want the pollen grains of this plant to come over there. So what do I need to do is I need to bag this up. I need to bag the whole thing up such that no pollen grains can land on this, and also it is to make sure that the stigma which is there, it is becoming receptive. Basically, it is becoming mature enough to take that pollen grain whenever the chance comes. Okay, so the next third step which we have is what? Bagging. Bagging. Okay, now after bagging, what do we do, Baba? After bagging, we would be removing the bag. We are going to remove the bag and what? And dust the pollen grains over here. So I'll be removing the bag and I'll be dusting the pollen grains right over here from this particular plant. From this particular plant, I will be putting that. So dusting of the pollen grains, or basically transfer of the pollen grains, that is going to be done from what? From the desired male plant, right? Now after that is being done, I have to make sure no other pollen grains comes and lands over it. So what do I need to do? Again, I have to bag it up. So the next process after doing this would be what? Rebagging, Baba. It will be rebagging. I will be rebagging it. And after rebagging, I also have to do one more very important thing, which is known as tagging. After this, I have to do one more very important thing, that is known as tagging. What are we tagging over here? We are tagging from which parent did we do the cross, so that we know we have an idea about what type of progeny is going to be formed later. You get it? These are the steps that you have to write. And also please understand, if at all it is not bisexual, it is unisexual flower. There is only one sex. In that particular case, you do not have to do emasculation. Only in bisexual condition you do that. So here I have written everything whichever is needed for you guys to write. Make sure you get this PDF. You'll get the PDF by joining the link given in the description section. That is about, there might be a link for to download the lecture notes and things like that. Join the WhatsApp channel. So join the WhatsApp channel and get your PDF. Works. So this is the whole thing. I hope this is super clear to you. Yeah. And the name of the experiment is artificial hybridization. Also remember, it is sometimes known as controlled cross-pollination as well, because in a controlled way you are doing cross-pollination. Okay. So not be confused if at all that came. And also remember, if in any place it is written that what is that technique used in crop improvement program to produce desired quality of plants, it is basically this only, that is artificial hybridization. Always remember that. Now importance of such experiment, they prevent contamination by unwanted pollen and help in producing hybrids with what desired characters such as higher yield, disease resistance, better quality, etc, etc. Works. Perfect. Now the next question that we have over here is, picture related to pollination is given below. Answer the questions that follow. So this is a picture that is given. What is happening? An insect is there sitting on the plant, right? Now, will this pollination confirm fertilization? Justify your answer with suitable reasons. What do you think? Every time whenever pollination happens, do you think that will lead to fertilization? What do you mean by pollination, Baba? There is stigma, there is style, and there is ovary. Suppose the pollen grains are landing up over here. This is what you mean by pollination. This is pollination, as in transfer of pollen grains from the male to the female part, right? What do you mean by fertilization? Where basically the male gametes which are there inside the pollen grain will be fusing with the female gametes that is there in the ovary. That is known as fertilization. So, pollination has to happen, after which pollen tube will be forming. The pollen tube will be getting inside and transferring the male gametes. But always remember, this is not necessarily going to happen. After pollination, some pollen tube might form, some might not form. So pollination does not guarantee fertilization. Always remember, for fertilization to happen, obviously you need pollination, but every pollination which is there will not be guaranteeing you fertilization. This is a very important thing. Okay, so so answer for this is, pollination only brings the pollen grains to stigma. It is just a transfer process, right? Fertilization occurs if the pollen is compatible, it germinates on the stigma and forms a pollen tube that reaches the ovule as well. Listen, even after forming the pollen tube also, listen, there is also one more question. After pollen germination, is it for sure that double fertilization is going to happen? Listen, even if the pollen tube is being formed, we cannot definitely say that yes, fertilization will be happening. It might happen, it might not happen. You get it? It might or it might not. Always remember it. So yes, even if it germinates on the stigma and forms a pollen tube that reaches the ovule and transfer the male gametes, then probably fertilization could be taking place. So if the pollen grain is incompatible, it will not germinate and fertilization is not going to happen. So anyway, if the pollen grain lands, suppose pollination has happened, but do you think fertilization is happening in this condition where the pollen grain itself is incompatible? So there are certain pollen grains which are not compatible, right? In that case, the pollen tube itself is not going to be formed. If at all you are getting confused, the only answer to this question is no. Okay, pollination does not guarantee fertilization. And I am telling you one more thing additionally, even if pollen tubes are formed, that does not guarantee fertilization. Even if compatible pollen pistil interaction occurs, pollen tube form, that does not guarantee double fertilization. Okay, there is more to it. Multiple pollen tubes are formed. It should be coming inside the ovule and doing the whole process. Then only we'll say that yes, double fertilization can take place. Cool. Now, what are the floral rewards provided by the plants to the insects to revisit? So, what is the reward that is given by the plants so that the insects can come again and again and again? First thing that we know is nectar. Nectar is one thing. Second thing, pollen grain itself. So, ma'am, if at all it gives pollen grain, so how does pollination will be happening? We just want to transfer the pollen grains, right? It will eat up everything. No, it does not eat up everything. There will be some pollen grains on its surface as well, which could be used for transfer. So, yes, nectar, pollen grains are given. Apart from that, you might have seen, you might have heard about yucca and moth. There, it provides a safe space for the moth to lay the eggs. So, it can be in the form of shelter also. Okay, so the floral rewards which are there can be nectar, can be pollen grains. Apart from that, it can be shelter as well, which you see in yucca and the pollen grain. Okay, yucca and the moth. Now, mention the characteristics of flower in this pollination other than the floral rewards. What is the characteristic of a insect pollinated plant? For an insect to pollinate, obviously you have to make sure that the flowers which are there, they have a good odor. Okay, they have a good fragrance out there so that it can attract the insects. Apart from that, most of them, my dear children, would be large and colorful in nature. If at all the flowers are small, they will come together to form the inflorescence. They will come together, form a cluster of inflorescence. Always remember that. And if at all the plant is being pollinated by, by flies and something like that, then in that particular case, it will be having a foul odor, because foul odor is going to attract the flies. So please know about all these few things out there. So large, colorful and fragrant flowers to attract the insects. Sticky or spiny pollen grains. Yeah, that is also there. So the pollen grains which are there, it has to stick on the surface of the pollinator, right? Then only pollination will happen. So which is why it will be sticky and spiny pollen grains. Stigma is sticky to capture the pollen brought by the insect. Yes, stigma is also going to be sticky such that whatever pollen grain is there on the surface of the pollinator, that can be transferred into the stigma also. Now, foul smelling flowers in plants pollinated by flies and beetles. And please write, if at all it's small flowers, then it will be forming cluster of inflorescence. Works? Works, ma'am. Perfect. Next question. Complete the given tabular column with respect to the male and the female gametophyte of angiosperms for the mentioned character. So this is the male gametophyte column. This is the female gametophyte column. This is the character that is given over here. You have to give the answer. For example, number of cells present in the male gametophyte. What is male gametophyte over here? First, let's decode that. Male gametophyte, Baba, is the one that is going to form the male gametes later. It is going to form the male gametes later. So the male gametophyte which is there, it is pollen grain. It is the pollen grain. Okay, ma'am. Then what about the female gametophyte? Again, the female gametophyte is the one that forms the female gametes. So what is that? It is the embryo sac. Always remember this thing. Okay, now what we are asked, number of cells present in the mature male gametophyte. In the male gametophyte, that is the pollen grain, my dear children. How many cells do you remember it's there? There's a very big cell which you call as vegetative cell. There's a small cell that is known as generative cell. This generative cell, in this particular case, you have just two cells, 1 and 2, right? Always remember, this generative cell can later divide into 2 and give rise to 2 gametes, right? It is the one that forms 2 gametes. So then in that condition, 1, 2, 3 cells will be there. So either you can write 2 or it can be 3. 2 or basically 3. Always remember that. Now, don't just write 2 and come. So mature, we'll finish this first, yeah, then we'll go there, okay. So number of gametes present in. So how many number of gametes are there, Baba, in one pollen grain? There are 2 number of gametes, so I'll write 2, okay. Now type of cell division involved in the development of. What is the type of cell division that is involved over here in the development of the pollen grain out there? I can say it is mitosis. So, if you are very much confused, okay, if you are very much confused, you can write through microsporogenesis. Through microsporogenesis, meiosis happens and you get microspores. The microspores undergo mitosis to form what? To form the pollen grains. So actually it is mitosis itself, but if at all you're confused between meiosis and mitosis, be specific. In microsporogenesis, for forming the microspores, you have meiosis. Later, it will be undergoing mitosis for forming the pollen grains. Okay. So actual answer is mitosis. If at all you can give like that itself, it will be more correct. But yeah, if you want to explain the previous portion also, you can. But make sure you underline mitosis specifically. Okay, to make the pollen grains, you need to do mitosis itself. Before that, yes, meiosis is happening. Now, ploidy of the cells of. What is a ploidy? It is diploid, haploid. What is the case over here? Always remember, all the cells which are over here are going to be haploid in nature. They will be having only half the number of chromosomes, which is why it is denoted as N. So 2N is diploid. So half of 2N is going to be what? N, that is going to be haploid. Number of chromosomes over here is always going to be haploid. So I'll say ploidy of the cells is going to be what? Haploid, Baba. And please mention haploid is denoted by N. Cool. Is this particular thing being done? Let's tally it over here. So just a second, okay? Where is it? Yeah. So mature male gametophyte has number of cells 2, 1 vegetative and 1 generative, or 3, 1 vegetative and 2 male gametes. Done. Number of gametes present, there will be 2 only, 2 gametes are there. Type of cell division, mitosis from microspore to the gametophyte. So I have mentioned over here, mitosis from where to where? From microspore to gametophyte. Before that, it is meiosis. Okay. Now ploidy of the cells would be haploid. Khatam. Okay. Now let's come back over here and let's talk about the mature embryo sac. Number of cells present in the mature embryo sac. How many number of cells are there? We say, no, always we say 7-celled, 8-nucleate condition. Do you know about it? Let's see. So over here, I have a quick animation. So suppose this is the embryo sac. 3 cells are at the top with 3 nucleus. It is known as antipodal cells. These 3 cells are known as antipodal cells. They are towards the chalazal end. This end, okay, is known as chalazal end. Always remember, they have the nucleus. All the nucleus over here, all the nucleus individually, if I talk about the nucleus, all of them are going to be haploid in nature. That means I say that these 3 cells are haploid in nature. Agreed? Agreed. Now, because since they just have one nucleus, so all of them haploid. Now here you have 2 cells, you call them as synergid cells. What do you call it? 2 synergid cells. Again, these cells are also going to be haploid in nature. Because here it is N, here also it is N, haploid. Fine. Going ahead, you have one more cell, that is the X cell, or you also call this as female gamete. This is also going to be N, haploid in nature. Okay. Now what do you have? Now you have a very big cell. And this very big cell has 2 nuclei. So the very big cell looks like this. This entire thing that you see over here is a very big cell. What is the name of this very big cell? Do you have the name also? No. So I'll mention it over here so that you do not forget it. It is known as the central cell. The central cell, Baba, how many nuclei does it have? It has 2 nuclei. None of them had 2 nuclei, but this guy has 2 nuclei. Each nuclei, I told you, is haploid. So what is going to be the ploidy of central cell? Haploid, N and N. N plus N, it becomes what? 2N. So this cell is the only one which is having diploid number of chromosomes. Rest all of them are haploid. If you talk about the nuclei personally, they are haploid only. They are known as 2 polar nuclei. Both of them are there, which is why 2 polar nuclei. Got it? Got it, ma'am. Okay. Now how many cells are there here? 1, 2, 3, 4, 5, 6 and 7. And 7 cell. So can I write over here that basically, just a second, I'll go forward, okay? So there are 7 cells. Number of cells present, 7. 3 antipodal, 2 synergid, 1 X cell and 1 central cell. Done. Number of gametes which are present over here, my dear children. How many number of gametes are there? Only this X cell is the female gamete. So I'll just say 1 gamete is there. So, okay, 1 gamete is there. Type of cell division involved over here in the formation of female gametophyte. Do you know how these cells are being formed? It is with the help of mitosis. So 3 mitotic division happens, and that is how. 3 mitotic division happens, and that is how you get these many cells. Always remember, 3 mitotic division. And when the mitosis happens, one more thing that you need to remember is that the nucleus keeps on dividing. Here, it's a very unique feature. The nucleus keeps on dividing. You call this as free nuclear division. Later on, the cell walls are being formed like this. So, it is free nuclear division that you see. Free nuclear division is seen in the case of female gametophyte. Why am I telling you this? You might get MCQ questions based on this. Anyways, okay. So, you had that. Now, type of cell division, I told you mitosis from megaspore to the female gametophyte. How does megaspore form? Through, again, meiosis. So here also microspores form through meiosis only. Here also megaspores forms through meiosis only. But later, both of them, they are going to undergo mitosis. Here, it is thrice. Okay. Now, ploidy of the cells which are there. Haploid. What about that? All of the cells are haploid except what? The central cell. Except the central cell which is going to be diploid because it has 2 nuclei. Works? Works, ma'am. Fine. So always use do not go around writing the answers without thinking for these type of questions. You have to think and then only give your answers. Do not be like fast I have to do. No, don't do that. Fine? Okay. Now the question, there is one more part of this question, that is, which advantage offered by the seeds help angiosperms to adapt to changing environmental conditions? So they are basically asking you about the advantage of a seed. Okay, seeds of what? Angiosperms, as in flowering plants. So what advantage does seed of the flowering plants give to them such that they can survive even if the environmental condition is super bad? What could be that advantage? What is there in the seed? Seed have a seed coat. Seed has seed coat, which is going to provide what? Protection. Protection against anything that is there. So protection and dormancy. The seed coat protects the embryo and allows to remain dormant during unfavorable condition. Apart from that, suppose it is dormant, it does not have any food. Does it have a food resource? Yes. Seed, my dear children, always have stored food in the form of either endosperm or maybe in the form of cotyledon, which can help it survive. Now, apart from that, if at all you talk about the seed, basically seed helps in easy dispersal. So seeds are adapted toward dispersal. They are very lightweighted, which is why they can be dispersed by wind to different locations where the conditions might be a bit more favorable, and it can allow that organism to grow up there. So these are the few things which are there that you can write for the advantages of the seeds which are there. Cool, ma'am. Works. Okay. Here 2 times mitosis happens, one to form generative and one to form vegetative cell. That is 1 mitosis. Second, the generative cell again divides into 2, that is again 2 mitosis. Here after end of 3 mitosis you get this thing. Okay, always remember that. And these 3 cells which are there, they are known as egg apparatus. Why egg apparatus? Because the egg cell is over here and these 2 synergids are going to help in the process of fertilization. Why? Because the pollen tube are going to be formed through the synergids. So the synergids are going to disintegrate, and in synergids you have one more very important thing that is known as filiform apparatus, which provides the path. It provides the path. Works? Works, ma'am. Cool. Now explain the different outbreeding devices developed by the flowering plants. So what are the outbreeding devices that are developed by the flowering plants, my dear children? Any idea? First of all, outbreeding means what? The ones that will be preventing self-pollination and promoting cross-pollination. Okay, so why do you think that plants need to prevent self-pollination? Because suppose there are some toxic traits in me, and my kids are being hung. Basically, in the case of plants, if at all it's like that, there is some bad plant over there, and the plant reproduces. What is going to happen? The same features are going to be there in the small baby plant also, and it might not be able to sustain really well. So which is why self-pollination could be really bad at times, and the plants would be having something that is known as inbreeding depression, as in because of continuous self-pollination, they will be having some very toxic characters within them such that they will not be allowed to stay in a particular environment, they will be dying off. Okay, so which is why to prevent this and to promote cross-pollination, we have outbreeding devices which are there in the plants, and you are being asked about that. So how do you frame your answer for this? Listen, mechanism to promote cross-pollination, the first one is time difference. Pollen release is happening, but the stigma is not receptive. So even if the pollen release happens, since the stigma is not receptive, pollen tube is not going to be formed. So that is one method in which I can prevent what? Self-pollination. Second thing, okay, so the next thing that we have is spatial separation. What do you mean by spatial separation? So over here, you have your where is it? No, it is not working. Okay. You have the female part, right? This is the female part. Suppose you have the male part over here. It's not too long. It's just somewhere over here. What is going to happen? The pollen grains which are there, they have to move like this and land on the stigma. That is not clearly very much possible, right? So that is what you mean by spatial separation. Plants, my dear children, have adapted a way where the anther is positioned in a way such that it cannot transfer the pollen grains. So anther and stigma are positioned differently. That is spatial separation. The third thing that we have is self-incompatibility. What do you mean by that? It's a genetic mechanism which the plant uses, such that even if pollen grains land on the stigma, it will not allow the pollen tube to be formed. There is some genetic mechanism that is blocking from the pollen tube formation that is there. That is known as self-incompatibility. Genetic mechanism preventing what? Self-pollen from fertilizing the ovules out there. Very, very important. Now the fourth one that we have is production of unisexual flowers. Production of unisexual flowers. How does that help, ma'am? So suppose one plant is just having the female flowers, as in it is just having flowers with the female part. Obviously, it has to rely on some other plant for fertilization to happen, right? Yes. So this type of plants which have two separate sexes, first of all, please know them. They are known as dioecious plants. What do you call them? Dioecious plants. Example, papaya. Example, papaya. So papaya plant, if at all you have seen, there is female plant and there is male plant. There is female plant as well as male plant. So in this particular case, male and the female flowers are going to be on different plants, and that is definitely going to help in what? Promoting cross-pollination out there. So here what all things are going to happen, my dear children? In the case of papaya, autogamy is definitely not happening. Autogamy means self-pollination from here to here. So since there is no pollen grains, which is why there is no autogamy that is going to happen. What about geitonogamy? Geitonogamy basically means from here, there's a flower and there's another flower. Do you think pollination can happen between two flowers of the same plant? No. Again, there's just female part over there. How do you think pollination can happen? So autogamy and geitonogamy is prevented. What is promoted? Xenogamy. Xenogamy means pollination between two different plants altogether. Two different flowers of two different plants. Okay, so always remember, in dioecious plants, autogamy and geitonogamy is prevented and xenogamy is possible. Okay, this is dioecious plant. The example for this is papaya. Now you have monoecious plants. Monoecious plants means a plant having both the sexes, female as well as the male sexes. Okay, and how do you think here you can prevent cross-pollination? Sorry, here you can prevent self-pollination. You cannot exactly prevent self-pollination, but let me tell you, to some extent it is possible. Suppose in a plant, okay, in a plant, I am saying flowers are not like this. The male and the female flowers are not together in the same flower. Rather, on the same plant, you have two different flowers, one with female flower and one with male. One with female and one with male. Okay. So that is being seen in the case of castor. So monoecious plants are the one that has both the sexes in one plant. Either it can be in this way or it can be in this way. We are talking about this. In this, anyway, autogamy is going to happen. Okay. Here, what are you saying? There are less chances. You are making the chances less for autogamy to happen and for promoting cross-pollination. From other plants, here easily things can be coming up. There is less competition of self-pollination, right? You get it? So to some extent, you can basically do that. And this is the example of castor. In castor, it's not like this, it's like this. There are separate male and separate female flowers, but on the same plant. Again, over here, autogamy is prevented, but geitonogamy is not. Geitonogamy is not. Pollination can still happen from the same plant parts, but yeah, it is reduced to so much extent, and it helps in proper cross-pollination to happen from other plant also, because there is no pollen grains of itself, right, in the same flower. So example for this is castor and maize. CM, Chief Minister, you can remember, male and female flowers on the same plant prevents autogamy, but not geitonogamy. Know about this. If you do not understand this, you will not be able to answer this. Okay. Works. So these are the things that will be promoting cross-pollination. These are the outbreeding devices out there. Cool, ma'am. Let's go ahead. Now, double fertilization is a unique event taking place in flowering plants. Briefly explain. How do we explain double fertilization? Any idea? Very, very simple. Listen to me carefully. So, I'll come back over here, and I hope you do not mind if at all I remove this. So, what exactly happens is that I told you your pollen grain which is there, it has two gametes, right? So there is one gamete and there is another gamete. This is male gamete, this is also male gamete. Two male gametes are there. 1, 2. When the pollen tube formation occurs over here, what happens is that one male gamete which is there fuses with the female gamete. Okay, the first one. Now there is one more male gamete. One more male gamete that is there is going to fuse with the two polar nuclei of the central cell. So first fertilization, second fertilization, 2 times fusing is happening. That is why I call it double fertilization. What do you get when the male and the female gamete are going to fuse? You are going to get a zygote. Okay, what is the ploidy of the zygote, ma'am? So always remember, gametes are going to have half the number of chromosomes. So here the male gamete is having N. Female gamete will also have N. N plus N, you will get 2N. Diploid over here. Okay, diploid zygote is being formed over here. What about this? So here, my dear children, in the case of the male gamete, it had haploid number of chromosomes. Here the two nuclei had haploid number of chromosomes. 2 nuclei has haploid number of chromosome. So what is going to happen? N plus N plus N. You are going to get something that is having 3n number of chromosome. And what do you call this particular cell as now? What do you call this particular cell which has 3n number of chromosome? It was a central cell fused with the male gamete. Now you will call this as primary endosperm cell. Primary endosperm cell, which is triploid in nature. Which is what? Triploid in nature. This nucleus which is there, I'll just call this nucleus with the fusion of all this thing. When the nucleus is being formed, I'll call it primary endosperm nucleus. So primary endosperm nucleus is of primary endosperm cell. This primary endosperm cell, Baba, later it will be forming endosperm. Why do you need endosperm? Endosperm is super important for the zygote, for providing nourishment to the zygote, such that the zygote can form an embryo. That is how it is usually going to go. Please remember, this is super important. Works. Okay, so basically this type of fusion which is happening over here with the male and the female gamete, I'll call this fusion as syngamy. I'll call this as syngamy. This fusion which is happening between 3 nucleus out there, or basically 2 cells out there, I'll call this as triple fusion. But why triple fusion? Since 2 nucleus are being fusing. What is the end result of this? Primary endosperm cell with triploid nature, okay? Or you call it primary endosperm nucleus, if at all you're just talking about the nucleus. Anyway, the cell which is there, which is being formed, it will be later forming endosperm, and the endosperm forms first. Endosperm is going to be, primary endosperm cell is later going to form endosperm tissue, and this endosperm tissue is going to help with the zygote, such that the zygote will be transforming into the embryo. Cool. So that is double fertilization. Now again, how do you frame this answer? So in flowering plants, 2 male gametes enter the embryo sac through the pollen tube. Agree. One male gamete fuses with the egg cell to form a diploid zygote. This is known as syngamy. Okay. The second male gamete fuses with the 2 polar nuclei in the central cell to form the triploid primary endosperm nucleus. And this is of the cell, that is primary endosperm cell. The primary endosperm cell later divides to form what? The endosperm tissue. Okay, and this endosperm is going to help in nourishment of zygote to form embryo. Fine. Because 2 fusions occur in the same embryo sac, it is known as double fertilization. A feature which is very unique to what? Angiosperms. Cool. Done, ma'am. Now, there is one more question that is given to you. Arrange the following terms in correct developmental sequence. So, pollen grains, sporogenous tissue, microspore tetrad, polar mother cell, and male gametes. That means you are being asked about one very important thing, that is microsporogenesis. The steps involved in microsporogenesis. So, what is the step? First thing that you have is basically lot of cells over here. These cells are known as pollen mother cell, PMC, or I can also call it microspore mother cell. Either way, these cells which are there, they belong to something, a tissue which is known as sporogenous tissue. So tissue is made up of multiple cells, right? So these cells are going to form sporogenous tissue. You can remember like that. So basically, if I want to go one by one, I'll say sporogenous tissue is there. In that you have pollen mother cell. This pollen mother cell which is there, it is diploid in nature. It is what? Diploid in nature. It will undergo meiosis. Meiosis happens in 2 stages. First stage, it will give you 2 cells, and in this stage itself, the number of chromosome becomes half. It was 2n over here, it becomes half over here. Okay, now these cells again divide, you get 2 cells over here. The number of chromosomes remains the same. Here also it divides, again you get 2 cells. Here the number of chromosomes remains the same. This is going to be known as meiosis 2. Okay, at the end what do you get, Baba? These 4 cells, first of all, you need to understand, you call them as microspores, and the microspores are always close together, which is why I'll call them as microspore tetrad. What do I call it? Microspore tetrad. They are a group of 4 of them. It's like that. The microspore, it's not like all of them were individual first and then they form the tetrad. No, they form the microspore tetrad the first. Later, they are going to dehydrate. They will be moving away from each other, giving rise to microspores. Now, these microspores are going to form what? The pollen grain. So, the first one which is there is going to be sporogenous tissue. The second one which is there, it is going to be the pollen mother cell. The third thing which is going to be there is it's the microspore tetrad. The microspore tetrad later form the microspore, forming the pollen grains, and the pollen grains will be having what? The male gametes. So this is how it is going to go. Works? So that is exactly written over here. Go through it. Fine? Fine, ma'am. Okay. Now, this is a question bank HOTS question that I saw. That is, double fertilization is a unique feature of angiosperms, and the products of this double fertilization are zygote and primary endosperm nucleus. We just saw it, right? In context to this, when a hexaploid plant is pollinated by a tetraploid plant, find out the ploidy of zygote and PEN through a schematic illustration. What? Listen, what are you fusing? Hexaploid plant. What do you mean by hexaploid? That means 6N. Okay, it is pollinated by what? Tetraploid plant. That means the pollinated by. That means it is going to be the male plant. Male plant is what? Tetraploid. Tetra means 4. So it is 4N in nature. The female is what? 6N in nature. Now find out the ploidy of zygote and PEN. Okay, how do you think zygote is being formed? With the help of female gamete and the male gametes fusion, right? What is the ploidy of female gamete over here? Gametes are always going to have half the number of chromosomes which is there. So if this was 6N, half of 6N is equals to what, Baba? It is going to be 3N. Okay, it is going to be 2 cancel 3, 6, 3. Okay, so it is going to be 3N. Similarly, if it was 4N over here, half of 4N is going to be what, Baba? Again, you do the calculation, it becomes 2N. Now what do? You do not do into, you do plus. Okay, 3N plus 2N, they are going to come together, right? 3N plus 2N, number of chromosomes, they are going to come together. What do you get? 3 plus 2 is equals to 5N. It is going to be pentaploid. Pentaploid, where this is about the zygote. Now you have been asked about one more thing, that is primary endosperm nucleus. Again, how did primary endosperm nucleus form? Primary endosperm nucleus is basically a combination of the male gamete, and male gamete, we know it was 2N in nature, and it is a combination of what? It is a combination. So instead of doing this, I'll do plus, otherwise you will get confused. Okay, here also I'll do plus. It is a combination of male gamete as well as the 2 polar nuclei, or basically I can just say the central cell. Okay, one polar nuclei will be having half the number of chromosome. If 2 polar nuclei is there, central cell has 2 polar nuclei. One polar nuclei will be having 3N, just like any other nucleus out there. There is 2 nuclei, so I'll add one more 3N. 2 polar nuclei there. 3 plus 3 is equals to what, Papa? 6. Now 6N is the totality of the central cell out there. Now this 6N plus 2N is equals to what? 6N plus 2N. 6 plus 2 is equals to what? 8N. This is going to be the ploidy of the primary endosperm nucleus. Again, I am telling you, primary endosperm nucleus is being formed when 2 polar nuclei of the female. One polar nuclei will be having half the number of chromosomes. Another polar nuclei will also have half. So 3 plus 3. Half is this number, right? So 3 plus 3 is equals to 6N over here in the central cell, right? And the male gamete is having half the number of chromosomes, that is 2N. So basically 6 plus 2 is equals to 8. You got it? So this is going to be octaploid. You need to write your answer like this exactly. I've given everything which is needed for you. So yes, I'll just write 1 by 2. Cool? Cool, ma'am. Perfect. Now, one more question is there. I thought I'll discuss it here, and that is double fertilization is reported in plants of both castor and coconut, groundnut. Double fertilization is happening both in castor as well as groundnut. Okay. However, the mature seed of groundnut is non-albuminous. Groundnut which is there, it is non-albuminous. What do you mean by that? Non-albuminous means mostly when I talk about a seed, seed has what? Inside it, it has embryo. How did the embryo come? Embryo came with the help of zygote. Zygote transformed into the embryo, right? Now, to provide nourishment to the embryo, or basically to form the embryo, you had endosperm. You had some endosperm. Some of the organisms use up the endosperm completely. Completely they use up the endosperm to form the embryo. From zygote to embryo transformation, you need endosperm to do this, and some utilize the endosperm completely. You do not have any endosperm left inside the seed. you do not have any endosperm left inside the seed, you call this as non-albuminous condition. There is nothing, no endosperm up there. Now when you talk, this is the case of groundnut, because groundnut is non-albuminous. Now what about castor? In castor, my dear children, the endosperm which was there, it is not completely utilized. It is still there. It is still there in that particular case. Okay, so since endosperm is still persisting, I will be calling this as what? Albuminous. I will be calling this as albuminous. Please understand, groundnut as well as castor, both of them, they are dicots. Dicots means basically they usually dicots use up the endosperm to form two cotyledons. So they usually have two cotyledons. Mostly endosperm is totally utilized for this purpose only, for formation of two cotyledons. But in the case of castor, even after being a dicot, it has two cotyledons, but also it has some endosperm left behind. That is why I'll call this as albuminous. Usually, okay, leaving back of endosperm is found in the case of monocots, but this guy is an exception. This guy is an exception, and I'm not saying mostly it happens, I'm saying usually this is the case of monocots, but yes. So explain the post-fertilization event. So how do you explain it? Right. In both castor and groundnut, double fertilization occurs. Zygote develops into embryo. Primary endosperm nucleus develops into endosperm. In castor, the endosperm persists in the mature seed. Hence, the seed is albuminous. Right. Here in groundnut, the endosperm is completely consumed by the developing embryo. Hence, the mature seed is what? Non-albuminous. So, you just have to give the explanation of the definition out there. Now, what do you have? Banana is a parthenocarpic fruit, whereas oranges show polyembryony. Explain, how are they different? So, parthenocarpy means what? Usually, Baba, what happens is that we have ovary, inside that we have ovule, right? So, ovary which is there, it transforms into a fruit, and ovule which is there, that transforms into a seed after fertilization. This is after fertilization. Until then, you will call this as just ovule and ovary. After fertilization, you will call this as seed and fruit. But in some cases, my dear children, where is it? Yeah, in some cases, without fertilization, as in there is not going to be any formation of seed over here, still the ovary will be transforming into fruit. Ma'am, how is that possible? Under the influence of some hormones, okay? So under some influence of some hormones, such as auxins and things like that, here the ovary transforms into a fruit without fertilization. So fruit without fertilization is known as parthenocarpy. Seed formation without fertilization, that is known as apomixis. Please know the difference, okay? Seed and fruit difference is there. Now, that is about banana. Now, when you talk about oranges, oranges show polyembryony. What do you mean by that? So, in the case of orange, my dear children, again, you have the ovary, you have the ovule. Inside the ovule, no, sometimes the nucleus cells might be there. There might be cells of nucleus. And here you have the embryo sac where you have the female gamete and things like that, right? Sometimes, inside this female gametophyte, that is embryo sac, the cells of the nucleus might get inside it. Once they get inside it, they are going to form the embryo. And you know what? They will be forming multiple embryos. Polyembryony is what you call this. And this is usually found in citrus fruits such as oranges, mango, etc., etc. Ma'am, over here, since it is getting inside the embryo sac and forming the embryo, here also fertilization is not happening, right? No. So can this be an example of apomixis also? Yes. So apomixis can happen in this case. Can happen in this case where polyembryony is being seen, but it is not necessary that every time it has to be done in this way only. Apomixis can happen in another way where the female gamete which is there in the gametophyte, instead of being haploid, it is actually diploid. The female gametes are usually haploid, right? But here it is diploid, which is why it does not need any male gamete. And that's also a way in which you can, basically you can transform this female gamete into what? A zygote. A zygote can form in embryo there without fertilization. So that is one way of apomixis. This is the second way of apomixis, which you call as polyembryony. Cool? Yeah. Apomixis is generally seen in Asteraceae and grasses. Polyembryony, a specific condition, is seen in the case of citrus fruits. Okay, ma'am, how do you write the answer? So, the ovary develops into fruit without fertilization, that is parthenocarpy. This occurs under the influence of what? Plant hormones. Can it be induced? Yes. So, in this fruit, do you think seeds will be there? No, Baba. Fertilization didn't take place, no. So, that's why seedless varieties of fruits will be formed over here. Oranges is polyembryony. More than one embryo is formed in a single seed, and additional embryos arise from the cells of the nucleus that I just told you. Hence, seeds are present containing multiple embryos. And that's your answer. Got it? Great. And with this, we complete one chapter, and now we get entry into the next chapter. I think we took so much time. Okay, but yes, in some chapters, 5-mark questions are not there, so probably it should be ending up really quick, and I'll be fast from this point onwards. Just give me a quick second. I'll just come by drinking some. I'll drink some water and come back. Okay? Yes. Okay. Let's go ahead with human reproduction. The first question is, draw a neatly labeled diagrammatic view of the female reproduction system. Over here you have to mention each and everything, as in the ovary, the parts of the fallopian tube, and every single diagram labeling should be there, including the uterine fundus region. Okay, so I'm not explaining much. The diagram is given. You have to practice it. Okay, and by the way, do you want a separate video on the important diagrams? You can let me know in the comment section if at all you need it. So in this 5-marker, you can get one diagram from this chapter. Always remember that. Okay, so draw a neatly labeled diagram of sectional view of mammary gland. This is the mammary gland. Again, please know the flow of it generally, which is like this. Dal fell on mat. So basically, write mat first, and then you put dal. What is mat standing for? This is mammary lobes. Mammary lobes have alveoli. Alveoli opens into tubules. Tubules opens into duct. Duct is going to form ampulla. Ampulla is going to open into lactiferous duct. That is how it goes. So dal fell on mat, or mat dal. Whatever you want to remember, you can remember. And yes. So 2A is there. Here alveoli, there ampulla. So ampulla ends with L. So you can remember like that also. That after this A you have L, that is lactiferous duct. So yeah. So here also the flow is the same itself. So see these things. You have multiple lobes. The lobes are opening into small alveoli. Alveoli, my dear children, will be opening into what? Tubules. Tubules which are there, these small things which are there, they are forming what? A duct over here, and the duct is going to come together from multiple ducts are going to come together, form the ampulla. Ampulla will be forming what? Lactiferous duct. Over here you have the nipple, you have the areolar tissue, and the back side you have rib. In between the ribs you have muscles out there, and fat is also towards the side. Okay, fat is also there towards the side. So this is general idea. Yes. Yeah? Yes. Next thing, write the schematic representation of spermatogenesis. This I can explain to you right now, because I think it's something that might need explanation, right? So let's go ahead. First of all, when I say spermatogenesis, the first thing that should be coming into my mind is it starts from puberty. And over here, what do you have? You have some cells which are known as spermatogonia. These cells are normal cells. It will be deployed in nature. Now, these spermatogonia which is there, it will undergo mitosis. What will it undergo? Mitosis, to form multiple cells, to increase their number, right? Now, when they increase the number like this, one cell among them becomes differentiated. I'll say it matures. It feels that I want to form the sperms later on. This cell is going to be known as what? The primary spermatocyte. The primary spermatocyte. How many number of chromosomes are going to be there? Baba, 2n only. Why 2n? Because this is mitosis. This is not meiosis, it is mitosis. This primary spermatocyte is going to divide. And from here onwards, meiosis is going to happen. And you know meiosis happens in 2 stages. So this is the first stage when you get 2 cells. What is an haploidy of these cells? It becomes half. After the first meiosis, it becomes half. Haploid numbers. So from this point onwards, it becomes haploid. And what do you call these cells as? Secondary spermatocyte. From primary, you got secondary spermatocyte. Now what happens? You need to do meiosis 2. In meiosis 2, what is going to happen? Let's see. You get 2 cells over here, you get 2 cells over here. Here the number of chromosomes remains the same. Here also the number of chromosome remains the same, right? And this is going to be what? Meiosis 2. This is going to be meiosis 2. What do you call these cells as? You call these cells as spermatids. Ma'am, isn't it known as spermatozoa? No. These are spermatids. They are usually globular in shape. They have not attained the elongated shape until now. So how do you remember who came first, spermatids or spermatozoa? Spermatids, they are like kids. They are immature. They are small kids. So now the immature. So until now you had spermatogenesis. Still you have spermatogenesis. Listen, this is a last stage which is there, where The spermatids, the immature spermatids are going to transform into mature sperm. What do you call them as? You do not call them sperm, rather you call them as spermatozoa. What do you call it? Spermatozoa. And ma'am, is this technique having a specific name? Is this process having a specific name from here to here? Yes. This is known as spermiogenesis. It is known as spermiogenesis. This is very, very important. Do not get confused between spermatogenesis and spermiogenesis. This is spermiogenesis, that is spermatogenesis. Later, the spermatozoa which are being formed, they will be moving out of the seminiferous tubule, and that is known as aviation, that is spermiation. Okay, that is movement of the sperm outside, or spermatozoa outside the seminiferous tubule. Works? Works, ma'am. Perfect. So always remember that until here it was 46 number of chromosome, but from here it becomes half the number of chromosome. From one spermatogonia, how many sperms? 4 sperms you get. Okay. Now we go on to the next question. Where's that thing? Okay, just a second. Yeah. Draw a labeled diagrammatic sectional view of human male reproductive system. Again, you have to draw the whole diagram up there. Make sure you do not do any mistake. Do you need my help? I'm so sorry for doing that, but yes, I have cold, right? So if you need my help, I can help you with that in some ways. So listen, first of all, you make the testis. The testis has testicular lobules. The testicular lobules will be having seminiferous tubules. The seminiferous tubules which are there, they are going to open into a duct. The first duct out there, which you call as rete testis. What do you call this as? Rete testis. Now rete testis says, oh my god, the sperms want to move out, let me exit, let me exit, let me exit. So who helps in that? It is vasa efferentia. E for exit, you can try to remember. It is trying to exit. These are vasa efferentia. But you know what? They are blocked over here because now you have the third duct out there, which is known as epididymis. This is actually 6-meter long duct out there, and this is where the maturation of sperm actually happens. Now from here you get a duct that goes into the body. So everything was happening in the scrotum until now, but now it goes into the body. What is this duct? This is known as vas deferens. So D for distance, it is traveling through distance. Where does it go, Baba? It coils around the urinary bladder. So this is going to be the urinary bladder, which looks like this. Okay, it coils around that, comes over here. It is going to receive a duct, an opening from a gland. This is known as seminal vesicle. Similarly, a seminal vesicle is on that particular side also. Now both of them unitedly are going to open, having a common duct. This duct's name is what? Ejaculatory duct. Ejaculatory duct. So this is what? A gland, accessory gland, you call it seminal vesicle. Now we have a duct. 2 of the ducts form and opens up over here. What is this particular thing opening? This is basically the urethra. This is the urethra. So ma'am, urine comes through this and sperm also? Yes, they have a common passage. Urethra is being covered by the male part, which you call as what, Baba? What do you call this particular part as? This is the penis. And over here you have the glans penis, the enlarged portion of the penis. And here you have a loose fold of skin, which is known as foreskin. Okay. Now, here I told you there is one gland. So there are 2 glands, 2 paired glands. You call it as seminal vesicle. Seminal vesicle. After that, there is a donut-shaped gland over here. Do not write donut shape, just remember it. It's single, it is not paired. You call this as prostrate. Prostrate gland. And then here you have again a paired gland, and that is known as bulbourethral gland. Bulbourethral gland. You also call this as the Cowper's gland. Okay, and that is exactly how. So this side and that side also you have to make and explain. Cool. Works. So yes, you had to mention these parts, so I have mentioned almost all of it, so you know that. Next thing, given below is a flowchart showcasing ovarian changes during menstrual cycle. Fill in the spaces with the hormonal factors. By the way, I have solved the questions in 2-marker, 3-marker session also, where I have explained about menstrual cycle. Watch those videos. Okay, so everything is linked. So obviously every single time when a video is being made, we cannot go on repeating things, otherwise the video will be too long, right? So which is why you need to understand some part of this chapter will be there in another video. Please refer to that video. Okay, and here I am going to talk about the next thing. So given below is a flowchart of ovarian changes during menstrual cycle. Fill in the spaces with the hormonal factor. Let's see. So this is for the menstrual cycle. Primary follicles which are there, they mature into Graafian follicle under the influence of A. So what is that that helps in the maturation of follicles? What is that hormone? Follicle stimulating hormone, right? It is follicle stimulating hormone that is going to help with this. So I'll just write it FSH. They help in the maturation. Now what? The Graafian, where is it go? The Graafian follicle which is there, it is going to rupture, and that is how you get the ova. So Graafian follicle and then ova is written. So you have to guess it is about rupturing of Graafian follicle to produce ova. Who helps in the rupturing of Graafian follicles? Ma'am, estrogen. No, Baba. Estrogen is going to tell LH. The LH is going to go on increasing basically because of estrogen. LH is going to have a spike. This LH spike is leading to what? Ovulation or the rupture of Graafian follicle. So I should be writing LH, right? Yes. So over here the answer for this question is going to be what? B is going to be LH, luteinizing hormone. Done. Now what do we have? Now you have to think, okay, I'm not going to tell the answer. You have the ova. Basically now it forms the corpus luteum. How does ova form the corpus luteum, ma'am? Imagine Graafian follicle which was there, it ruptured. Ova is being released, or basically secondary oocyte is being released. What is left behind? It is the corpus luteum. That is what you mean by this. Now the corpus luteum, arrow this, and it is written C. What is this thing which is released by the corpus luteum? What do you think is released by corpus luteum? It is a hormone that is known as progesterone, and progesterone will make the endometrium even more thickened. So secondary oocyte is being released. Any point fertilization can happen. Suppose fertilization happen, there should be someone to nourish the embryo. Who does that? Endometrium, which is why the endometrium has to be very thickened up and also should be secreting things, which is why you need progesterone. Progesterone majorly does it. So corpus luteum releases two hormones actually, progesterone and estrogen, but majorly progesterone. So you can write both progesterone and estrogen, but majorly please write this one is the major hormone which is going to play an important role over here. Works. Now, women experience two major events in their lifetime, one at menarche and one at menopause. Mention the characteristics of both the events. At menarche, what is the menarche is what? The start of the reproductive phase. It is the start of the reproductive phase, right? Menopause is when it's the stop, it's the end of the reproductive phase. So beyond that, she cannot reproduce and give fertile offsprings out there. So basically that is menopause. So write the characteristics of both the events. How do you write it? It's very simple. By the way, I have written everything which is needed for you to understand this particular question. And menarche, the first onset of menstruation at puberty, marks the beginning of reproductive phase in the case of females. You have to write that just all. Menopause is the permanent cessation of what? Menstruation, as in permanent stoppage of menstruation. It marks the end of reproductive phase, isn't it? Yes, ma'am. Done. That's all. That's all. Now, draw a sectional view of seminiferous tubules of human and label the following cells in the seminiferous tubules. So, sectional view of seminiferous tubule is something that you might have missed. Do not miss it and please know it. First of all, before decoding the sectional view, you should know these things also. Cells that divide by mitosis to increase their number. Which cells are there which increase their number in seminiferous tubule? During spermatogenesis, first one, spermatogonia, they increase their number, right? So can I write spermatogonia over here? Now these things which are being asked, you have to mark it there. So you have to write it first, then you have to mark it there. That is how you get the marks. Now cells that undergo meiosis 1. Primary spermatocyte, which is differentiated in form, it is the one that will undergo meiosis for the first time, right? So the one that undergoes meiosis 1 is primary spermatocyte. Okay. Cells that undergo meiosis 2, secondary spermatocyte, that is the one that will undergo meiosis 2 to form spermatids. So I write secondary spermatocyte. Okay. Cells that help in the process of spermiogenesis. Spermiogenesis is what? Spermatids to spermatozoa formation. And ma'am, what cells are there that helps in that spermiogenesis? It is not spermatids, not spermatozoa. Basically, the cells which are there, they are Sertoli cells. Remember, Sertoli cells are there for nourishment of the spermatogonia and the entire process which is going to take place there. It is the one that takes care of it. especially the last process, that is spermiogenesis. So under the influence of Sertoli cells, so you know what happens? FSH hormone is there. FSH, follicle stimulating hormone, is there. It acts on the Sertoli cells. Sertoli cells are going to release some factors. These factors are going to make sure that the hormones are enough for the whole process of spermatogenesis to happen. So for maintaining the hormones up over here, you need to make sure factors are being released. How do you get hormones? There is one more LH, luteinizing hormone. In the case of males, this is how it works. LH goes to act on Leydig cells. Leydig cells releases the male sex hormone, that is testosterone, or you broadly call them androgens. These androgens are now, this is happening inside the seminiferous tubule, this is outside. Okay, sorry. Sertoli cells and everything is there inside the seminiferous tubule, and this is happening outside. So androgens from outside comes inside the seminiferous tubule for the whole process to happen. But to make sure that their amount has been maintained over here, you need some factors. Who release those factors? Sertoli cells. Especially those are being needed in the process of spermiogenesis. So cells that help in the process of spermiogenesis are going to be what? Are going to be Sertoli cells. Why? Because they are the one that releases the factors which make sure that there is enough amount of testosterone over here. So can I write Sertoli cells? Yes. Do I need multiple understanding? Yes. I do need understanding of multiple concepts, and only I can write this answer. So please know that. Okay. Now cells produced due to spermiogenesis. Answer. Cells produced due to spermiogenesis. At the end of spermiogenesis, what do you get? Spermatids transform into spermatozoa. So cells produced due to spermiogenesis is what? Spermatozoa. It is spermatozoa. Got it? Yes. Now how do you draw this diagram? This is how you are going to draw it. So you are having spermatogonia right over here. The spermatogonia which is there, it is going to divide, going to form primary spermatocyte. The primary spermatocyte which are there, they are going to divide, form secondary spermatocyte. So here it's just cell which looks almost the same. Right? It looks almost the same. So you have to form multiple cells over here. In between that, you have to make other type of cells. These cells are what? Sertoli cells, that provides the nourishment and also produces the factors which make sure that the level of hormones are being maintained up over here. Now, towards the end, my dear children, you can show spermatozoa as well as spermatids. Spermatozoa and spermatids. Spermatids actually here in this picture has been mentioned like this only, but you can basically show a globular structure also. It's not a big problem. Okay, so please do that. But according to NCERT, whatever is given in NCERT, go according just to that. So yes, fine. When you write the answer, write here A, mark here B, C, whatever is there accordingly. So this was also a question that was given in your book. Now apart from it, there are a few other things. Menstrual cycle, as I told you, hormones involved in gestation, parturition, month by month, development data. All of this is super important. In 2 and 3 marker session, I have explained it. Go watch it. Go anyway, any chapter you have to watch the 1 marker as well as all 2 and 3 marker questions. Otherwise, there are chances that you might miss out on concepts. So do not do that mistake. Okay? So I'm not explaining it here. Otherwise, it will take too much of time, right? And it will be repetition. So yeah. Now what do we have? Reproductive health. This chapter have 5 marker questions, Baba. No, there are no 5 marker questions, only 1, 2 and 3. So it's already been covered in the 1 marker and 2, 3 marker session. Now what do we have? Molecular basis of inheritance, and between that you have principles of inheritance. We'll do it after this chapter. So here also you have some 5 marker questions coming in. Let's see those. Mama. Okay. Children, I hope you guys are understanding. As I said, my energy is a bit low, which is why I'm not able to, tell some jokes and things like that. But I hope that you are really getting engaged, you're understanding things well. Anyways, exam time, so you have to pay attention, right? So yeah, that's all. Let's go ahead. DNA is genetic material in both prokaryotes and eukaryotes. Yet the packaging process in their cells is not the same. Explain why. DNA is a genetic material in both prokaryotes and eukaryotes. Is it? Yes, Baba. In prokaryotes as in bacteria. Okay, the primitive organism. Eukaryotes as in us. In us we have DNA, they also have DNA. But one very important difference that you always need to remember is that in them, the number of the amount of genetic material which is there, it is going to be very less. For example, Escherichia coli 4.6 into 10 to the power 6 base pairs. 4.6 into 10 to the power 6 base pairs. Our case 6.6 into the power 9 base pairs. So we have too much things in here with us. We have less genomic content. That is one very important thing, which is why packaging can be pretty much easier in them and packaging for us. Listen, why it is difficult for us? Because we have a cell, inside the cell we have nucleus, inside the nucleus this DNA has to be fitted in. And when I say DNA has to be fitted in, our genomic size is more. It is almost 2 meters long. Can I fit this 2 meter long DNA inside the small nucleus? It's very difficult. Which is why it is difficult in us. But in the case of prokaryotes, do they have a nucleus? No. They do not have any nucleus. Their genetic material is going to lie around in the cytoplasm itself. There's a specific region which is there, but is not membrane bound and things like that. Which is why it does not have to be made too compact. So first thing is that prokaryotes do not have large genomic size. Second thing is that they do not have membrane bound organelles such as nucleus and things like that. Got it? Yes. So DNA is genetic material in both prokaryotes and eukaryotes, but its packaging differs due to difference in genome size and also the cell organization. First point. Now, in prokaryotes, DNA is naked, circular and present in the nuclear region, the open region which is there, specifically where genetic material is there, you call this as nuclear region. Do you think they have histone proteins? No. Histone proteins are needed whenever you want to wrap around the DNA such that it becomes compact in nature. So here there is no need of histone. In prokaryotes, no histones are there. And because of small genome and absence of a nucleus, you do not need that. Now in eukaryotes, DNA is associated with histones, okay, and almost 200. So DNA wraps around the histone protein. So 200 base pairs of DNA are being covered in this only. This whole structure which is being formed is known as nucleosome. Okay, DNA wraps around histone octamer. 8 histone proteins will be coming up, and that's how DNA is going to wrap around. This whole structure is known as nucleosome out there. These nucleosomes, they coil further, so you will have multiple nucleosomes. So you might be able to see something like this popping up. Here you have nucleosomes, and basically the DNA will be wrapped around like this. It shows like a bead on a string like structure. There might be beads on a string. Basically it is nothing but the chromatin. The chromatin will later condense to form the chromosome. Everything, the chromatin, chromosome, whatever you're talking about is nothing but the DNA and the proteins. It's just the DNA and the proteins. Always remember that. So yes, these nucleosomes coil further to form the chromatin, which finally condenses to form the chromosome. So the large amount of DNA can be efficiently packed inside the nucleus. That is why both of these things are different. I hope that's clear. Yes, this is your model question paper 1 question. Okay, please know the structure of nucleosome. This is very, very important in 2 and 3 marker. I have explained it. Yeah, fine. Now explain Griffith's transforming principle experiment. How do you explain Griffith's transforming principle experiment? You might be knowing about it. Please remember, I have told Griffith's transforming principle experiment over here. That does not mean you should not be looking for Avery's experiment and Hershey and Chase. Hershey and Chase could be coming in, we never know. So please know about these 3 experiments. Here just Griffith's experiment is being talked about. Okay, so what exactly is being done is that one strain of a bacteria is being taken. What do you call this bacteria as? Streptococcus pneumoniae. It causes pneumonia. Okay, now this Streptococcus pneumoniae which is there, it has 2 strains, one a rough strain and one smooth strain. The rough strain which is there, it is a plain strain out there. Smooth strain which is there, it has an extra covering around it, which is made up of polysaccharide, as in carbohydrates basically. It is mucus polysaccharide, which makes it or gives it a smooth appearance. Now, this guy, since it has an extra covering around it, it is the demon, or I'll say it is the one that causes death. It is the virulent form, the one that causes death in the organism. If someone is infected with this, it will be non-virulent, which is why those organisms can be easily living. So over here in Griffith's transforming experiment, what was being done that first thing that he did is he took a live R strain, as in he took an R strain, live one, and he put it inside the mice. I call it as Puchu. My students might know it. So inside Puchu, when it was put, Puchu lived, my dear children. Why? Because R strain is non-virulent, does not cause any problem. Okay. Then S strain was taken and then it was put inside the mouse. What happened? Puchu died. Why? Because it was the virulent form. Okay. Then what did he do? He treated S strain with heat. He treated S strain with heat, and obviously that will be bringing some changes. And then he put inside Puchu. Puchu was so happy. Puchu didn't die. Okay, it didn't die. Okay, so then Griffith was like, okay, when I put live R strain, it didn't die. When I put heat killed S strain, then also it didn't die. Why not I put a combo of it? Let's see what happens then. He put that, and to his surprise, he found that Puchu died. And one more thing was noticed. He put live R strain and heat killed S strain. But inside Puchu, he found live S strain. Did he ever put live S strain? No. So basically something might have been coming from here into the R strain and might be transforming this live R strain into what? S strain inside the organism. So there is something that is getting transferred, and that something which was getting transferred could be the genetic material, because when that thing was there, it is giving information to produce something that will lead to the death of the organism. So this thing that was getting transferred was considered to be the genetic material, but he didn't know whether it was DNA, RNA, protein that was genetic material, which is why Avery, MacLeod, McCarthy came into the picture. They proved it partially, okay, but then people didn't believe it. Then Hershey and Chase came with their experiment on bacteriophage and E. coli, and then they proved it. Please know, these two experiments are equally important. Please go through it. If you have any doubts, ask me. I'll make a separate video also. I do not mind it. So please do that. Okay. So this is what you have to write. The live R strain bacteria were transformed into virulent S strain. Griffith concluded some heritable material was transferred from S strain to R strain. So please know about that. Works? Works. Fine. Man knows. Okay. Now state the arrangement of different genes of lactose operon in E. coli, lac-operon, and describe the role of lactose in regulation of lac-operon. Now this is also super simple. Listen to me. So first thing, so I'll explain with this diagram. That will save a lot of your time. So there is a gene over here which you call as inhibitory gene. What do you call this? Inhibitory gene. This gene is a regulatory gene. Okay, so this position you have a regulatory gene. It regulates. It can regulate positively, negatively. But over here, since it is going to regulate negatively, I'll call it as inhibitory gene. This inhibitory gene has a promoter for itself. Onto this promoter, when RNA polymerase will come and bind, what is going to happen? Transcription will be happening, and whatever mRNA will be found has to be found. And here after translation, you get the protein. What is this protein? Repressor protein. What protein? Repressor protein. Okay, done. Until here clear. Now you have another set of gene over here. For that you have a promoter region, and you have one more region which is known as operator. O for operator. This type of setup is usually found in prokaryotes. Okay. And over here you have multiple genes. Z, Y, A. I remember it as Zyada. Zyada means more. So Z, Y, A, that is the flow of things. Z basically, which is there, it codes for an enzyme that is known as beta-galactosidase. Ma'am, what does this enzyme do? So E. coli, suppose E. coli needs some energy. What do we do when we need some energy? We drink some juice and things like that. We get carbs, right? Glucose basically. Similarly, when this organism E. coli needs energy, it has to take either glucose or basically something that could give glucose. So over here, suppose it has lactose in the medium where it is growing. It takes up the lactose and breaks down the lactose with the help of beta-galactosidase into what? Glucose and galactose. So lactose is made up of glucose and galactose. So when I break down lactose, I get glucose. You get it? So that is the use of beta-galactosidase. Now, first of all, lactose is outside in the medium. It has to be taken inside. For that, you have to increase the permeability of the cell, which is why you have permease enzyme. When this enzyme is produced, the permeability increases and you have what? More lactose. Now, there is one more enzyme that is known as transacetylase. It's not this, it is transacetylase. Transacetylase deals with everything toxic that comes in between. Obviously, when you are doing so many things, there are chances there are some toxic buildup happening up there. So, deal with that. So, we have transacetylase. Okay, these are the three genes which is there. Z, Y, A. What is this? Beta-galactosidase. What is this? Permease. And what is this? This is transacetylase. Okay. Now, suppose I am growing the organism, that is E. coli, in a medium that does not have lactose. Obviously, do you need all these enzymes as of now? No. So, do you think transcription should be happening? No. Which is exactly why the repressor protein over here will be going and binding on the operator. Now suppose this is my hand and this is RNA polymerase. RNA polymerase will come bind over here to do the transcription. But do you think it will be able to do the transcription? Why? Because this guy is up over here, the operator is up over here. Sorry, repressor is on the operator, which is why there is no possibility of transcription to be done, which is why all these things are not going to be formed. In this condition, I will say it is switched off, lack operon. This is lack operon, and it is what? In switched off condition. What type of regulation is this? Negative regulation. This is negatively regulating it, right? So this is negative regulation that is happening up over here. Now suppose I have changed the medium and the medium has lactose in it. What is going to happen? The lactose slowly will be getting inside the cell. Once the lactose gets inside the cell, it says to the repressor protein, hey Macha, don't go and bind there, da baba. You come here, you bind with me, because they both are best friends. So always remember, the repressor protein which should be binding up there, it's now free, it's not binding there. Rather, the lactose protein is coming, sorry, not lactose protein, lactose is not a protein, lactose is a carb. Okay, the lactose which is there, that is binding up with the repressor out there. In this condition, what is going to happen? Transcription will smoothly happen because over here there is no blockage. Which is why I'll say that lactose which has just got in, it is what? An inducer. It is what? An inducer. It is helping in transcription to happen, right? So lactose is an inducer, always remember that. Apart from lactose, you have different form of lactose. There is allolactose that can also act like an inducer. Please remember that. Okay, done. So this is an inducer. Now what happens? Beta carotene is being formed, permease is being formed, transcytase is formed. When permease is being formed, it increases the permeability, more and more lactose will be coming in. But ma'am, there's a question, how did the first lactose come in? So it's, when I say that it's switched off condition, it's not completely switched off. Little amount of permease will be there. Okay, little amount of transcription will be happening. It's a general thing. In your NCERT it might not be written, but please know that. Okay, so please know all about this. Please know the flow of this. Very, very important. Okay, Piposia, whatever you want to remember, you can remember. Piposia. Okay, very, very important. Then this is when I say it's switched on condition. And remember, in your NCERT it is written lack operon shows negative regulation. There is positive regulation also, but it is beyond as to what is to be taught over here, which is why it is not. Is what NCERT also says. Do not worry. Okay, it's not just me, it's also NCERT. Works? Works, ma'am. Perfect. So that is being mentioned over here. Write your answers properly. I've given you all the answers, how to frame your answers. Just know about it. Okay. Now, another 5-marker question, that is, given below are sequence of nucleotides in a particular mRNA and amino acids coded by it. Okay. 5 AUG, UUU, UUC, GAG, UUA, GUG, UAA. And methionine, phenylalanine. Okay, so AUG codes for methionine, UUU phenylalanine, UUC also phenylalanine. So you are being asked what are the properties of genetic code that can be correlated from the given data. So what are the properties that you can understand from this? First thing, AUG which is there, it is what? Going to be an initiator codon. It is the one that initiates the process of translation such that amino acids are being coming up. And apart from being initiator codon, it also codes for an amino acid that is known as methionine. So can I say AUG which is there, it has what? It has a dual function. It has a dual function. This is the first thing that I can write. Now, if I see UUU, UUC, 2 codons are there coding for the same amino acid. What do I call this particular feature? Multiple codons coding for same amino acid. This feature is known as degenerate feature. This is known as degenerate feature when multiple codons code for the same amino acid. Now, do you see any full stop, commas, anything up there over here? No. So can I say that it is read in a contiguous fashion, as in a continuous fashion? That is what I mean by contiguous fashion. Okay. Now the fourth thing that is up there. What is the fourth thing? Any idea about the fourth thing? If you see, each codon codes for only one amino acid, right? GAG is not coding for anything else, just glucine. Oh, sorry, glutamine, not glucine, glutamine. Okay, or basically over here, just a second, okay? It is glutamic acid. Okay, now we're coming to this. UUA is also coding for leucine. So it goes like that. What do you call this feature when an amino acid is when a codon is coding for a single amino acid? This is known as unambiguous nature of codons. Unambiguous nature of codon. What do you mean by that? One codon codes for only one amino acid. Listen, multiple codons can code for one amino acid, but one codon is definitely coding for one amino acid. That is known as unambiguous nature of the codon. Now, what feature do you have? If at all you see this, UAA is not coding for anything. So what is this codon? This is basically, and after this there is no amino acid also being formed. So can I say UAA is a stop codon? Yes, I can say that. UAA is what? A stop codon. It does not allow translation to further go up beyond that. Okay, so it's a stop codon. Apart from UAA, do we have another codons? Are there which are stop codons? Yes. So UA, UGA and UAG. Okay, UA, UGA and UAG. These are the few things that are being formed. Always remember that. Okay, so, and one more very important thing. If at all you see all the codons which are there, they are what? Triplet in nature. They are triplet codons. They come in a number of 3 groups. Okay, so the codon is triplet. 61 out. We have 64 codons, by the way. After that, 61 of them are going to code for amino acid. 3 of them, UAA, UGA and UAG are stop codons. So know that. AUG has dual function, know that. Some amino acids are coded by more than one codon, hence it is denaturated, correct. The codon is read in mRNA in contiguous fashion, correct. UA, UAG and UGA terminator codons, correct. Now, it is not given over here, but still I am telling you. The codons which are there, they are going to code for the same amino acids all throughout all different type of organisms out there. This is known as universal feature of the codons. If at all you get a question that this codon is coding this amino acid in bacteria and the same amino acid in human beings, what feature does it show? It shows universality of this particular codons. So they are universal. Exceptions are always there. Exception is what? The mitochondrial codons and some protozoans. So in mitochondria also you have codons. There you see a different whole thing happening. Similarly in protozoans also you see a different thing happening. Okay? Okay, perfect. Okay. Next thing that we have is a police plan from the crime department approached the forensic science department to help identify the culprit. DNA samples were collected from the crime site and of the suspects. Explain the crime site and of the 2 suspects out there. Explain the process involved if the culprit has a monozygotic twin. Can this method give results? Okay, first of all, let's go line by line. Explain the process involved. If at all there is something happening, suppose there is a crime that has taken place over there. I am the criminal. I did the crime. You are also there. I said that you did the crime. I said that you did the crime. Let me name you. Your name is Raghav. Okay, your name is Raghav. Any Raghavs up over here? Please let me know. Okay. So over here, me and Raghav are the suspects. Okay. And from the crime site, basically from the crime site, we got some blood. This was being taken. This was being taken and it was assumed that it is of what? The murderer. That is me. But as of now, no one's know that. So basically, the DNA was being checked out. Okay. And they made a complementary strand of it. What did they do? They made a complementary strand of it and they added radioactive thing over here. This complementary strand is known as what? A probe. It is known as a probe. This was already been done and is kept there. Now, you have a doubt between, the police had a doubt between me and Raghav. So basically, first of all, our DNA was isolated. So I'll write isolation of DNA. That was being done. After isolation of our DNA, using restriction enzymes, our DNA was being cut. Using restriction enzymes, the DNA was being cut. The same restriction enzyme was used over here also, if at all cutting was there. Done? Done. Now after that, using gel electrophoresis, our DNA strands were separated. Okay, so now I'm going to make the scenario. It looks like this. Suppose this is me and this is Raghav. This is the DNA that we have, the DNA bands that are being formed, okay? Cool. Similarly, this DNA from the murder site also, from here also, I took the DNA, I cut it with the same restriction enzyme, I made them separate. Okay, listen, this and this exactly matches, but as of now, I cannot prove it. Listen to me carefully. What has to be done next? So gel electrophoresis was been done. After that, you have to transfer it onto a nitrocellulose sheet. You have to transfer it onto a sheet. So this is nitrocellulose sheet. That is known as southern blotting. You call this as southern blotting. After you transfer it up, what do? You basically put the radioactive probes. This probe which was there, it is being put over there. Radioactive probe. And this process is known as hybridization. Why? Because you are putting a probe and it will bind to the complementary base pairs out there. So what exactly happens is that, listen to me carefully. The probe goes and binds up over here. The probe goes and binds up over here. It binds up over here and it will also bind up over here. You might ask why? Listen, we all have some repetitive sequences. There are some sequences which repeats in our body. In you, it might be repeating in different number of times. In me, it might be repeating different number of times. Does not mean that we do not have any sequence which is not similar. We will have it. But over here, if at all you see that, if I have a quick comparison between this and this, it is exactly similar. Both of them looks exactly similar, the bands. Here it is different. With that, what I get to understand is that probably the murderer is jasmine itself. Murderer is what? Jasmine itself. But again, you put the radioactive probes. After that, you need to do one more thing. That is, you need to view it under an autoradiography machine or UV light thing. So please know that also. Autoradiography thing or UV light. So DNA fingerprinting works on that. That is, we have unique DNA out there. Okay, we have some unique DNA out there which makes us all different. And basically with the help of that we can find out the culprits. We can find out if at all there is who is the father, who is the mother and things like that as well. So please know about that. And for this particular process, you will require things such as blood, hair follicle, skin, bone, saliva or sperm, etc. These are the specimens that is generally being used. And also remember, in the blood, RBC is of no use. In blood is made up of multiple cells, right? Red blood cells are of no use because red blood cells does not have nucleus. That means no DNA as well. Please remember that. So process involved is DNA fingerprinting. I hope you know that. Here what do? Isolation of the DNA. Then digestion by restriction enzymes, you cut it, separation by gel electrophoresis, transfer to the membrane or southern blotting, hybridization with the DNA probes, and visualization. So how do you get to know that this probe is there? Basically, you made it from the DNA that you got from the site, right? That is why it is going and matching up there. So yes, got it? I hope you have got it. Each step you have to write. Now, there's one more part of this question. If the culprit had a monozygotic twin, what do you mean by monozygotic twin? As in the ones that look similar, very similar, right? So basically when they look similar, that means inside that the DNA is also very much similar. Why? Because there was a zygote, this particular zygote split into two and they develop into two embryos. So whatever genetic material was here, it's the same over here also. So in this particular case, do you think DNA fingerprinting will be of any use? No. Because both the twins will be having same type of genetic material. It will be very difficult to find out. So if the culprit had a monozygotic twin, no. No. Why? Because DNA fingerprinting cannot distinguish between monozygotic twins. This is because monozygotic twins have identical DNA sequences. Hence, their DNA fingerprints are exactly the same. There is one more type of twin where basically two zygotes. So basically in that particular case, you might have seen those people who do not look similar, right? So in that particular case, ovum was released from both the ovary. Okay, sperm came and fused both the ovaries, two sperms, separate here. Okay, now you have got one zygote here, one zygote here. So that is individual zygotes that are there. Over here, only one zygote. In monozygote twin case, only one zygote is there. This zygote itself divides into two. You get it? So that is the difference that is there. So please know the difference and answer accordingly. Okay. Now, I hope this type of questions are helping you well. You feel that, okay, I'm practicing some new type of questions as well. Yeah? Do let me know. I'll be really happy if at all I get to know that it's helping you. Now, answer the following. Construct a complete transcription unit with promoter and terminator on the basis of the hypothetical template strand. So what is given over here? This is going to be the template strand. Please read your question really well. Now, write the RNA strand transcribed from the above transcription unit along with the polarity. Okay. So first thing that you have to do is make a template strand. So make a transcription unit. So just realize whenever you have arrow on this side, it means that it is a 3' end. Okay. Okay, so where was I? Yeah, I'm so sorry. As I said, I'm not feeling well. Yeah, so this is 5' end and this is 3' end. So wherever you have arrow, that is 3' end. This is going to be the template strand, right? Now, I'm just making a rough diagram. Do not judge me, okay? 3' and 5' end. If this is my template strand, I would have to make a coding strand as well. That will be in the opposite polarity. So this will be 5' and that will be 3'. This is going to be what? My coding strand. Now listen. At the 5' under the coding strand, you have a sequence that is known as promoter region, onto which RNA polymerase is coming and binding and doing the process of transcription. Here onto the 3' end of the coding strand, you have the terminators region. In between, you have to fit all this in. So in between, in the 3' end, what do you have? A, T, G, C, and the list goes on. How do you make a complementary strand? A base pairs with what, Baba? A base pairs with T, T base pairs with A, G base pairs with C, and you have to make a strand like that. That is going to be a coding strand. This is how your transcription unit is going to look. Make sure you make the polarity really well. This is super important. And read the question. Here it is given, hypothetical template strand is given to you. If hypothetical coding strand is given to you, thing will be different. What will be your answer if at all coding strand is being mentioned over here? Think for that as well. Okay, just know the scenarios. Now, this is a transcription unit. What do you have to make? You have to make an RNA thing that is transcribed. So basically, which one is going to undergo transcription? Template strand is the one that undergoes transcription, right? So you have to make a complementary RNA strand. Do you make it again from this strand? Listen, you just have to replace wherever T is there, you have to replace U, because you're making an RNA and it will be complementary RNA, right? So wherever T is there, you will have U out there in the new RNA strand. So this is how it is going to look. Do not be worried. If at all you're confused, come on, Baba. Yeah, this is a promoter, that is a terminator and everything. Here you have the new RNA strand being made. So if at all you see this, it was TAC here. You had UAC. So wherever T is there, you're replacing it by U. That is the only difference out there. And when you make a new RNA strand, it has to be from 5' to 3' direction. And you will always use the template strand for that. Coding strand is not going to get transcribed. Remember it. Else works. Perfect. Now list the salient features of human genome. This is being asked usually. So please know the salient features of human genome. The human genome contains how many base pairs? 3,164.7 million base pairs out there. 3,164.7 million base pairs. The average gene size consists of what? 3,000. A gene, if at all you see, it is having 3,000 base pairs. But there is an exception, and that is dystrophin. It has 2.4 million base pairs. Everything is in million over here. Now, total number of genes is what? Estimated to be 30,000. How was the previous estimate? Previous estimate is what? 80,000 to 1,40,000, but it is not, it's just 30,000. Now, the functions are unknown for over 50% of the discovered genes. For 50%, okay, for over 50% of the discovered genes, we do not know their functions. Their functions is not known. Less than 2% of the genome codes for protein. When I say, okay, I have discovered many genes, say 30,000 genes I have discovered. For 50% of them, I do not know what is their function. And not just that, when I talk about the whole genome, only less than 2%, okay, it's not more than, please remember it is less than, these terms are important. Less than 2% of gene actually codes for what? Protein, know that. Now, repeated sequence make up very large portions of the human genome. That is why we say less than this codes for protein, rest everything is what? Could be repeated sequences. What are these repeated sequences? They are stretches of DNA that are repeated many times, 100 to thousands. They thought to have no direct functions, but they definitely helps us in understanding the evolution pattern which is there. Okay, they helps us in understanding evolution pattern. Now, so over here, okay, so they do not have any direct coding function, but they shed light on chromosome structure, dynamics and evolution. Chromosome structure, dynamics and evolution. Now, one more point is there. Chromosome number 1 has the most number of genes. What is the most number of genes? 2968. So over here only 7 is being mentioned. 2968. Here do not see 7, just everything is there. Okay. Chromosome number 1 has the highest number of genes. That is the first one to be sequenced also. And chromosome number Y, that is found in the case of males, XY, Y gene. Okay, sorry, Y chromosome which is there. It has the least number of genes, and that is 231. Do not go and write 213. It is 231. So here it is 2968. There it is going to be 231. Very, very important. Now, scientists have identified 1.4 million locations where single base DNA difference is there. That is single nucleotide polymorphism is what you say is that. Okay, so over here 1.4 million locations is where you find it. These are the very important things that you need to know. This is the salient features of human genome. Works? Works. Next thing. Explain the process of translation in protein synthesis. So what is translation, Baba? By the way, let me just see how many questions are left. So are we soon going to finish it up? Yes. Okay. Explain the process of translation in the protein synthesis. How do we explain that? First of all, for translation to happen, what do you need? You need an mRNA. mRNA is in which direction? 5' to 3' direction. Okay. Now what do you need? mRNA which is there, it will be having codons. AUG and the list goes on. You need anticodons. From where do you get that? From a tRNA. So you need tRNA also. Okay, tRNA has an anticodon loop, and basically it will be having amino acid attached to it. Fine. And what do you need? You will need, apart from this, you will need ribosome. Because ribosome provides the site where tRNA will be coming and binding. So it looks like this. Your mRNA is between two ribosomal units. The tRNA will be coming and binding in the specific sites of these units and helping in the whole process to happen. The first thing that needs to be done over here is this, is known as charging of tRNA. What do you mean by that? It means that tRNA has a 5' end and a 3' end. Okay, it's a RNA which is a 5' end and 3' end. At the 3' end, the amino acid will be coming and binding. This process is known as charging of tRNA. It is also known as amino acylation. What do you call it? It is known as charging of tRNA, or it is also known as amino acylation. This is one very, very, very important thing that you need to remember. Now what? Now there is another thing that is taking place over here. There is anticodon which has also been prepared. Please remember the secondary structure of tRNA. We thought it is like a clover leaf model, but actually it is not. Okay, it is an inverted L. It looks like an inverted L. And also tRNA is known as soluble RNA, which is why sRNA is also what you call it. Fine. So this charging of tRNA, amino acylation, requires a lot of ATP. Remember that also. So this has already been prepared. Every tRNA which is there, it has an amino acid joined to it. Charging of tRNA, this has to occur in the first place, in the first phase. This is not first step, I say it's the first phase. In the first phase of translation, this has to be already done and kept. Now what? So now we have the mRNA. Onto this mRNA, one smaller ribosomal subunit comes and attaches. Small ribosomal subunit. Then you have the AUG sequence, initiative codon sequence. Over here, the tRNA will come and attach. Then what? Then the next ribosome, the larger ribosome will also come and attach over here. Larger subunit of the ribosome comes and attaches. And now we say that the initiator complex have been found. The ribosome which is there, always remember it is made up of rRNA, ribosomal RNA. Apart from that, there are 80 proteins which are being involved. 80 proteins are also going to make up this ribosomes are there. So first the small ribosome attached to the mRNA, then the tRNA, then basically the larger subunit attached. Now we say that initiator complex is being set up. So we will read until this point so that you have an understanding. First is activation or charging of tRNA. Amino acylation of tRNA is the process of attaching an amino acid to the corresponding tRNA. This is known as charging of tRNA. It occurs in the first phase. Next thing, initiation. This is the first phase. Now we are going to begin up. So in initiation, mRNA binds to the smaller ribosome subunit. Okay. The start codon is recognized by the initiated tRNA, the first tRNA out there, carrying what? Methionine. The larger ribosomal subunit assembles, forming the initiation complex. Done. Now what happens, my dear children? You have formed the ribosome. Slowly the ribosome is going to move. So until it had, see, each tRNA has an amino acid, right? Suppose a new tRNA is coming. What is going to happen? These two amino acids which are there, they are going to join, and this bond is going to break, which means that this tRNA is free to go, and you have a new chain of amino acid being formed. So same thing is going to be repeated again and again until a terminator RNA, that is basically your UAA, the stop codons, anything is being reached. Until that particular point, this will be going in. But you should know that what is that something that will be helping in the formation of bonds between these amino acid, right? So that is the next thing that we are going to see. So elongation. tRNA brings amino acid to the ribosome, matching codons on the mRNA through its anticodon. Okay. Peptide bond, that is the bond between two amino acid. Peptide bond forms between the adjacent amino acid, catalyzed by the enzyme. Okay, you do not have to write the name of the enzyme, just know it is catalyzed by the enzymes out there. Now, the ribosome also acts like a catalyst. The ribosome as a whole, it also acts like a catalyst. Why, my dear children? Because it has, as I told you, it is made up of rRNA, and there is one type of rRNA which is called as 23S rRNA. 23S rRNA. This is there in the bacteria. It is basically acting like a enzyme which you call as ribozyme. So this particular thing also helps in the formation of peptide bond. In the case of what? In the case of bacteria, the rRNA. Okay, for the formation of peptide bond. So generally, peptide bond is formed by the enzymes which are present. You know, multiple proteins are present in the ribosomes. So yeah, out of that, anything can help in the formation of peptide bond formation. In the case of bacteria, you have 23S rRNA, which you also call as ribozyme, helps in the formation of peptide bonds. Now, ribosome moves along the mRNA in 5' to 3' direction. In which direction does it move? From 5' to 3' direction, it is going to move. Please have an understanding of that also. Got it? Perfect. Now what? Termination. When a stop codon, that is UAA, UAG, UGA is reached, at that particular point of time, no tRNA will come and bind over there. Release factors might come and bind, but no tRNA comes and binds over there, and that's exactly how you got a polypeptide. Got it? Yes, that's all. Those are the things that you need to remember. Now, one more thing which is there is that this is an mRNA. Before the 5'N, before the 5'N, and after the stop codon, before the initiator codon, after the stop codon, or after the 3'N, you have some regions which are known as UTR, untranslated regions. They are not going to undergo translation, but they are super important. Why? For the stability of the mRNA, for the initiation process to happen very smoothly, for everything, for the process of termination to happen smoothly, you need UTR. Untranslated regions, they are not going to get translated. Works? Works. Perfect. Now, question number 9, which is DNA replication is said to be semi-conservative. Why? Describe the experimental proof of Measles and Install to show DNA replication is semi-conservative. First of all, Measles and Install did their experiment on which organism? E. coli. Okay, it was E. coli. So, E. coli, my dear children, was grown in a medium which had N15. So over here, the two actors which are there, one is N15 and one is N14. Nitrogen basically is what? Is a part of our DNA. Yeah, nitrogen is basically there in our DNA. So nitrogen has been taken up. If at all I grow an E. coli in a medium which is having nitrogen, that nitrogen will be taken up for the formation of DNA. Okay. So there was a medium whereas NH4Cl is used where the N can be N15 or N14. And over here, what do I do is, I have a logic. The logic goes like this. What I am trying to prove is that if I say this is parental DNA, the parental DNA is going to separate out and a new strand is going to be formed for sure. But will the old strand be also there? Yes. Only half of the old strand is there, which is why I call this as semi-conservative mode of replication. And I am trying to prove this. Not me, actually Measles and Install tried to prove this. How did they prove this? So basically, they grew the E. coli in a medium that was containing N15. Now, N15 will be taken up for forming the DNA, and N15 isotope is a heavy version of isotope. So what is going to happen is that when I put this DNA in cesium chloride medium, in a medium that is cesium chloride, CaCl medium, the DNA is going to come somewhere over here. They move down, they segregate somewhere over here. Why? Because they are heavy. They are very heavy. That is why they move over here. So I get a heavy chain. Okay. Now basically I change the medium. I grow the organism in N14 medium. What is going to happen? And every E. coli is going to divide in every 20 minutes. So after 20 minutes, I saw that there was no lower band with heavy band. There was actually a hybrid band. How? Listen to me. So these two strands which are there, red in color, they separated. The new strands formed up. The new strand was N14. The older strand was what? N15. Which is why now it's not like this. Rather, it's like this. This is non-parental type. Do they look like the parental type? No, they look different. And similarly, their heaviness is also different. Over here, they're somewhere in the middle. So I got an understanding, but still I want to see once more. Again, I let the E. coli divide. So 20 more minutes. So 20 plus 20, I have 40 minutes now. Every cycle is of 20 minutes. What did I find? These two strands are going to separate one green strand, one red strand. Similarly, one red and one green. New strands are going to be formed, but what strand? N14 only, because I'm still growing in N14 medium. Now what you will find? You will be finding a heavy chain, a hybrid chain. Apart from that, you will be finding a light one. So N15 was a light one, N14 was a light one, which is why you will be finding a light chain also. So now they do they have any parental thing? If at all I compare with this, yes. But if at all I compare with this, no. In the last generation, I have nothing that resembles like the parent. Okay. Is the density different? Density is also different over here. So please know that. So with this conclusion, with this understanding, got to know that, yes, it is. Since he found hybrid variety every single time, he got an understanding that, yes, parental, DNA is going to be there. One strand, only one strand of the parental DNA is at least going to be there. Otherwise, you will not find hybrid, no. You'll only find the new ones and only the light ones. You have the hybrid for a reason. Got it? Yes. Now, similar type of experiment was done by Taylor and his colleagues. But they did not use nitrogen, rather they used radioactive thymidine. Please know that. They used radioactive thymidine over here. And just a second. Yeah. Work. Come on. Okay, there's some problem. I'll fix it. So radioactive thymidine was used, and the plant which was used was faba beans. You also call it as Vishya faba. Please know about that also. Okay, generic thing. Fine. Now you have the next question out there, and I think it's time to fix the... Okay, next question, it went. Okay, and it goes like this. Name the process involved in the following. So you have to tell the process name which is involved in the following. One parental DNA strand is conserved by the daughter cell. Semi-conservative mode of replication, right? Only one strand is there, other strand is new. Separation of DNA fragments. With what process did you do separation of DNA fragments? Gel electrophoresis. Yes, with that process you do separation of DNA. Now removal of introns in the hnRNA. hnRNA means what? When I say transcription is happening, DNA transcribes into hnRNA. So whatever amap shanap is there in the DNA, it's transcribed over here. So there might be coding regions, exons, and there might be non-coding regions, introns. Everything will be there here also. But when you say, I am going to form an mRNA which will undergo translation, I have to make sure there are no introns. So if introns were over here, I have to remove these introns with the help of splicing. This process is known as splicing. Now, attachment of Rho factor in to RNA polymerase during transcription. What do you call that? So listen, transcription, you have terminator and a promoter region, right? So in bacteria, for attaching to the promoter RNA and for starting the process of initiation, you need a factor that is known as sigma factor. Similarly, for termination, you need another factor that is known as Rho factor. And you call this process as Rho dependent termination. Okay, Rho dependent termination. That is what you call this process. Now, polymerization of amino acids to form polypeptide on mRNA. On mRNA, when you form a polypeptide, what do you call this as? You call this as translation. So this is exactly written over here. This was the question, so I thought I should be putting it over here so that you can also get a practice. Okay, so please remember, go through each and everything, very important. Okay, now what do we have? Okay, other important concepts which are there, which you should not be missing. As I said, Hershey and Chase, different type of RNA polymerase, there are 3 of them. Please know about that. I have explained previously. and genetic content in different organism. Again, in 1 marker you will be able to see this. In 2 marker you will be able to see this. In 1 marker you will be able to see this. Hershey and Chase experiment I have done in master stroke. But yes, again, if you need some help regarding that, I can help. Okay, do let me know about that. I'll do help you. Yeah? In a separate video, I can do that. So until now, are you facing any trouble? Is it okay? Are you understanding? If at all you are understanding, please let me know. Okay, if you're not understanding, please let me know that also. That is very much important. Okay, I'll just drink some water and I'll be right back. Okay, dear children, I would have still fallen. I'm sorry. Yes. So, yeah, we were here. Principles of inheritance and variation. Let's go. So, 2 and 5 marker questions are there. 2 marker I have already discussed, and the 5 marker you will be seeing over here. First question that we have is, T.H. Morgan selected fruit flies for his genetic experiment. Why? Why did he take fruit flies for his experiment? First of all, they were very easy to grow. Second thing, they had a very short lifespan. So, if at all I want to observe some changes, I just have to wait for a few weeks. Okay, just 2 weeks, because in 2 weeks the lifespan is going to end up. Next thing, a single mating will be producing too many progenies, so basically that was also helpful. Next thing, there's a clear differentiation between male and females. It was very easily distinguishable, so that is also one very important reason. So these are the things, please write it down somewhere if you forget. Now, what are chromosomal disorders? Give an example. So listen. Chromosomal disorders are one and Mendelian disorders are the other one. Mendelian disorders basically means what? Suppose I have a chromosome over here. This chromosome and gene is there. This gene has some mutation, which is why the proteins are not being made properly. This is Mendelian disorder. This leads to Mendelian disorder. Chromosomal disorder means I needed to have 2 chromosomes, but I have 1 more chromosome. Extra things being made. Or probably I need to have 2 chromosomes, but I have only 1 chromosome. Chromosomal disorder. Suppose I have only half of it, other half is not there, leading to what? Chromosomal disorders. These are the things which comes under chromosomal disorders. So the disorder in human beings occurs due to absence or excess or abnormal arrangement of 1 or more chromosomes are called as chromosomal disorders. And the best example for that is Down syndrome, Turner syndrome, and also Klinefelter syndrome. Down syndrome, Turner syndrome, and also the Klinefelter syndrome. Got it? It's very simple, okay? That is how you have to write this particular question. Now, this question is actually easy compared to others. So let's go ahead with the difficult ones. Slowly we'll move it, okay? Now, give reasons for the following. A simple cut results in non-stop bleeding in hemophilia affected individuals. Why? So, if someone is affected with hemophilia, no? If I get a small cut down there, the blood is going to go. Usually, when we get a cut, there's clotting happening in our body, right? It clots suddenly. Okay, we have proteins for that. But in them, it's not like that. Why? Probably because the proteins that are responsible for clotting are not being made. That means the genes that are going to code for those proteins might be affected. You get it? So I will say that a simple cut results in non-stop bleeding because a single protein that is a part of a cascade of proteins, a single protein which is a part of a group of proteins which is involved in clotting is being affected. That is not being formed. That is the reason why we have non-stop bleeding in patients who have hemophilia. Now, Turner syndrome affected females are usually sterile. So, Turner syndrome is the case where you should be having 2 chromosomes, as in 2X chromosomes for being a female. But over here, there is a loss of 1 chromosome, which is why we say this is XO condition. And since there is loss of 1 chromosome, I will say that this particular female, even though she has feminine characters out there, she will not be having a proper ovary. Her ovaries are going to be rudimentary. They are not going to be properly formed. So, I will say it is rudimentary ovaries which are going to be there, and this rudimentary ovaries cannot give rise to what? Ovum. Got it? That is why she is sterile. She cannot produce offsprings because the ovum itself, the gamete itself which is there, it's not of any use. So Turner syndrome affected females are usually sterile because they have rudimentary ovaries. They might not even produce ovum. That is also a case that could be there. So how do you frame the answer again? We need to know. Let's see. So females are usually sterile because they have only 1 X chromosome, leading to underdeveloped ovaries and lack of normal gamete production, which is there. Safe answer. Now, in Morgan's dihybrid cross experiments on Drosophila showed that flies having genes for yellow body, and white eyes exhibited lesser recombination. What do you mean by that? general thing that I want to tell you is this. Listen, suppose there are 2 chromosomes. My body is boiling. Okay, so there is A and there is B chromosome over here. There is capital A and capital B over here. These 2 genes are very close. These 2 genes are also very close. If at all I cut it and I exchange the parts, what is going to happen? Basically, I get a new chromosome which looks like this. Capital A and capital B comes on this side. Small a and small b comes on this side. Aren't these genes still together? Aren't these genes still together? Do you find any new combinations happening apart from, yeah, upper part is, yes, according to it, it will be new combination, but if at all you see these 2 genes, they are still close. So I'll say lesser recombination is happening there. Why? Because these 2 genes are tightly linked. They are tightly linked. When they are tightly linked, obviously lesser recombination will be there. And the example that I always use is that you and your best friend, if at all you're too close, there are less chances for someone else to enter your team out there, right? So lesser recombination out there. On the other hand, if at all the scenario was something like this, where you had capital A here, capital B here, small a here and small b here. These 2 genes are very far away from each other, right? Now if at all I cut this part and exchange, what is going to happen? Basically something like this is going to happen. Capital B, small b. And here since it's an exchange, small a, capital A. You see small a and capital B and capital A and small b is there. This was never the combination. So there is a higher amount of recombination out there in loosely linked genes. They are loosely linked and they are tightly linked. So here when I say in Morgan's dihybrid cross experiment on Drosophila, it showed that flies having genes for yellow and white eyes exhibited less recombination. That means probably they might have been very close to each other. They are very close to each other. Please remember it. Okay, and the other one, so there is one more thing, miniature wings. So miniature wing gene is very far from each other, which is why we say they have more recombination. The highest amount of recombination is how much? Do you know? It is 50%. So in this, they show almost around 37.2% recombination. That means it is too high. In this, basically only 1.3% recombination. Only 1.3% recombination. That means it is too less. So know that. Yeah. Okay. How do you frame the answer again? Yellow body and white eye genes in Drosophila show less recombination because they are located close to each other on the same chromosome, exhibiting what? Genetic linkage. That's all. Okay. Now we have one more question. Inheritance of skin color in the human show different phenotypes. Why? Inheritance of skin color. So do you think my skin color and your skin color is the same, or you and your friends is the same? No. Why? Because actually we show an exception to Mendel's law. Mendel said that one gene, one protein, right? But here it's not like that. Here what is going to happen is that multiple genes are going to code for one character, that is your skin color. So you have suppose capital A, capital A, capital B, capital B, capital C, capital C. Your genotype is going to be capital A, capital A, capital B, capital B, capital C, capital C. This is the genotype. This genotype shows highest skin color. Suppose your genotype is changed over here. It is not capital A, rather it is going to be small a. Similarly over here, it is not capital B. Here it is going to be small b. Here it is not capital C. Here it is going to be small c. What is going to happen? The genotype is going to change. It is going to be capital A, small a, capital B, small b, capital C, small c. This will be having lighter skin color than this. The more number of recessive alleles that you are using, my dear children, it will be lighter skin color. The lightest will be something like this. So here what do you see? Your skin color is determined by multiple genes out there, which is why since human beings will have different combination, we all have different skin color as well. Okay, so inheritance of skin color in humans show different phenotypes because this is showing polygenic inheritance. Skin color shows polygenic inheritance, but multiple genes has a role to play. And it is not just skin color, it also your height. Human height is also following polygenic inheritance. Okay, so it shows different phenotypes due to polygenic inheritance where multiple genes contribute to the phenotype, leading to a range of skin color variation. Different skin colors being formed out there. Works? Works. Okay, next thing. Accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why do you think accumulation of phenylalanine happens? Why do you think accumulation of phenylalanine happens? Because an enzyme which is needed for converting phenylalanine to tyrosine is not being formed. Okay, this enzyme the gene that codes for this enzyme is being mutated. Now, do you think enzyme is going to be formed? No. Do you think conversion will happen? No. So phenylalanine is going to be accumulated in the form of phenylpyruvic acid for the same reason. And this accumulation will lead to mental retardation as well. It will lead to mental retardation as well. So phenylalanine accumulates in the body due to defect in the enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine, causing what? Toxic buildup out there. Okay, this is also an example of pleiotropy. It's also an example of pleiotropy. I'll tell you, shall I tell you? Yes, I'll. So listen to me. Phenylketonuria, what happens is that phenylalanine which is there, it has to convert to tyrosine with the help of the enzyme phenylalanine hydroxylase, PH enzyme. Now this enzyme is not being found because the gene is being mutated right there, right? So this thing is going to be built up, okay? They convert into phenylpyruvic acid and they get accumulated in our brain and causes mental retardation. It causes mental retardation. Now one more thing is being affected. Tyrosine which is there, it is super important for a pigment to form in our body. And this pigment is responsible for our hair, for our skin and our hair pigmentation, hair coloration, right? Since this pigment is not being found because the tyrosine is not being found, your skin and your hair will be having reduced pigmentation. Reduced pigmentation. So one gene is being affected and two characters are affected. One gene is affected, which is why two characters are affected. This is known as pleiotropy. So usually according to Mendel, one gene, one character. But here one gene, multiple character can also be affected. So please remember it's an example of pleiotropy as well. Works. Pleiotropy, polygeny, do not get confused. Now, few autism-linked recessive gene blood diseases occurs in human population. Among them, some are related to qualitative and some are related to quantitative problem of synthesizing blood proteins. Explain. So, let's see. I'm tired. Yes. So, they are autosome-linked recessive gene disorders. So, I'm just dividing into two. One is going to be sickle cell anemia. Another one is going to be thalassemia. So what is going to happen over here and here? Know the difference. You have chromosome number 11 where you have HBB gene. You know HBB gene codes for what? Codes for beta globin chain. Beta globin chain of hemoglobin. So ultimately your blood, your RBC has hemoglobin and hemoglobin is made up of alpha chain and beta chain, right? And this hemoglobin is inside the RBC, right? So here the beta globin chain which is there, okay, it is not being produced nicely, as in there are some changes in the beta globin chain. What changes and why did it happen? Because there were some mutations over here. There were some mutations over here. Okay, let's see what is the problem that is occurring over here. Okay, so it was supposed to be GAG codon at this 6th position. So there are multiple codons. The 6th codon was supposed to be GAG, which codes for glutamic acid. Glutamic acid. Now, my dear children, instead of GAG, it is GUG. There is a point mutation. There is a change in the single base pair. Point mutation. Instead of A, it is U, which is why the codon will be now coding for another amino acid that is known as valine. And each amino acid has to come together to form what? A polypeptide. Over here, valine is going to come in this particular case and going to form the beta globin chain. Do you think it's a normal beta globin chain? No, it is not. It is a distorted one. Now this beta globin chain comes along with an alpha chain and forms a hemoglobin. So this hemoglobin is not proper. This hemoglobin goes into an RBC. In this RBC, the normal shape should have been different, but here it is going to be different. Just a second. Okay, dear children. Sorry, someone came in. Okay. So RBC is over here. Suppose it has this bad hemoglobin, what is going to happen? There will be a change in the structure of the RBC. So RBC has a biconcave shape, but now it will be spindle in shape. And when it becomes spindle in shape, what is the problem? It will go and bring clots in the blood vessels out there. And also remember, all of this change in the shape is happening because of one big reason, that is, there is a problem with the hemoglobin. When there is a problem with hemoglobin, there will be reduced oxygen carrying capacity. There will be less oxygen out there, which is exactly why the shape of the RBC also becomes changed to a spindle shape. Got it? This is the basic logic out there. For sickle cell anemia, always remember this is an example for point mutation that is happening over here, and A comes before U, so it was A first and then it was replaced by U. Similarly, G comes before V, so glutamic acid was the first thing, now it is replaced by valine. Right? Here, it is an autosomal recessive disorder. Why? Because these genes which are there, HBB genes, it is on chromosome number 11, which is an autosome. They are autosome. They are not sex chromosomes. They are regular chromosomes, that is known as autosomes out there. And basically, I call it a recessive disorder, which means that both the chromosomes should be affected for someone to have this particular disease. Otherwise, they will be just a carrier. Otherwise, they will be just a carrier. So in recessive disease, both the chromosome have to be having the recessive alleles, then only the disease will be got. Otherwise, that person would be just a carrier. Got it? I hope that is clear. And also over here, what is the problem? The quality of hemoglobin which is being produced, that is a problem. So it is a qualitative disorder. It is not quantitative. It's a qualitative disorder, that is in the case of sickle cell anemia. Now when we talk about the other disease, that is thalassemia, what is the problem up there is that there are two chromosomes. This is chromosome number 11 and this is chromosome number 16. On chromosome number 11, what do you have? You have HBB gene, HBB. On chromosome number 16, my dear children, you have basically HBA gene. That means this chromosome will have genes which will be helping in the formation of beta globin chain, and here you have the genes that is coding for alpha globin chain. And there are 4 of the genes over here. There are only 2 gene locations over here. Please remember that. So I'll call this as HBA1 and this is HBA2. HBA1 has 2 genes, HBA2 also has 2 genes. Now listen, here the problem is that suppose this gene is being affected. What is going to happen? There is a normal gene that will still help in the formation of beta globin. So beta globin chain is going to be produced, but the quantity of this is reduced. Suppose both of them are affected, then beta globin chain is not going to be produced at all. You get it? So this is a quantity thing. Similarly over here, suppose one particular gene is affected, what is going to happen? Still alpha globin chain is going to be produced, but the quantity is going to be sorry, it is going to be less. Suppose 2 of them are affected, again the quantity of alpha globin chain which is produced is less. Suppose all 4 of them is affected, alpha globin chain is not produced at all. When alpha globin chain is not being produced at all, do you think you can make hemoglobin? No. If beta globin chain is not produced at all, do you think you can make hemoglobin? No. So remember always, this is going to be a quantitative disorder. It's what, my dear children? It's a quantitative. It's because of the quantity of the globin chains which are being produced. Okay? And when you have so many genes being affected, that is why when you say that you have a major thalassemia patient, if only one gene is affected, other gene is still functional, you say that you are minor thalassemia patient. So please remember that thing also. This is also autosomal recessive trait. Why? Because over here also it's the autosomes genes which are being affected. Got it? Now how do you frame the answer for this? In sickle cell anemia, mutation in the HBB gene on chromosome 11 causes the production of abnormal hemoglobin. This defective hemoglobin leads to sickle-shaped red blood cells, impairing the oxygen transport and causing blockages in the blood vessels. Here the defect is caused because of the error in the quality of the protein that is being made, right? Whereas in thalassemia, it is caused by mutations in gene responsible for hemoglobin synthesis. HBA1, HBA2 for alpha genes, HBB for beta genes. There can be mutation in one allele of a gene, one allele of a gene, or there can be mutation in what? In more than one allele or both the alleles of a gene. It leads to reduced or no production of specific hemoglobin chains out there, causing anemia and fatigue. So anemia and fatigue is for this particular thing, thalassemia. Sickle cell anemia, it's a life-threatening disease. It can lead to death also. Similarly here also, if all the genes are affected, it can lead to death. Thalassemia also. Here the defect arises due to errors in the quantity of the gene which is being produced. So yes, so please remember, it's a qualitative and this is a quantitative disorder. That's all. Cool. Next thing that we have is define aneuploidy. Give two examples of aneuploidy. What do you mean by aneuploidy? Aneuploidy means either the loss or gain of the chromosome. That is what you mean by aneuploidy. Now if it is a gain, you call this condition as trisomy. If it is a loss, you call this condition as monosomy. Now you have been asked, mention the karyotype of Klinefelter syndrome of an affected individual and write its symptom. So in Klinefelter's, what is going to happen, my dear children? You should be having XY chromosome, right? These are sex chromosome, but here you have one more X chromosome leading to XXY condition. Okay. Generally, in our body, how many chromosomes are there, Baba? 46 chromosomes are there. 46 chromosomes there. Out of 46 chromosomes, 44 of them are autosomes. 2 of the chromosomes, that is 45th and 46th, they are the sex chromosome. They are what? The sex chromosome. Always remember that. Now over here, when I say you have an extra X chromosome, how do you denote it? Basically, you can write like this, that is 45 autosomes, comma, XXY. This is known as karyotype. This is known as karyotype. So how many autosomes do you have? Sorry, not 45. Here it is going to be 44. You have 44 autosomes in XXY. This is 44th chromosome, 45th, 46th, 47. In total, do you have 47 chromosome? Yes. Why? Because it is a trisomy condition. You have an extra X chromosome. Sometimes instead of writing like this, they also write it like this: 47, comma, XXY. That means 47 chromosomes are there and probably the extra one would be this X. So you can either write like this or you can write like this. This is known as the karyotype of Klinefelter's. And I told you what happens in Klinefelter's, you have an extra X chromosome. This is a trisomy condition, right? And because of this, the males which are there, they have a overall masculine development. Okay, males will be having overall masculine development, but they will also have gynecomastia, as in there will be development of breast. There will be development of breast in this particular case. Apart from that, my dear children, what is the next thing that you have? Apart from gynecomastia, there will be underdeveloped secondary sexual characters. Do you think they can be fertile? No, they will be infertile. Why? Because there is no spermatogenesis and anything happening over here. Do you think they will have height and everything out there? Yes, they have tall stature with long arms and legs. And learning, okay, so this is all is not important. At least remember these characters out there. Apart from it, you have multiple other examples for trisomy, autosomal trisomy. Listen, this is a trisomy, okay, but this is allosomal. Why allosomal? The sex chromosomes are known as allosomes. So here the sex chromosome is extra, that is why allosomal trisomy. Similarly, you have autosomal trisomy, that is Down syndrome, where the 21st chromosome is affected. 21st chromosome is an autosome? Yes, it is an autosome. Okay, so 21st chromosome, my dear children, over here, when it is being affected, how it is affected? You should be having 2 chromosomes, right? But you have an extra copy of the chromosome. Trisomy of 21st chromosome, which is why this person will be having symptoms related to Down syndrome. But again, what is a karyotype over here? Usually you should be having 44 autosomes and then XX, that is for females, 44 autosomes and XY for males. Here you have 45 autosomes. One extra autosome is there. Yes, one extra autosome is there. So can I write over here 45 autosomes XY if it's a male who is having a XX is if a female is having Down syndrome, 45 autosomes XY if it's a male who is having Down syndrome. Now what about Turner syndrome? In Turner syndrome, it's a monosomy condition. You should be having XX, but you just have XO. There is only one X, there is no other X. So again, how do you show that? Autosomes are normal, 44A, but sex chromosome is like this. So you write 44A XO. So 44 and 45th chromosome, that's all. Otherwise, you can also write in total how many chromosomes are there, Baba? Instead of having 46, you just have 45. So 45, comma, XO. This can also be written, but I prefer this. This is uniform. This will be easy for you to remember. Okay? Yes. Perfect. So aneuploidy is a condition in which organism has an abnormal number of chromosomes, either due to what, gain or loss of one or more chromosomes. It is caused by non-disjunction. This is very important. How it is caused? It is caused by non-disjunction during cell division. Okay, there is no proper separation that is happening during cell division. That is why this particular thing is happening. Non-disjunction during cell division, that leads to aneuploidy. Okay, you might have also heard about polyploidy. Polyploidy is basically the overall number of chromosomes is being increased. The whole set of chromosomes number is being increased. That usually happens in the case of plants. Okay, it happens in the case of plants. And that is not because of non-disjunction during cell division. That is because of basically the karyokinesis took place, nucleus division took place, okay, but the cell division did not take place, which is why a cell will be having double the number of chromosomes. Okay? Okay. Now, represent schematically the results of one gene interaction between pure tall pea plant and pure dwarf pea plant. Pure tall pea plant and pure dwarf pea plant. How do we explain that? How do you show one gene inheritance? This is very, very simple. Okay. So, pure tall looks like this, capital T, this will be small t, small t. Here the gametes. So, first write the parents. You have to write it. Okay. I'm not writing it, but you need to write it. Now, the gametes. From this 2 chromosome, one can go into a gamete. So, either way it will be this. Similarly, here it is going to be this, right? Now you do the fusion, this and this, you get capital T small t, this and this, you get capital T small t, this and this, capital T small t, this and this, capital T small t. So what do you write? This is going to be the F1 generation, and you write basically all of them are tall. Okay? Phenotype, physical appearance which is there, all of them are tall. What is the genotype? Genotype is all of them are capital T small t. Why they are tall? Because the capital T is dominant over the small t, right? Now you do F2 generation. How do you do F2 generation? Basically over here, let me make Punnett square. So in Punnett square, you just have to write 2 parents. So one is, I am crossing between what 2 parents? I am crossing between capital T small t and capital T small t. Basically I am doing what? Selfing. So how do you write it? One parent you write it over here, capital T small t, another parent you write over here, capital T small t. When this and this fuses, you get capital T, capital T. When this and this, you get capital T small t. When this and this, again capital T small t. When this and this, you get small t small t. What exactly happens over here? Capital T, this is going to be tall. What about this? This is also going to be tall because you have a dominant allele. This is also going to be tall. So basically out of 4 of them, 3 are tall. So if at all, I have to write the phenotypic ratio, physical appearance ratio. How do I write? 3 of them are tall. So 3 bracket tall, colon, 1 of them is going to be dwarf. So phenotypic ratio is what? 3:1. Okay, ma'am, what is the genotypic ratio? Genotypic ratio, my dear children, if at all you see, 1 of them is capital T, right? Only 1. 2 of them which is there, it is going to be capital T small t, so capital T small t, and 1 of them is going to be small t small t. So write like that and you have got the answer. So this is how you are going to show. Please make sure you write all the ratios properly. Mention what does 3 stand for, what does 1 stand for. It's 3 of them tall, 1 of them dwarf. Done. We can get question related to this, as in basically either dihybrid cross or monohybrid cross, or basically incomplete dominance and things like that. Now categorize following as either autosomal or sex-linked disorders. So in sex-linked, in your NCERT you just have 2, and that is hemophilia and color blindness. So this one goes like sex-linked. This one, this one, this one. Thalassemia, phenylketonuria, myotonic dystrophy. All of them are autosomal. Now, in your NCERT, the sex-linked, autosomal, everything is recessive. Okay. Only one particular diagram has been given in your NCERT in the pedigree chart, and that example is of myotonic dystrophy. This is autosomal for sure, but it is autosomal dominant disorder. This is the only one which comes under autosomal dominant disorder. In your NCERT, one more thing is given, cystic fibrosis. They have not explained it, but you should know it is also autosomal recessive. So only one dominant disorder is there, and that too autosomal dominant, which is myotonic dystrophy. Okay, done. Now, what conclusions will you arrive after careful observation of the below given diagram? So, below given diagram has something like this: 1, 2, 3, 4, 5, 6. 6th position you have valin, you have GUG over here. Now, valin which is there at this particular position, you need to understand that basically Here, point mutation has taken place. You just have to explain about the sickle cell anemia. Say that point mutation took place, you had GAG, that code for glutamic acid, but now it is replaced by GUG, which is coding for valine. Explain that, and that's all your answer about. From the diagram, we conclude that a single base substitution in the mRNA codon has occurred. The codon becomes GUG, which codes for valine. So, in the beta globin chain, glutamic acid at the 6th position is replaced by valine. Just explain that. Got it? Yes. Now what? Identify the disorder by the following symptoms. Individuals with overall muscular development with gynecomastia. Overall masculine development is there. Overall masculine development. Okay. But they have gynecomastia, as in basically breast development. This is seen in the case of Klinefelter syndrome. So you have to identify the syndrome, and that is Klinefelter syndrome. Okay. Now, schematically represent the incomplete dominance. Incomplete dominance is basically what? It is not completely dominant. So in the case of capital T, small t, we saw that it was completely dominant. That is why here the character was tall. But in incomplete dominance, things are going to be different. How different? Let's see. So over here, you had a flower whose name was snapdragon flower, or you also call as Antherinum majus. Okay, Antherinum majus. So over here, you take the red plant, or red flowers, and basically white flowered plant. You mentioned it as small r. This is red and this is white. And you cross it. Let's see what is going to happen. Let's form the gametes first. These are the gametes. When you cross this and this, you get capital R, small r. This and this, capital R, small r. This and this, capital R, small r. This and this, again capital R, small r. Everywhere you have one capital R which will be dominant over the small r, right? So the feature should be what? Red. In the first generation, it should be red. But what do you think is the color? The color is actually, all of them are pink in color. Oh my God, why is that happening? Because capital R is incompletely dominant over the small r. This is an exception to Mendel's law. Okay, Mendel gave law of dominance, right? According to it, this was not the case, but this is an exception. Again, okay, they were shocked, so they crossed it once more. So capital R, small r, capital R, small r. Gametes are going to be capital R, small r, capital R, small r. Let's cross and see. This and this fuses, you get capital R, capital R. It's going to be red. Can I write red? Let's write red. This and this fuses, you get capital R, small r. What is this now? This is going to be again pink in color. Why? It's because it's incomplete dominant. Okay, when this and this was crossed, again capital R, small r, again pink in color. When this and this small r came in, and that is what? White in color. That is white in color. So what is the phenotypic ratio? Until now it was 3:1. All of them were red, right? Usually it should have been red, but now it is not like that. So phenotypic ratio, my dear children, is what? Is basically 1 red is to 2 pink is to 1 white. So 1 is to 2 is to 1. What about the genotypic ratio? Genotypic ratio is also like that. 1 capital R, 2 capital R, small r, and 1 small r, small r. Got it? That is going to be the genotypic ratio. So genotypic and phenotypic ratio are going to be exactly the same over here in the case of incomplete dominance. Done? Okay. Please note that they do not follow law of dominance. They do not follow law of dominance over here. Similarly, in the case of single gene, one gene inheritance, what do they follow? They follow law of dominance and law of segregation. Law of segregation means when you form a gamete, only one gene is going to go. Okay, only one gene or one allele is going to go into a gamete, not both of them. So yes. Now, the next question which is there, identify the genetic disorders. Okay, we have to identify. Sex-linked recessive disorder occurring in about 8% of males and only about 0.4% of females. 8% and 0.4, it's not even 4, 0.4% of females. Leading to defect in the cone of the eye. So listen, leading to defect in the cone of the eye, that means it has to be color blindness. It has to be color blindness. Why do you think it's the males who are affected more? Because males just have one X chromosome. right? They have another Y chromosome. In the case of females, you have two X chromosome. So if one is affected, other one is there to overshadow the effect, because it's a recessive disorder. In a female to have this, the female should be having both the genes being affected. So since it is a recessive disorder, even if one chromosome is affected, other chromosome is there, not there to overshadow, it will be showing up the disease. You get it? That is why males, they are generally affected. In the case of females, they can be carriers. They will be carriers, they will not have the disease, because there is one to overshadow the effect. So the first disease that we have is what? Color blindness. Now what do we have? Sex-linked recessive disorder affecting a single protein involved in the clotting of blood. Clotting of blood means hemophilia. It's a blood disorder, but it is sex-linked blood disorder. Okay, next. Inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. Phenylalanine getting accumulated in phenylketonuria. And phenylketonuria is again an autosomal recessive disorder. I told you, only two sex-linked disorders are there. That is color blindness and hemophilia. Rest, apart from that, everything. Let me take sickle cell anemia or thalassemia or basically phenylketonuria. All of them are autosomal recessive. Over here, your answer is phenylketonuria. Please know the symptoms of all of it. Okay, there will be mental retardation. Apart from that, in the urine it will be a foul smell, because this particular thing which is being accumulated, it's not absorbed by the kidneys, it is being moved out, which is why there is a smell. Okay, now autosome linked recessive blood disease, substitution of amino acid in beta globin chain of hemoglobin. Autosome linked recessive blood disease, substitution of amino acid in beta globin chain. Substitution word came, that means it is sickle cell anemia. Autosome recessive blood disease, again reduced rate of synthesis, reduced, that means quantity, that means it is going to be thalassemia. Got it? Yes, ma'am. Okay. Next, represent schematically the result of two gene inheritance. Now how many questions do we have over here? We have, okay, we are going to complete this. Okay, let's do. Two gene inheritance of seed shape and seed color. This is very easy. Okay, if you understand, try to understand, it's going to be super easy. I'm talking about dihybrid cross. That is why two gene inheritance, right? Dihybrid means I am going to talk about two characters at once. So seed, shape and color. Similarly, shape and color. Let's talk about shape first. Round. Here it is going to be wrinkled. Just think of tall and dwarf, just like that. Similarly over here, it is yellow. Here it is going to be green. Okay, how do you show that? Capital R, capital R, capital Y, capital Y. Similarly, small r, small y, small y. Which one is dominant? You know this one is dominant, right? Now, how do you form the gametes? Listen, this is important. This is where most of you do not understand things. So you have capital R over here, capital Y over here. When you make the gene from a pair of chromosome, only one will come. Sorry, when you make a gamete from a pair of chromosome, only one is going to come, one allele is going to come. So either this or either this. Whatever you do, it will be capital R from here. Here also, whatever you do, it will be capital Y only, right? Similarly, here, whatever you do, one will be small r from one and one will be small y. Agreed? Now, when both of them come together, it will be capital R, small r, capital Y, small y. Now you have to use your imagination. It looks like this. Capital R, small r, one chromosome came from here, one chromosome came from the other parent. Similarly, capital Y, small y. This is how it looks. Which is why all the plants are going to be what? Round and yellow in nature. They are going to be round and yellow. Why round and yellow? Because over here you have one to dominate, here also you have other one to dominate, right? Now, when you do selfing, in the first generation you only saw the parental character, first dominant parental character, that is round and yellow. So they did selfing. In selfing, what is going to happen? Let's do the selfing and see. Can I make it here itself? No, I would need another one. So listen, I have now round and yellow like this. Similarly, round and yellow like this. Now, you have to use your imagination. It goes like this, capital R, small r, and capital Y, small y. Listen, now when I form the gametes, from here one can come, it can be capital R, from here one can come, it will be capital Y, suppose. From here one can come capital R, from here one can come small y, suppose. From here one can come small r, from here one can come capital Y, okay. From here one can come and from here one can come Got it? These are going to be the 4 gametes up there. These are going to be the 4 different types of gametes. Why do you think they form? Because of law of independent assortment. In dihybrid cross, you have law of independent assortment, which says that whenever you are forming the gametes, the alleles of one particular character, that is shape, will be independently assorting from the alleles of another particular character, that is probably color. So does capital R say I will only go with capital Y? No, there is nothing like that. Which is why in future you will be able to see non-parental character. So you crossed round and yellow and wrinkled and green, but now you will see round and green, which was not there in the parental. Why? Because of law of independent assortment. Please know. Please know, this is very, very important. They ask you, in Mendel's experiment, how did he show non-parental character? Because law of independent assortment was being formed over there. Okay. In T.H. Morgan, how was he able to say non-parental character? Because crossing over between the genes was being seen. That is totally different. The linked genes will have less recombination. The loosely linked genes will have more recombination. That is what he said. This is different. That is different. Please know about that. But both of them are going to lead to what? Non-parental characters. Now, you have formed the gametes. Write the gametes on this side, because the same parent will produce, this parent will also produce these type of gametes. Now you cross them and it looks something like this. Where is it? It will be looking something like this, right? So when you cross capital R, it will capital R, capital Y, it will come like this. If at all you see, there are 4 into 4, 16 progenies will be produced. How many? 16 progenies. Now this is something that you have to remember for sure. And the ratio of this, phenotypic ratio, is what is there. Shall I remove this? Is it going to be a problem for you? Okay, I'll remove this. So it goes like this, okay? Where am I? Oh my God. 9 is to 3 is to 3 is to 1. 9 of them are going to be round and yellow. That is a dominant trait. One of them is going to show the recessive trait, which is what? Wrinkled and green. Wrinkled and green. Now 3 and 3 are over here. What are they going to be? If this is round and yellow, this is going to be round and green. It's going to be ulta, round and green. If this is wrinkled and green, it is going to be wrinkled and yellow. Got it? Wrinkled and yellow. So this character, did you see any time until now? No. So these 3 plus 3, 6 characters are non-parental thing. They are non-parental progenies. If at all in a question it comes that 64 progenies are produced. I'm telling you, I've repeated this question so many times, but telling you now also. If at all, they're asking you 64 progenies are produced. Out of them, how many of them are going to show non-parental character according to dihybrid cross experiment? So 64 are there in general. Here, out of 16, how many of them are non-parental? 6. So I'll write 6 by 16 into whatever number is given. If I do the calculation, it comes out to be 24. So 24 out of 64 will be what? Non-parental. Similarly, you should be able to do the calculation. Okay? Done. Perfect. Now, Mendelian disorders may be autosomal or sex-linked, dominant or recessive, homozygous or heterozygous. Pedigree symbols are used to analyze these disorders in human population. Mention the genotypes for the following pedigree symbols. Okay. So, you may get a question based on pedigree analysis as well. Make sure you solve it. Autosomal dominant disorder is this. And I am saying that this particular person, that is a man, he is being affected. Right? Because the color is different. Color is like that. So this person has to be affected. I'm talking about autosome and my genes are like this, capital A, small a. What should be my genetic combination over here? It should be either this or it can be like this. Because you have a dominant allele, the dominant allele will show the effect and that person will be having the disease. Here both the alleles are dominant, which is why again the person will be having the disease. So it can be like this or this, then only the person will be affected. See, you have to check over here and here. Similarly, autosomal recessive. Autosomal recessive disorder is what we're talking about, and here there's a lady, circle means lady, right? And she is not having the disease. That means it should not be in recessive condition. Listen, if at all it is in this condition, then you will get someone like this. They will have the disease. Why? Because it is an autosomal recessive disorder. Now you have to make sure that that person does not have the disease, because over here it is not being colored. So you have to do the ulta of small a, small a. That will be either this or this. You should be having one dominant allele coming up. So it will be just similar to this. Got it? Getting it? Next thing, sex-linked dominant. Sex-linked dominant means, my dear children, over here, suppose you have XA and XA. This is the dominant one, this is the recessive one, because you have small a over here, right? So when I say this is a guy has 2 chromosomes X and Y. And when I am saying that is X-linked dominant disorder, but this particular person does not have it, that means that this particular will be having a recessive allele. If at all the person had something like this, then he would have been affected. But here you are saying this person does not have this, right? So you have to make sure it's the recessive allele that you have to put. So over here that is being mentioned. Similarly, sex-linked recessive disorder. Now, sex-linked recessive disorder is given and both the male and the female are affected. If it is recessive disorder and if you want to make both the male and female be affected, you have to put the recessive allele in the case of male because only one X is there. In the case of females, you have to make sure it's not just on one chromosome but on both the chromosome. Then only she will be showing. Otherwise, in her, you know, the dominant allele will be taking up over that. So both the alleles have to be affected for her to be affected. Get it? So this is what you have to write. Is there a confusion? If there is a confusion in this question, go back once more and go through it. You will be able to do it. Got it? And this is very, very important. Very, very, very important. Cool? Yes. Next thing. Now, observe the representation of a dihybrid cross and answer the questions. This is the dihybrid cross experiment that we just did. Phenotypic ratio, we know it is what? 9:3:3:1. Do you know the genotypic ratio? It's very simple. 1:2:2:2:1 and then 2:4:2 and then again 1:2:1. So that is the genotypic ratio. Usually it does not come, but no. 1:1, 2:4:2, 1:1. Okay? Now let's come back over here. Okay. Identify homozygous dominant genotype in the progeny. Homozygous dominant. Homozygous means both the alleles have to be same. Homozygous, and I am saying dominant. So basically this is the condition. You cannot say this one, okay, because why? Both the chromosomes, both the alleles are not homozygous over here. You have to make homozygous, that means both the alleles have to be same. Cool? Next thing. Identify recessive genotype in the progeny. Recessive, if at all you're talking about recessive, it has to be this. It has to be this. Okay, there is no other progeny which is recessive. Recessive condition will always have both the alleles to be similar. Now, if at all you were asked about heterozygous dominant and all, you could have given the answer like this is heterozygous dominant. Why? Because here the alleles are different. Here you were asked about homozygous dominant. So again, yeah. Now, number of parental type of progeny. How many of them are parental type? You know the ratio 9:3:3:1. I told you 9 and 1. This is round and yellow and this is wrinkled and green. 9 plus 1 is equal to what? 10 of them are like parents. How many recombinant type progenies are there? 3 and 3. 6 of them are going to be non-parental, or basically what do you call as recombinants. Got it? So this is the thing that was given, so I thought I'll explain. Fine. Next thing. So here the answer is also given. If at all you want to see, you can check it out. Now, I think this is going to be, no, second last question of this chapter, okay? This is a hot question. A cross between a normal couple resulted in a son who was hemophilic and a normal daughter. Let's do. A cross between a normal couple. So that means, okay, resulted in what? A son who was hemophilic. Son who is hemophilic. That means you have to color it up, okay? And a normal daughter. And a normal daughter. Normal daughter is why you're not coloring it up. The daughter was married to a normal man. Daughter is married to a normal man. Okay. To their surprise, the grandson was hemophilic. Now their son, grandson means basically, okay, their daughter's son, grandson. To their surprise, the grandson was having hemophilia. Okay. Represent the cross in the form of pedigree chart. I have already done that. Give the genotype of the daughter and the husband. So, if I say that the male is having the hemophilia, that means he is something like this, Baba. Agreed? One gene is going to be affected, that obviously is going to be the X chromosome. X chromosome, do you think they can get from the father? No. X chromosome you get from the mother only. Y chromosome you get from the father. So, here, if I am making it like this, you should understand I should have one normal X chromosome to overshadow the effect. Then only she will not be affected. Here it is mentioned that she is not affected. She could be a carrier. She can be a carrier. So her genotype is like this. Only if the condition is like this, then you get one X chromosome from here, one normal Y chromosome from there. This affected X chromosome would cause disease in the case of the guy. You get it? Use the logic. You never can make it something like this. Otherwise, she should also be having hemophilia. Okay, because this is what? A recessive disorder. So since she has an extra X chromosome that overshadows the effect, which is why she is not growing. Now, if at all it's just normal XX, it's normal XX, then how will he get the disease? He's disease, that means he have got one X chromosome at least, right? So, yes. So this would be the genotype of the female, and that is a daughter. And you're asked about the son, right? And her husband, daughter and the husband. So husband is normal, it will be just XY. Husband is normal XY and female daughter is like this. Okay. Now write the conclusion you draw of the inheritance pattern. This is what? Sex-linked recessive disorder. And since it is recessive, it will be skipping generation. So over here the parent does not have it, but here the child had it. Here the parent does not have it, it has it. Okay. Now sex-linked recessive disorders, in that case the males are the ones which are mostly affected. So you can write that also. Okay. So I have written everything. Write the conclusion you draw. X-linked recessive, it is through transmitted to carrier females. It mostly occurs in males, and trait can skip a generation because it is recessive. Cool. Now we have one more question, the last question from this chapter, and that is, refer to the representation of a pedigree chart of a particular trait and answer the question that follows. So here is the question. So there is a circle and what do you call a square, and then you have to mention the rest. So which genetic disorder does the pedigree represent? Which genetic disorder does the pedigree represent? So over here you see there is skipping of generation. Here the parent does not have it, but the children have it. Right? Skipping of generation is there, that means it is going to be recessive. Now, is it sex-linked or autosomal? In sex-linked, obviously the males are the one which are mostly affected. In the case of autosomal, it can be male and female. So over here you can clearly see in one generation both male and female that is being affected. So I can say it probably could be autosomal. And let me tell you, in your NCERT, this particular pedigree chart is given, and directly they are saying it's what? Sickle cell anemia. So they have given you two pedigree chart. Please know it. One is myotonic dystrophy, which is autosomal dominant. It will have a diseased person in every single generation. This is autosomal recessive. This will be skipping generation. So as I said, here the parents do not have it, here the children have it. Here the parents do not have it, here the child has it. You get it? So please know that. And that is myotonic dystrophy. It's autosomal dominant. This is recessive. Now, which genetic disorder does the pedigree chart represent? Sickle cell anemia. Mention the cause of the above trait. Just say single mutation is happening out there. This is what we studied about sickle cell anemia. Is caused by point mutation. Due to this mutation, valine replaces glutamic acid at the 6th position of the beta globin chain. Okay. Now write in 3 features of sickle cell anemia. It's an autosomal recessive. Red blood cells become sickle shaped under low oxygen tension. Heterozygous. Okay. Suppose, my dear children, there is a heterozygous condition. One person has something like this. This gene is affected. This one is normal. Okay. So what is going to happen? Basically, suppose 2 heterozygous people are marrying. 2 carriers are marrying. They do not know that they are carriers of hemophilia. Listen, if at all I do this cross, it comes out to be something like this. Very, very important. This is same for thalassemia and everything. Phenylketonuria, because all of them are autosomal recessive, right? So 2 people can be carriers. They might not have the disease, but the child might have. How much percent of the child? Let's see. This and this crosses, you get HBA, HBA. A is a normal one because that's why the person would be normal. They will not have the disease. This and this crosses, you get HBA, HBS. Here, in this particular condition, again, they would be carrier. They have one bad gene, but the other gene is there to overshadow the effect. This and this crosses, you get again HBA, HBS. Again, over here, what is going to happen? What is the thing that is you're going to see? Is that, okay, is that basically again here they are going to be carrier only. They do not have the disease. Now, when lastly this and this crosses, you get HBS and HBS. And when this comes in, when HBH, HBS comes in, that means that person will be having disease because this is a recessive condition. So out of 4, 1 is affected. So I can write 1 by 4 is equals to what? 25% of the progeny is going to be affected. Got it? As simple as that. So please know that 25% chances of a progeny being affected is there. Cool? Cool. And now we go up to the next chapter, that is human health and diseases. My dear children, I strictly believe that it is definitely helping you out. Please let me know if it is helping you out, okay? In the comment section below. Okay, my dear children, let's go ahead with the next chapter, that is human health and diseases. And the first question that you have over here is, what are lymphoid organs? Write the functions of the following in response to immune system of our body. Now this question is very simple, very straightforward, but you should know the logic and some keywords out there. So always remember, the lymphoid organs are the ones where your lymphocytes are going to develop, they are going to become mature, and also they start doing dishum with the antigens or with the pathogens out there. So their major function is to kill the pathogens, right? So the exact thing is happening over here, but in a structured way. So there are two types of lymphoid organs. First one is the primary lymphoid organ, okay, and the second one would be the secondary lymphoid organs. In primary, you just have two of them. Always remember, only two. One is your bone marrow, and the other one which is there is going to be the thymus. Now, bone marrow is something that is found inside your bone. Inside each of your bone, my dear children, you would be seeing something. There is red marrow and there is yellow marrow. Yellow marrow contains the cell which is responsible for formation of fat. Apart from that, the red marrow contains some stem cells. Some cells which has the potency to become different kind of cells. That is what you call as stem cells. Now, over here, they are hematopoietic stem cell, which means that they are going to form different forms of the blood cells. Not any other cell, just the different forms of blood cells. You know, there is RBC, there is WBC, there is platelets and all of it. So all these kind of cells are being formed inside the red marrow of your bone marrow. Now, when I say red blood cells, white blood cells are being formed, you should know that under white blood cells comes what? Your lymphocytes. That is your B and T lymphocytes. Okay, under white bloods comes your B and T lymphocytes. Apart from that, you have neutrophils, you have so many other cells, macrophages, everything out there. Now, so we got an understanding that, okay ma'am, I got an understanding that B and T lymphocytes, which are major cells out there, they are definitely formed inside the bone marrow. So are they just formed over here? Yes, they are just formed over here, but also you need to understand they mature as well. They become mature over here. But here there is something that you have to note. The T lymphocytes which is there, it goes to the thymus. Now, thymus is somewhere over here that is behind our sternum. So, sternum is a place where all your ribs are going to be joined. Okay, so behind your sternum, or what you call as chest bone, you have a small little thing which you call as thymus. So, the T lymphocytes which is there, it comes over here into the thymus and becomes mature over here. It is formed there, but it comes over here for maturing. B lymphocytes stay there and matures there itself. Now, one more very important thing that you need to know about primary lymphocytes is that primary lymphoid organ is that this is the place that the lymphocytes which is there, it becomes antigen sensitive. What do you mean by that? Antigen sensitive means they know that they would be having some, some cells which they have to fight against. Okay, they are becoming antigen sensitive. They know that these are the pathogens, these are enemies, and I have to kill them. But do they start killing them over here itself? The answer is no. They just become sensitive. They do not start their work over here. So then, ma'am, where do they actually meet the pathogens and kill them? It is going to be the secondary lymphoid organs, where these cells, my dear children, are going to become what? The effector cells. Effector cells that means that they are going to show their effect of killing the antigens. So always remember, whenever the term antigen sensitive comes, it's in the primary lymphoid organs. Whenever the term comes that they become effector organs, where do they become effector organs? In the secondary lymphoid organs. Got it? Now, under the secondary lymphoid organs, you have multiple things. For example, the most famous one that is there is lymph node. Is what? Lymph node. So basically, you know, just like how we have our arteries, veins and capillaries, that is a blood vessel out there, we have lymphatic vessels. We have lymphatic vessels, which is responsible for carrying anything extra which is there in between the cells back and giving it back to the heart. That is the responsibility of the, the lymphatic vessels. The lymphatic vessels which are there, they would be having some places like this. Okay, some places like this, which is kind of enlarged. Why do you think they have places like this? Well, in these places you have so many lymphocytes already standing up there. Why do you think that? Because whenever the lymphatic vessel is basically carrying any extra fluid between the cells, and suppose there is some pathogen in between those particular cells, they also come in through the lymphatic vessel out there. Now there should be someone to trap them. Who is that? It is going to be the lymphocytes which are there in the lymph nodes. So don't you think this is a place that they are going to show their effect, that they become effector cells, they are going to do dishum dishum? The answer is yes. So you have lymph nodes or lymphatic vessels throughout your body, and you might have seen when you go to doctor checks over here and touches and see that whether it's swollen or not. What is he or she checking? He or she is checking basically, are your lymph nodes swollen? If at all it's swollen, that means there's an infection going on and probably your body is reacting to it. You got it? Okay, fine. So that is one very important thing. Apart from that, you have spleen. Now spleen is just beside our stomach, my dear children. Spleen is what you call popularly known as the graveyard of RBC. This is where the old and worn out RBC is going to go and die. Now when I say that it has blood supply to it, and when I say that RBC are coming, I can also imagine that there will be some pathogens also which are there in our blood that will be coming to the spleen. Now here, my dear children, within our spleen there are soldiers lined up. Soldiers as in lymphocytes lined up there. So whenever some pathogen comes in, those pathogens are being killed, or those antigens are being killed. So can I say again that here is a place where they become effector cells? Absolutely yes, I can say that. So that is clear to you. Now we have the next thing, that is MALT. MALT stands for mucosa associated lymphoid tissue. What do you mean by that? So basically our intestinal tract overall, the innermost lining of the intestinal tract, which you cannot see, which is innermost, my dear children, which lines up everything, it is known as mucosa layer. It is known as mucosa layer. Now this mucosa layer can have a group of lymphocytes around there at different locations. For example, we have that in our tonsils. So tonsils that you see, my dear children, is a part of mucosa associated lymphoid tissue, and here, my dear children, you would be having so many lymphocytes, which is why whenever something comes up, we have throat pain and things like that, probably because our body is reacting to it. Okay, our body is fighting for us. Similarly, so under MALT comes tonsils. Apart from that, there is one more thing, that is Peyer's patch. Have you heard about it? Peyer's patch. So this is again found in the mucosa layer, but found where? Not over here. It is found towards the intestinal region. So it is found towards the intestinal region, which is having high number of lymphocytes out there. Apart from it, you also have appendix, which is there. Always remember, mucosa associated lymphoid tissue occupies around 50% of the whole lymphoid organs. So 50% of the whole lymphoid organs, basically secondary lymphoid organs, are coming under MALT, mucosa associated lymphoid tissue. Got it? So now, how do you frame this answer? First of all, what are lymphoid organs? Organs where origin, maturation and proliferation. Origin as in they are formed, maturation as in they are going to mature, they are going to differentiate, they will be knowing that what I have to do in future. And proliferate as in increase the number of the lymphocytes occur over here in the lymphoid organs. Now primary organs such as bone marrow and thymus produce antigen sensitive lymphocytes, okay, which migrate to secondary organs such as spleen, lymph nodes to become what? Effector cells whenever an antigen comes into an action. Now bone marrow, which is there, it is a main site where all blood cells including lymphocytes are formed. Agreed, okay. Provides microenvironment for B lymphocyte maturation. I told you B lymphocytes stay there and mature their self, correct. Thymus, primary lymphoid organ for T lymphocyte maturation. Here the T lymphocytes mature, which is why you have T name over here, because it is maturing in the thymus. Now immature T cells become antigen sensitive T lymphocytes and later differentiated into what? Effector cells. Later, not here. It will become effector cells in the secondary lymphoid organs. Also remember, the size of the thymus is going to reduce as we are gonna grow up. Okay, and the thymus would be somewhere over here. Now, we are asked for the next thing, that is spleen as well as lymph nodes. Secondary lymphoid organ that filters blood and traps blood-borne antigen, contains lymphocytes and phagocytes that help activate antigen-sensitive cells into effector cells. Or basically, I can just say that they become effector cells over here and start killing the antigens. Lymph nodes, same thing, that is there. Trap antigens from the lymph entering the body. Lymph as in the extra fluid, interstitial, in between the cells, whatever fluid is there, that is known as lymph fluid. Okay, so that runs in the lymphatic vessels. So provide site for lymphocyte antigen interaction. Again, they become effector cells over here. Now this is the picture that is given of a spleen. So my dear children, if at all you see, this is your stomach and just beside it you have this spleen right over here. Similarly, if at all you see this, these are your lymphatic vessels. The green color thing is a lymphatic vessel, and in between that you can see small round things. These are the lymph nodes which are highly supplied with lymphocytes. Apart from it, what do you have? MALT, mucosa associated lymphoid tissue, which is the one that contributes majorly. It contributes 50%. So that is a very big number. Got it? Got it, ma'am. Perfect. Now we go to the next question, that is, give the schematic representation of replication of retrovirus. Now you might be asked to draw this whole thing. Okay, you might be asked to fill in some steps over here. That means that your concept should be superbly clear over here. So please listen to me whatever I am trying to tell you. So first thing first, you have your retrovirus, that is your HIV. Do you know the genome of HIV? What is HIV genome made up of? For example, in our case we have a double-stranded DNA, right? But what about HIV, my dear children? It has single-stranded RNA. What does it have? Single-stranded RNA. How many of them are there? Ma'am, 2. So ma'am, 2 of them do not come together to form complementary double-stranded RNA. No, they remain single-stranded only. So always remember, you just have to write ssRNA. That means single-stranded RNA. Okay. Apart from it, it has also the protein code. So since there are chances that you might forget, I am just writing it. It has single-stranded RNA. This is what? The genome. Okay, perfect. Now, it also have the protein covering, that is the virus protein coat is there, agreed. Apart from that, there will be some proteins also, as in some enzymes also within this. Now, the virus which is there, it is going to affect the normal cells. Always remember, HIV virus, first of all, will be going into macrophages. Its actual target is T helper lymphocytes. Its actual target is T helper lymphocytes. Why? Because T helper lymphocytes is something that is a mediator in our immune system. It's a mediator in our immune system, right? So if at all that guy is damaged, our immune system will go very low. And that is the exact thing that the virus wants. Okay. So it is the T helper cell which is the target, but initially if at all it wants to increase its number, first of all it will go and attack the macrophages. What phages? Macrophages. Those are also different type of cells. So it came and attacked the macrophage. What does it do over here? When it comes and attacks, it will be transferring its single-stranded RNA along with some very important proteins which are there inside into the cell. This is our cell. This is our macrophage. Now, the target is to utilize our machinery. How can it utilize our machinery? It has to get inside the nucleus and basically be part of our DNA. Because whatever is there in our DNA, that is going, whatever information is there, that is going to be transcribed, translated, and the proteins and everything are going to be made, right? So if at all it wants to use our machinery, the first thing that it would have to do is, this is an RNA, it has to transform itself into DNA. Ma'am, is that possible? RNA to DNA? I have heard about DNA to RNA. That is transcription, right? DNA transcribes into RNA. That is transcription. Can't we do the opposite of it? We can, only if at all we have an enzyme for that, and that is known as reverse transcriptase. Okay, it reverses the transcription process, which is why it is known as reverse transcriptase. Did the virus have it? Yes, the virus brought this particular enzyme also. I told you there are some proteins inside, right? So this is one such thing. So what does it do? It does reverse transcription with the help of very important enzyme, that is reverse transcriptase. And remember, this is against what is followed in the central dogma. The central dogma says from the DNA you get the RNA, from the RNA you get the protein, and basically DNA to DNA is known as replication. This is what is central dogma. But now since ulta is happening, I'll say this is against the central dogma, and it is because of the enzyme reverse transcriptase of the of the virus up there. Now you have got the DNA. What does the DNA do? DNA will be slowly getting inside the viral DNA, incorporates into what's the host genome out there. That is our genome. It gets inside again with the use of some enzymes only. You call this as integrase. It's not given in NCERT, so you did not learn it. Okay, so it now incorporated over here. After incorporating, it is going to use our machinery, and what does it do, Baba? It will form new viral RNA. It has to form RNA again, right? It has to come back in this form, which is why whatever was there initially will be formed here. So new viral RNA is being produced with the help of information given by the DNA. Now new viral proteins are also being produced, and finally you have the RNA, you have the protein covering, and they would be moving out. So please remember these steps. New viral RNA is produced by the infected cell, and new viruses are produced again. Got it? This is the basic funda. You should be knowing each and everything about it, otherwise you might lose your marks. Now I'll be showing you similar question that has come this year, okay, and it goes like this. Just a second. Okay, let's solve this. So what you have to do is fill in the missing data in boxes 1 and 2, that is this and this. Give an example for retrovirus. Retrovirus is basically HIV. So example for retrovirus is what? HIV, human immunodeficiency virus. Now name the disease caused by the retrovirus. It is AIDS. What do you mean by AIDS? Acquired immunodeficiency syndrome. That means you acquired it during your lifetime. You got this disease during your lifetime, and it makes your immune system deficient. That is why acquired immunodeficient, okay, syndrome, that is there. Syndrome means group of symptoms will be there. Since your immunodeficiency, what is going to happen? There will be a group of symptoms also being shown up. Now name the diagnostic test for the disease. What is the diagnostic test? It is ELISA. What is it? ELISA, enzyme linked immunosorbent assay. So over here I have written everything. Enzyme linked immunosorbent assay. How does it work? It works on the principle of antigen antibody binding. Antigen antibody binding, as in, I'll just give you an example, so please remember that, okay? Antigen antibody binding, that is a principle of how enzyme linked immunosorbent assay works. Suppose I already have some setup like this, where I have the antibodies which are produced against the HIV. I know that these are what? Antibodies which are produced against HIV. Now I'm testing whether Pappu has HIV or not. What do? I take some blood from Pappu, okay, and basically if Pappu has HIV, what is going to happen? In that blood there will be some antigens, as in there will be some pathogens, and these antigens will be coming and attaching over here, if at all they are correct match, because these are HIV specific antibodies, right? So onto this the antigens from the blood will be coming and binding. Again, we will not be able to see it, which is why we will need another antibody out there which is also pre-prepared. Now this antibody which is there, my dear children, it will be linked to an enzyme. It will be linked to an enzyme. Okay, now this antibody comes, it is linked to an enzyme. Now I want to see whether the actual binding has taken place. What do? I'll be putting some coloring substance. When I put the coloring substance, if at all the enzyme reacts to it and gives a color change, that means that whole of this reaction has taken place. That means that the person has HIV/AIDS. You get it? So it is dependent on antigen antibody binding principle. Okay, now we come back to this question and we fill in the missing data which is there. Now this time you have to try. Retrovirus came. Virus infects a normal cell. Okay, it infected a normal cell. Now viral RNA is introduced into the host cell. Agreed. It is now inside the host cell. Reverse transcription absence. After that, what happens, my dear children? After reverse transcription, what do you get? Viral DNA is going to be formed over here. That is the next step. Now viral genetic material in the nucleus of the host cell, as in the viral DNA is now inside the host cell. What will happen? The viral DNA incorporates into the host genome, and after that what will happen? After it incorporates, it will give information to form the viral protein as well as the viral RNA. So viral protein and viral RNA is being formed. The viruses comes out of the host cell because everything is now assembled, and that's how it goes. So if at all you know the flow, you will be able to write the answer. If not, you will not be able to write the answer. So viral DNA is formed, that is the first one. Next thing, viral components such as proteins and RNA is being synthesized. Then similar to that, we have one more question. Now this is all from the question bank, and they have been put under the 5-marker category, which is why I thought I'll explain it to you. Okay, so a person has been diagnosed as HIV positive. Okay, they have been diagnosed as HIV positive. Name the diagnostics test. It is ELISA. Now write the full name of the pathogen: human immunodeficiency virus. Human immunodeficiency virus. Now name the genome of the pathogen. What is it? What is the genome? Single-stranded RNA, right? Single-stranded RNA, or so you call it as ssRNA. Now name which particular cells of this person are likely to get destroyed. What is the cell that are going to be likely to get destroyed? It is first macrophages, but my dear children, ultimately it is T helper cells. Ultimately it is T helper cells. cells. So even though if we do not write this, at least write this. At least write this. This is going to be the major target. This is the one which is why we have less immunity, because this guy is being targeted. Macrophages, initially they are being attacked, and they are basically, we say they are factory of HIV. Why? Because HIV viruses, they are going to replicate the number over here and then attack the T helper cells. But if at all you get a question like this, I will prefer you write this first. You write this first, and probably you can write macrophages, and bracket you can write factory of HIV. That's all. First this, okay? Now suggest a partially effective treatment to treat him. So HIV, do you think HIV/AIDS has a treatment? Okay, there is no fully curative treatment that has been found out until now. Okay, there are things that are ongoing, but generally the partially effective method that we have is having antiretroviral drugs. Okay, so antiretroviral therapy is something that we have, because it's a retrovirus, so against it we have antiretroviral therapy. So you can just remember that. I hope that works. Yeah? Okay, perfect. Now we'll come back to the questions that we were doing. So yes, we were here. Differentiate innate immunity innate immunity and acquired immunity. First question, innate and acquired. Innate means what? Something, immunity that you have from your birth, and also it is not specific. So example, when I say that I am born with skin, this is an example of innate immunity. I am born with skin, and the skin prevents entry of anything inside my body, right? And do you think it's specific, such that do you think it will allow a virus, you bacha, you come in, and bacteria, you do not come in? Is it like that? Is it specific? No. It's the same for all. It acts the same for all of them. So this comes under what? Innate immunity. Basically, you need to remember they are from birth and they are non-specific in nature. Okay. Under this, you have multiple barriers. This is your homework. Please go and study the different barriers of innate immunity. Very, very important question. Physical barriers, as in your skin as well as your mucus, and then you have physiological barrier, for example, HCl being secreted or your tears being secreted. All of them, they come under what? Physiological barriers. Cellular barriers, for example, you have macrophages, you have polymorphonuclear leukocytes, that is known as neutrophils, and all of them. Okay, natural killer cells. Now you have cytokine barriers, as in cells releases some things which are non-specific. It is known as, in example is interferons. All of them, please go through it once. Okay. Now acquired immunity is something, it's not that we get from birth, we acquire it during our lifetime, which is why you call it as acquired immunity. And this is going to be super specific. It is going to be super specific. For example, I tell you that in our body, whenever some antigen comes, okay, whenever we say that, okay, I've got an infection, when the antigen comes in, my body is going to react to it in two ways. One is, it will be showing humoral immunity. The second thing it will show is cell-mediated immunity. Okay, humoral immunity, as in, basically it is going to release, or basically I can say humoral immunity is the when, basically, seeing a pathogen, my body will be producing some B lymphocytes. And this B lymphocytes in turn are going to produce two other things. One is going to be the antibodies, and the other one which is there is going to be some memory cells. So antibodies and memory cells are being produced. These antibodies are going to target the antigens and are going to target to destroy them. What about the memory cells? Memory cells are produced in a way so that it has an idea. Next time the same antigen comes in, I have to be more careful. I have to produce the antibodies in more time. So this might be for the first time it might be seeing an antigen out there, right? So obviously the first response, the primary response over here is going to be less. But the next time the same antigen comes into our body will react. So which is why every time when we have some infection for the first time, it will react very badly. The second time the same thing comes on, our body knows how to tackle it. Why? Because there is memory cells being produced over here. Why do you say that this is humoral immunity? Because, my dear children, B lymphocyte, which is a type of a white blood cell, it is going to release antibodies directly into the blood. And blood is what? A fluid. Blood is what? A fluid. And you also call this fluid as humor. That is the interchangeable term that is there. Humor. So since in your humor or basically in the blood you have antibodies floating around, and that is there for trapping the antigens. You call this type of immunity as humoral immunity. This is one type of acquired immunity. Didn't you acquire it during your lifetime? Yes, you acquired it, right? You acquired it during your lifetime, you got some infection, and that is when all of this thing started happening. Similarly, you have cell-mediated immunity. Here in cell-mediated immunity, the major guy that plays a major role is T lymphocytes. T lymphocytes. Now over here also, whenever it comes across some antigens, it is going to tell the other cells, hey, you have to be active, you have to go kill the antigen. Such as it will signal the B lymphocytes to produce antibodies, or probably it directly can go and kill the cells. Anyway, that can happen. Apart from it, it will be also releasing one very important thing, that is memory cells. Again, memory cells, such that the body has an idea. So one thing that is common in both of them is that memory cells are being produced. That means the body has a memory of it, that something has come. The next time you have to be very, very careful out there. Got it? So this is humoral immunity and this is cell-mediated immunity. This comes under acquired immunity, and this is very specific to specific antigens. Specific type of antibodies are going to be produced. A specific pathogen, specific type of T lymphocytes are going to go act. Why it is known as cell mediated? Because over here T lymphocytes mediates the other cell, or itself goes and kills the antigens out there. So there is cell to cell interaction out there, which is why you call it cell mediated immunity. I hope you got it. Now if at all you had to give difference between, so you have to say innate immunity is present from birth, acquired develops after an infection or basically during a vaccination. Vaccination is what basically you get the weakened terms of pathogens, weakened type of pathogens, and that is being put inside your body such that it can ignite a response, right? So either an infection or a vaccination. And here in innate it is non-specific, but here it is very specific to particular antigens. Here there is no memory, here there is immunological memory. So example is skin, phagocytes as in your macrophages and things like that. Over here you have antibodies, T cells as an example for it. Cool. Now you have the next question, which is refer to the below given diagram and answer the question. First of all, the diagram of antibody is super important. You should be knowing it. This is an antibody. Okay. Over here, this long chain that you see, this very long chain that you see over here, these chains are known as what? Heavy chains. What do you call it? Heavy chains. So when I say H2, that means two heavy chains. H2 stands for two heavy chains. Okay, what about L2? L2 is basically these two light chains out there. These two light chains out there, they are known as what? Two light chains. Got it? This is super important. Now over here, my dear children, you should also be able to see that there is a binding site. Binding site for what? Antigens. Antigens are going to come and bind over here, and that is how the whole reaction is going to take place, right? So this is the antigen binding site. A and B is what? The antigen binding site. Do not forget it. Very important. So name the molecules trapped in the lymph nodes responsible for the activation of lymphocytes that bind to A and B site. What is the one that is responsible for activation of lymphocyte? It is going to be the antigens, and they bind at the A and the B site. Works? Works, ma'am. Perfect. So how do you write the answer? You write the answer like this. Okay, make sure you write it. Mention the cells involved in accomplishing the acquired immunity. It is B lymphocytes and T lymphocytes. B does humoral immunity, T lymphocytes show cell-mediated immunity. Then there's a question. If at all I have an organ transplantation, okay? You might have seen in most of the cases the organ which is transplanted is getting rejected. You call this as graft rejection. Who is responsible for that? B lymphocyte or T lymphocyte? It is mostly T lymphocytes which is responsible for that. Cell-mediated immunity is responsible for graft rejection. Graft rejection. And since we're talking about antibodies, also remember antibodies are also known as immunoglobulins, IG. That is why you say IgA, IgG and things like that. IG stands for what? Immunoglobulin. Antibodies that is being produced, it is nothing but proteins. It is nothing but proteins, or basically polypeptides. Polypeptides come together to form proteins. So please know that. And how many antibodies do we have? You can remember it like this: GAMED, gamed. IgG, IgA, IgM, IgE, IgD. The first antibody which travels from the parent, that is from the mother to the child, through placenta is IgG. That is why first position. Now the second antibody that travels from mother, okay, mother's milk to the baby, from the colostrum, it is IgA. Okay, so you can remember IgA, A for Amma. A for Amma. So this is passed through colostrum to the baby. Now M function is not given, D function is not given, E is given. E is there for allergy. E, allergy. Please remember it. Okay, and if at all, whenever something comes in and you feel that, okay, I do not know about it, go refer your NCERT. This is very, very important. Okay, now name the cells involved in the humoral and cell-mediated immune response out there. So in a question paper, instead of C question like this, they have put another question. Name the cells involved in humoral and cell-mediated. So you should know. In humoral, it is carried out by B lymphocytes. They produce antibodies, and also they produce what? Memory cells. These antibodies bind to pathogens and neutralize them. Okay. So please remember, they produce antibodies and also memory cells. Okay. Now cell-mediated immune response, this response is carried by T lymphocytes. No antibodies are produced over here. Instead, T cells directly attack and destroy the infected cells or the cancer cells, or signals other cells, or signals the other cells, and also produces what? Also produces memory cells. So please know about all this thing. Works? Works, ma'am. Perfect. Okay. Now this question we solved. We'll come to, I think, the last question of this chapter. Fill in the missing data. Escheriasis causative organism is what? Ascaris. What are the symptoms of this Escheriasis, my dear children? Escheriasis. Escheriasis is basically caused by Ascaris lumbricoides, which is a roundworm, and it affects your intestine. It causes intestinal blockage. This is the first point that should be coming to your mind. Since there is a blockage, always remember, and also this particular guy goes and perforates our intestine, which is why there will be internal bleeding. Okay, intestinal, first of all, intestinal blockage. Then you talk about what? This thing, intestinal blockage. After that, internal bleeding is something that you have to say. And since internal bleeding is happening, you will be suffering from anemia as well. So will you be able to write that? Please write it. Okay, internal. So I'll write it over here. I think that will be good. Internal bleeding, intestinal blockage, and then anemia. At least these points you have to write. Now what do you have? You have name of the disease you have to tell. Trichophyton is the causative organism, and basically appearance of scaly lesions on the various parts of the body is there. Scaly lesions of the body leading to itching. Trichophyton, epidermophyting, fighting for what? Microspore, small spore. They are all fungi that causes ringworm disease. It is not caused by any worm, it's caused by fungi, but it looks like a worm because the structure is like this on the skin, right? So I'll say it is ringworm disease. Okay, see question. Typhoid causative organism is dash, and it has high fever, weakness, headache, stomach pain, and constipation. So what causes typhoid? The bacteria whose name is what? Salmonella typhi. Salmonella typhi. Now what do you have? Pneumonia. Pneumonia is caused by Streptococcus pneumoniae or Haemophilus influenzae. What is the thing that you have? Pneumonia is the place where your lower respiratory system is going to be affected. That is means your lung is going to be affected. In your lung, what is being affected? Basically your alveoli. Your alveoli, which is there, which is responsible for exchange of gases, that is being filled by mucus, which is why there is no proper exchange of gases. So can I say that fluid filled alveoli is seen over here, and the symptoms, if at all you go talk about, so there will be what symptoms if at all fluid filled alveoli is there. So this is not a symptom. The symptom you have to write, right? The symptom will be breathing difficulty. You will have fever, and everything, very common thing is what? Fever. Apart from breathing difficulty, fever, what do you have above? You might have cold, or basically you can just write cough. Apart from that, what do you have? You might have headache also. Okay, if not headache, if you are very scared for writing headache right there, because some things are common between common cold and this, you can go with the extreme character. That is what? Your nails and lips turn what? Bluish gray in color. So please write all this thing and you'll get your marks. Okay, so each and every part of this particular table is important. You can get similar type of questions also, so please know about all the diseases. Now the one that I have got is right over here. E is a disease. It is caused by rhinovirus. It causes nasal congestion, discharge and sore throat, okay, and also cough and headache. So what is this thing? It is going to be common cold. This is going to be common cold. So I think I have got this or something else. Okay, so these are things that you have to write. I hope this is clear to you. If at all until now it is super clear, definitely let me know and let me just drink some water and I'll be right back. Okay, and we'll go on to the next chapter. That is, okay, first of all, evolution, there is no 5 marker, which is why it is not going to be discussed over here. Microbes and human welfare also, there is no 5 marker. There's only 2 and 3 marker. We have discussed it already. Biotechnology principles and processes, my dear children, again you have 5 markers, so we have to start discussing this. So I'll just drink some water and I'll be right back. Okay, perfect. Yes, so explain the process of preparation of competent host for transformation with recombinant DNA. Listen, competent host means a host which is ready to take up the recombinant DNA. So what is the process that I will be utilizing for making a host competent, for being ready? So in the case, this is usually discussed in the case of bacteria, and I'll tell you in bacteria what you do is, suppose you have the organism up there, and basically in this medium you also put the DNA which you want to insert, right? And this medium is going to be a medium that has calcium in it. Why calcium? Why calcium? That is the first and the foremost thing that you should be doing. So okay, so bacterial cells are treated with divalent cations, as in basically CaCl2, to make them competent. Over here, when I say divalent cation, you should know the charge is over 2+. That is why I call it divalent cation. It is calcium. The calcium ions are going to neutralize what? The negative charges. On what? On the DNA and also on the cell membrane. Listen, the DNA and the cell membrane, both the charges have to be neutralized. Otherwise, do you think they will get inside? No. Basically, they will repel. There is no chances of the DNA passing through it. So we have to make sure that the neutralization happens, and that is one major role of Ca2+ ions, neutralize the negative charges on the DNA and the cell membrane. And also need to understand the second function of calcium, and that is it increases the permeability. That means it increases the permeability. There will be some pores in the cell membrane that is created because it is being treated with the calcium ions out there, right? So now there are some pores over here. Negative charges is also, it is neutralized over here. Everything is okay. There are few pores over here. The next thing that you have to do is, first of all, you have to keep in a medium that is having ice. Okay, now you shift the thing into a hot medium. That means basically you are giving a heat shock. You are giving a heat shock, and then again what do? You treat it back with ice. So the thing is that whenever you do that, there is basically opening of the pores very much, okay, and there will be closing of the pores when you treat it back with the ice. So since there is temperature change out there, the permeability of the membrane is going to be changed, which is exactly why whenever it opens, the recombinant DNA is going to get inside, and basically when I put in the ice, it will be closed, which is why it cannot move back outside. You get it? So this is the thing that we generally use. So the mixture of cells and the DNA is then put into ice, then given a heat shock, 42 degrees Celsius for 90 seconds, just for 90 seconds, and then back cooled on ice. This allows the recombinant DNA to what? Enter the bacterial cell and not to move out as well. This is how you make the cells competent generally. Okay, this is how you make a bacterial cell competent. Works. Now, next thing is that mention the restriction enzyme that can be used for insertional inactivation of antibiotic resistant gene over here. So, oh my dear children, listen to me. This is what? This is a plasmid, right? This thing that you see is a plasmid. That is PBr322. PBr322 has an ORI site, that is origin of replication. Apart from that, it has ROP site. This protein is super important for the replication process to happen. Always remember that. Okay. Now ORI ROP site is done. Here you have 2 sites. If it has this gene, it allows it to be resistant to ampicillin, as in basically I can grow an organism that has PBr322 and it will still survive. Why? Because it has a resistant gene, ampicillin resistant gene. Ampicillin is what? An antibiotic. So if when I have a gene against it, is basically like having a superpower against it. So this is ampicillin resistant gene. This is tetracycline resistant gene. This is another antibiotic resistant gene that is out there, right? Okay. Now listen to me. these are enzymes. These small things that is seen over here, these are enzymes, and these enzymes have specific site. That means these are the places where they will make a cut, and if at all needed, they will, whenever needed, we can insert the DNA at this particular site out there, right? So PVU2 has a site in the ROP site. BAM-H1 and SAL1 have a site over here. So if at all I am cutting anything with BAM-H1, I'll make a cut over here, I'll insert my new gene over here. When I insert my new gene, my dear children, do you think it will be tetracycline resistant anymore? No. Why? Because at this place I am inserting something else. I have lost the older gene which was making the cell resistant to tetracycline. Now is it tetracycline resistant? The answer is no. So if at all I want to make sure that the plasmid does not have tetracycline resistant, what do I have to do? I basically have to cut this particular plasmid with either BAM-H1 or SAL1. Now, suppose I do not want the organism to have ampicillin resistant. Again, what do? I can cut it with PVU1 or PST1, because anyway if I make a cut over here and I insert a gene over here, it would not have the older gene which is ampicillin resistant. This process of inserting something and inactivating the other thing out there is known as insertional inactivation. What do you call it? Insertional inactivation. I'm inserting something, which is why something else is getting inactivated. So if at all I want to inactivate ampicillin resistant, I have to use either PVU1 or PST1. If at all I want to inactivate tetracycline resistant, then I have to use either BAM-H1 or SAL1. These things are something that you have to remember. Now there can be a question, that is, basically I do not want to inactivate any antibiotic gene out there. Which are the sites that I can use? I can use EcoR1 site, I can use CLAV1 site, I can use HIN3 site, because they are not coming under what? Any of the resistant genes out there, right? So EcoR1, CLAV1, HIN3 and also PVU2 But if at all you're cutting with PVU2, you will not get replication protein being made, and that is super important for replication process to happen. So please know that. Okay, so your question is, first question is, mention the restriction enzymes that can be used for insertional inactivation of antibiotic resistant gene. So what are the restriction enzyme that I can use for insertional inactivating these two resistant gene out there? I can use PVU1, PST1 for ampicillin resistant, okay, BAM-H1 and SAL1 for inactivating tetracycline. This is your answer. So exact thing is written over here. Got it? For that you need to know the location. We will draw it once and we'll remember it. Okay. Now one more question is there. How many cloning sites are present in the given PBR322? As in, how many sites for the enzymes are present in PBR322? So how many sites are there? Let's see. 1, 2, 3, 4, 5, 6, 7, 8. 8 cloning sites are there, and you can just write it. Number of cloning sites in PBR322 is 8. 8 unique restriction sites are there, and these are the restriction enzyme sites. Got it? Please write this. And since if at all you are getting confused by any chance, I can draw it and I can help you remember this. So this is your PBR322. Here what do you have? You have origin of replication. Okay, here what do you have? You have ROP site. Here what do you have? You have tetracycline resistant gene. Here what do you have? You have ampicillin resistant gene. Done? Okay. Now we start making things. Here, my dear children, you have PVU2. What do you have? You have PVU2. Here you have BAM-H1. Here you have SAL1, 1. BAM-H1, SAL1. Here you had PVU2, right? Here you have PVU1, and here you have PST1. PVU1, PST1. Okay? 1 here, 2 of them here, 3 will be over here. So the first one that you have is EcoR1, which is very famous restriction enzyme. Then you have a dundee like this is known as CLAV1. Then you have something like this is known as HIN3. So listen to me very, very carefully over here. 1, 1, 1, 1. Everything is 1. Here it is 2. Here, there it is 3. Okay, 1, 1, 1, 1, 1. But here it is 2 and there it is 3. Here you just have 1 restriction enzyme site. Here you have 2. Here you have 3 of them. 1, 2, 3. You can remember like that also. This is super, super important for you to know. And remember, an enzyme will be having only 1 site over here. Okay, a particular restriction enzyme will be having 1 cloning site. Multiple restriction enzyme can have multiple cloning sites, as in basically when I say PVU2, do you see PVU2 elsewhere? No. It will only make a cut over here. Suppose you had a PVU2 site elsewhere also. What is going to happen? It will make a cut and will destroy the whole plasmid out there. We do not want that to happen. So per
per restriction enzyme, only one cloning site is there. But in one particular plasmid, you can have multiple cloning sites of other restriction enzymes. Please know that also. Done? Done, ma'am. Perfect. Clear. Let's go ahead. Now, give reasons for the following. You have to give reasons. Restriction enzymes are called molecular scissors. Why do you call them molecular scissors? Restriction enzymes, these enzymes that we saw, you call them molecular scissors because they are going to cut your DNA. There are two types of restriction enzyme. One is endonuclease, one is exonuclease. Endonuclease goes within the DNA strand and makes a cut in the middle. Exonuclease cuts towards the outside. Anyway, isn't it cutting the DNA? Yes. Which is why you call them restriction enzymes as molecular scissors. So, because they recognize and cut the DNA at specific base sequences, that is recognition sites, just like scissors cut at precise points, you will call them as molecular scissors. And also, since they are cutting in the big molecules such as DNA, RNA and things like that, that is why you call them molecular scissors. You please write that also. Okay, now DNA ligase is used in genetic engineering. Why is DNA ligase being used? So when I make a cut and I put this particular gene somewhere else, do you think it will slowly fit in nicely? Suppose I've cut this particular plasmid and I am inserting a new gene over here. Do you think it will perfectly stick? The thing is that you will need something to glue it up really tight, and that is the role of DNA ligase. It is there for ligating things. Okay, it is there for ligating things. So DNA ligase is used in genetic engineering because it joins the two DNA fragments by forming what? Phosphodiester bonds. It helps in the formation of phosphodiester bonds between their sugar and phosphate backbones, helping to seal the foreign DNA into the vector. So these key points are super important. Over here also, why they are known as molecular scissors? Because they cut the molecules. Molecules, big molecules such as DNA. Also write this, very important. Here they form the phosphodiester bond, sealing the sugar phosphate backbone out there, right? Next thing, chilled ethanol is used in DNA isolation. So suppose, my dear children, I want to isolate the DNA from a cell. What do? I take a test tube over here and I put chilled ethanol. In this chilled ethanol, your DNA will be what? Precipitating out, or basically moving out, separating out. That is why I used chilled ethanol in DNA isolation. Okay, so because DNA is insoluble in cold ethanol, so you have to explain like this. It is insoluble. Okay, DNA which is there, it is insoluble. And since it is insoluble, do you think it will mix up? No, it will not mix up. Rather, it will be precipitating out from the cell extract, making the DNA visible and easy to collect. So this point is important over here. DNA is insoluble in cold ethanol. Works. Okay, smaller DNA fragments move farther in a gel slab. So if at all you have seen gel electrophoresis, my dear children, it goes like this. You have wells over here, you have cathode over here, and you have anode over here. Cathode and anode. Always remember, cathode is negatively charged, anode is positively charged. Anions are negative, but anode is positive. Always remember. Your DNA ends with A, so it moves towards A. You can remember like that. DNA is also negatively charged. Negative will repel. What will happen? They will separate. The smaller ones which are there, they move the farthest. The larger ones which are there, they will move slowly. So who moves the farthest? The smaller one. Why? Why do they move faster? Because they will be having less inhibiting effect. Always remember, in gel electrophoresis, you use agarose gel. And the DNA is not going to float around the agarose gel, it is going to show the sieving effect. It moves within the agarose gel. Sieving effect is something that you should not be missing, right? When it shows the sieving effect, it gets the least hindrance. It will be able to move very easily. That's the reason why smaller fragments would be able to move faster, or basically farther. So because during gel electrophoresis, smaller fragments face less resistance from the gel matrix and therefore migrate faster and farther towards the positive electrode, that is anode. Now one more very important thing. Who moves first? This is also a question. So sometimes we confuse this. When I say smaller fragments move faster, people confuse it. Does it move first? So from the well, it can be either the longer fragment or basically the shorter fragments that can move first. We never know. It can be anything. So if at all the question says, the first one to move is a smaller fragment, there is no clarity regarding that. The one that moves the farthest is basically the smaller fragment. That is something very sure of. Cool. Now, thermostable DNA polymerase is used in polymerase chain reaction. Thermostable DNA polymerase, as in a DNA polymerase which can resist high temperature, is being used in polymerase chain reaction. Why do you think so? Because in polymerase chain reaction, my dear children, there is denaturation. Your DNA, when you say that you have to replicate the DNA, first of all, you have to separate them. And for separating them, you need a degree that is 95 degrees Celsius, which is too hot. Okay, after denaturation, basically, I have to do the annealing process. It also happens at high temperature, around like 55-65 degrees Celsius, which is lower than this, but yes. And then elongation, which again requires, elongation of a new strand, again requires almost 75 degrees Celsius. Now, for making a new strand at this particular temperature, obviously, you have to make sure that the DNA polymerase used is what? Thermostable. It can resist high temperature. Okay, that is the reason. So because enzymes like Taq polymerase, as in the DNA polymerase which you extract from Thermus aquaticus is known as Taq polymerase, can withstand high temperature used to denature the DNA strands, allowing the PCA cycle to continue without the enzyme breakdown. Okay, so you have to make sure the enzyme does not break down at these high temperatures out there. It's not just for denaturing the DNA strands, but also for the process of extension and everything, you need high temperature, right? And you have to make sure that the enzyme does not break down during these conditions. Now, here I have a picture so that you can remember things a lot better. This is chilled ethanol and your DNA is insoluble, which is why it is moving out, okay? Here, gel electrophoresis, the smaller fragments, if at all you're able to see, they move the fastest. Here, this is DNA ligase. It's like a glue. It glues up the things by making the phosphodiester bond. Cool? Cool, ma'am. Perfect. Next. Genetic engineering is a core technique of biotechnology. In the background of this, complete the following table. Okay, let's complete the following table. Over here, what do you have? Gel electrophoresis dash visualization of the DNA fragments. Here, the things which are used are being written. So, gel electrophoresis, in gel electrophoresis, what do you use such that the DNA becomes visible to you? You use a strain which is known as ethidium bromide. What do you use, Baba? You use ethidium bromide. So please write that, ethidium bromide. Now next thing, dash DNA polymerase is being used and dash is the technique's name. This is going to be the technique's name. Okay, no, this is going to be the technique's name. Over here, gel electrophoresis, gene gun and everything is written. So this is going to be the technique. This is going to be the use of it, right? The use of it. So basically, what is the technique where you use DNA polymerase? You are going to basically call this technique as polymerase chain reaction PCR. Okay, what do you get after the end of it? You get amplification of the DNA, right? Your DNA is going to get amplified. So amplification of DNA. When these type of question comes, read to the entire question and understand what is being asked over here. Do not make it ulta sida. Okay. Now gene gun method is there. Gene gun is basically a vectorless method. A vectorless method for transferring DNA inside what? The plant cells. Inside the plant cells. How do you transfer it inside the plant cells? So plant cell has a cell wall, which is why you use a gun over here. Otherwise, you could have just injected, just like animal cells, you do micro injection. But in plant cell, you have gene gun, where what do you take? That is the next question. What do you take over here? What is the thing that you use over here? You take, okay, you take particles of gold and tungsten. Particles of gold and tungsten. And what do? You wrap it around with the DNA. You wrap it around with the DNA that you have to insert. What will happen later? Transfer of recombinant DNA into the host nucleus will be happening up there. Gene gun is also known as biolistic method. Please do not forget it. This is found only in the case of a plant cell. Only in the case of a plant cell. Got it? Got it, ma'am. Now, isolation of DNA is there. What do you use for isolation of DNA? That such that it precipitates. It is chilled ethanol. So E is going to be chilled ethanol. Got it? Perfect. Okay. So exact thing I have written over here. You can just go through it once more. The next question that we have is, given below are the diagrams of plasmid A and B. This is plasmid A and this is plasmid B. Now observe meticulously and answer the questions that follows. Okay, let's see. So this is PBR 322 and this is another plasmid which is given in NCERT, like Z gene, okay, blue-white colonies, you might have heard about it, right? Now, which plasmid is or are you going to select for the cloning and why? For cloning and why? What do you think is the answer for this? Which one will we use for cloning? Listen to me very carefully. I will be using plasmid B for cloning. The reason is very simple. in plasmid B, suppose the LAG-Z gene over here, I am inserting something else here at this particular position. What is going to happen? LAG-Z gene is not going to produce the same thing that it was producing earlier. What was it producing earlier? It was producing beta galactosidase. What was it producing? It was producing beta galactosidase. Now this beta galactosidase, which is usually being produced, what does it do? If I add a chromogenic substance as in a coloring agent, this beta galactosidase will act on it and give a blue color thing, Now suppose I am inserting some gene over here. I am inserting my gene over here, and then to check whether the plasmid has got the gene or not, it has become recombinant or not, what do? Basically, again, I'll put the chromogenic substance. If at all there is no color change happening, I'll just easily get to understand that that over here I have got my gene being inserted, because there is no color change. Otherwise, it would have given me a blue color thing, right? Over here, the steps are pretty much very long, and I'm going to explain that, do not worry. So the thing that I will prefer is plasmid B, because it is very easy for me to use. Plasmid B is selected because it has LAG-Z gene, which helps to easily find the recombinant colonies. When a foreign DNA is inserted into the LAG-Z gene, the gene becomes inactivated. What happens? It becomes inactivated. This allows blue-white screening. The white colonies contains recombinant DNA. The blue colonies contains the non-recombinant DNA. So basically, it's very easy for me to understand. Now, what is insertional inactivation is the next question that is given. Insertional inactivation is common for both of them, where what is happening? It means the gene stops working because a foreign DNA is inserted into it. Now, if you want to give an example, you can give. Example, in plasmid B, when LAG-Z gene stop producing the enzyme beta galactosidase, after insertion of what? The foreign DNA out there. That is why I get the white colonies, right? Right. Now, will the number of culture plating be same or different over here? Comment. What do you mean by that? So here, my dear children, where is it? So here, in this particular case and this particular case, the number of plating that you require for finding the recombinants, will you think it will be the same or different? That is a question, and we have to find the answer. For this, you need to have patience. For understanding this, you would require patience, and also this is a very important concept which is being asked, and you guys mostly get confused between this. So make sure you listen to me very, very carefully. So it goes like this, okay? This is the condition for plasmid A, and this is the condition for plasmid B. So I'll make the plasmid B first, okay? In plasmid B, my dear children, suppose here you had LAG-Z gene, okay? And here, now in this particular case, what do you have? You have the gene of interest. So this is going to be what? My recombinant DNA. This is not a recombinant DNA. Now what do? Basically, I put this inside a cell. Similarly, I put this also inside a cell. Later, I have a plate right over here, where I am going to add the chromogenic substance. Chromogenic substance. Okay. Here the cells which have the plasmid, the older plasmid out there, it will look like this. And here the cell which have the new plasmid, it is going to look like this. But can I identify just on the basis of the looks? No, obviously it is not visible to me. DNA, how will I get to know that? So over here, when I put the chromogenic substance, the thing is that this one will be turning blue in color, whereas this one will not have any change. It will be basically white in color. I say colorless. Over here it is blue. Why? Because still LAG-Z gene produces beta galactosidase, and it is going to act on chromogenic substance, which is why I get blue color colonies. How many plates do I require? How many number of plates did I require to basically find the recombinant DNA? Only one plating. In one plating itself, I got to understand that these ones are the white ones. Basically, it has a recombinant DNA. And this one is what? The non-recombinant. So this has been very, very simple for me. Now, let's talk about plasmid A. In that, my dear children, first thing that you need to realize is that you have to, so there is one cell over here, there is another cell over here, and there is one more cell, suppose. Okay, this cell, or basically I'll say, okay, this is ampicillin and this is tetracycline resistant gene. Okay, this is another plasmid, and this plasmid has ampicillin over here, and the blue one is the gene of interest. So my first task is to find the transformants, or basically I'll write in yellow, it will be easy for you to understand. The first task is to find the transformants, as in basically a cell did not have this plasmid before. Okay, I have to make sure that they take up the plasmid. How basically I do the competent DNA technique by doing heat shock and this and that, calcium and stuff. So some might have taken, some might have not taken the plasmid, some have not taken, some has taken. We know because it is clearly visible, but how do we get to understand when we're doing it under the lab? We cannot see the DNA out there, right? So for that, the first plating we do is with ampicillin. We do the first plating in ampicillin. Suppose when we plate it with ampicillin, this is not be able to survive. Why? Because this does not have the plasmid out there. It does not have a normal plasmid. It does not even have the recombinant plasmid. Listen, some plasmid might have taken up the gene of interest, some plasmid might have not taken up the gene of interest. Anything can go inside the cell. It can be this or it can be this also. Okay, so suppose over here it has not taken any plasmid at all. So this is going to be what? A non-transformant. This is not transformed. Transformed means basically to take up something new. It has not taken anything new. Okay. Now over here, these two are transformants. How do I understand this? Because this can survive and this can survive. Why can they survive? Because it has ampicillin resistant gene over here also. So what do I get to understand? That these two things which are there, these two cells are transformant and this one is the non-transformant. This is going to be the first plating. The next plating that I'm going to do is with, wait. So I have these two guys now, so I have to make that diagram. Let's do that first. Here, this one is this. Here, this one is this. Cool. This is my gene of interest and this does not have it. So basically I can clearly see and as of now tell that this is the recombinant cell with the recombinant DNA and this is the non-recombinant. Both of them are transformant because both of them got the plasmid which was initially not there. Both of them are transformant, but this is non-recombinant because it does not have the gene of interest and this has. Again, how do I identify it? The next plating that I am going to do is going to be in tetracycline medium. When I do in tetracycline medium, what is going to happen? This is second plating, okay? This is the first plating. This one will keep on increasing their number. Why? Because, ayyo, did it go? My God. It went. Okay, anyways, that one which was there, it will be, okay, calm. This one which was there, this one was non-recombinant. Why? Because it does not have this. It has the recombinant DNA. When I grow it in tetracycline medium, which one is able to grow? This one. Why? Because it has a tetracycline resistant gene. This one will not be having. So which one will I select? This one is something that I will be selecting. The one which is not able to grow is the one which has my gene of interest. It is not able to grow because it is not having tetracycline resistant. It has become insertionally inactivated. So this is my thing that I would take. You get it? For this you require 2 plating, whereas in the other case you just required 1 plating out there. So will the number of culture plating be the same or different? Comment. The number of plates will be different for both plasmid. For plasmid A, ampicillin tetracycline needs 2 plates, one with ampicillin for finding the transformant, such as who has taken up the plasmid, and one with tetracycline to find which one has taken up the recombinant plasmid, right? For plasmid B needs only 1 plate with X-gal. X-gal is the chromogenic substance which is there. Okay, X-gal is the chromogenic substance onto which beta-carotidase is going to react. So white means it is recombinant, that means it has been insertionally inactivated, there is no production of beta-carotidase. The blue means it is non-recombinant. Hence, plasmid B is better because it allows easier and faster selection of recombinants in just one step. Got it? That's all. Perfect. Now, we go over here. Study the diagram given below and answer the question that follows. What is EcoR1? What is EcoR1? It is a restriction enzyme. It is a restriction enzyme. And how did it get its name? Listen, EcoR1 stands for Escherichia coli, E. coli basically. The first letter of Escherichia is being taken, that is E. Coli, 2 letters. The species name, 2 letters have been taken, that is by CO. And R stands for the name of the strain, and 1 stands for the number of discovery, on the basis of number of discovery. So what is EcoR1? It's a restriction endonuclease. Endonuclease is something that will go cut in between, okay, in between the phosphodiester bonds within the DNA itself. So EcoR1 is a restriction endonuclease enzyme isolated from Escherichia coli. It recognizes specific palindromic sequence. It has a specific site where it will go and make the cut. What is the site? 5-GAATTC 3-. 5-, this is very important. GATTC, okay, it is 5-GAATTC 3-. This is the site that we have over here. And it cuts between what? Cuts between G and A. Here, this is the site. Here it makes a cut between G and A, and it's a palindromic sequence. Why? Because in a DNA, suppose I have GAATTC over here on the other side. This is 5-, 3-. Similarly, from here also, it will be GAATTC 3-. Always remember, this is super important. It's in 5-, 3-. Do not make a mistake when there. And it cuts between G and A. So it makes a cut over here, it makes a cut over here. Now, how is the action of exonuclease different from that of endonuclease? I told you, exonuclease are going to cut the DNA from the ends, whereas exonuclease cuts the DNA from the end. Endonuclease cuts the DNA from within this DNA itself. Okay, it removes nucleotides one by one from the terminal ends. It breaks DNA at specific recognition sequence. So first thing that it does is that it goes on and search for the recognition sequence, then makes a cut out there. Example, enzyme that trims the DNA ends. Example, EcoR1, HIND3 and things like that. Which one was the first restriction enzyme to be found? The first restriction enzyme to be discovered? It was HIND2. It was HIND2. HIND3 is found in PBR 322. HIND2 is the first one. It also have a recognition sites of 6 base pairs, just similar to this. Okay. In E. coli also you found the restriction enzyme, but you could not discover it completely. You just knew that there was something out there. But the first one to be discovered was HIND2. Please know that. Now, how are sticky ends formed on the DNA strand and why are they so called? Sticky ends are found when a restriction enzyme such as EcoR1 cuts the DNA. Okay, I'll just show it to you. So, over here, I had the picture, right? Why do you get sticky ends? So, suppose, my dear children, you have the vector DNA, as in the plasmid DNA. Okay, suppose the vector over here is the plasmid. And you have your foreign DNA. Now, these guys have to come over here. That means this side and this side should come. This side and this side could come in contact, right? So, what do? We cut it with the same restriction enzyme. Plasmid, I am going to cut it with EcoR1. And in plasmid, GAATTC site is being searched. It makes a cut over here between G and A. It makes a cut over here between G and A. Okay, done. What is going to happen? This L-shaped thing is going to go. This L shape, when I make a cut over here and I make a cut over here, this L-shaped thing is free over here. It's not attached to any sort of DNA. So this one will go. I'll get something like this, right? This is my vector DNA, as in my plasmid DNA. Similarly, in my gene of interest also, I made a cut using the same restriction enzyme. Here also you had GAATTC site, GAATTC site. Again, it cuts between G and A, G and A. The one which is not attached to the plasmid, or basically not attached to the DNA, is being cut. The L-shaped thing is being removed. Now, when I put one end and another end of the vector, this is the end of the plasmid, this is the end of the foreign DNA, and I keep it close. What is going to happen? They are going to be base pairing to each other. Why? Because they are complementary to each other. They are waiting for a hydrogen bond to be formed, and since both of them come together, you get everything fine. Now, for the last step, you need DNA ligase, but sticky ends formation is super important over here, which is why we cut the plasmid with the same restriction enzyme that we use for cutting the foreign DNA also. So sticky ends are formed when a restriction enzyme such as ecoarvin cuts DNA staggeredly between the G and A. This produces single-stranded overhangs. These are single-stranded overhangs, as in they're extra things which are there. They're complementary to what the sequences are there. They are called sticky because these ends can easily pair with the complementary basis of another DNA fragments, helping in joining foreign DNA with the vector DNA. Got it? That's all. Okay, that's all. As simple as that. And make the diagram and show it to them. It will be really useful. Now write 3 vector-free techniques that can be utilized to transfer recombinant DNA into a ready host cell. What is a vector-free technique that we can use without any vector? Any idea? So first one is micro-injection. Micro-injection is basically where I am going to so this is an animal cell. There is no cell wall over here. I can just use an injection which has my recombinant DNA and insert into the nucleus out there. That is known as microinjection. This is the case for animal cells where I do not want a vector. Okay, so directly injecting the recombinant DNA into the nucleus of an animal cell using a fine needle. That's all. Now the next thing that we have is biolistic or gene gun. I just told you for plant cell, you cannot insert like this because you have an extra cell wall, which is why you will need something else for entering things, right? That is known as gene gun method. Here high velocity tungsten and gold particles are coated with DNA and are shot into the plant cells. That is the exact thing that we do. Now the third thing is electroporation. Electroporation is basically electric pulses are created such that it creates temporary pores within the cell membrane, allows the DNA to enter. This is not given exactly in your NCERT, but please know that. Okay, otherwise you also have the calcium chloride medium where you are basically using divalent cations to make the bacterial cells take up the DNA. You can mention that also. That also works. It is also a vector-free technique which is there. Now, agrobacterium tumefaciens as a natural vector. So, listen to me carefully. The usual vector that we have is a plasmid. Fine. But in some cases, this is not the case. In usual, plasmids can act like a vector, but not all vectors are plasmid. We have vector-free techniques. We have some other vectors also. For example, in the case of plants, we use agrobacterium tumefaciens. Okay, this is in the case of plants. In the case of animals, we have retroviruses. Always remember, this is what vector that we have for animals. You know, HIV is there for transferring things. Similarly, disarmed retroviruses are being used for transferring things into the animal cells. Please have that logic in your head. Here you have been asked about agrobacterium tumefaciens, which is a natural vector in the case of plants. Why do you call it as a natural vector? That is a question. So agrobacterium tumefaciens, which is a bacteria, it has a plasmid. You call this plasmid as tumor-inducing plasmid. What do you call it? Tumor-inducing plasmid. I can also call it as TI plasmid. How does it induce tumor? Basically, it has a segment over here, and this segment is known as tDNA, that is transfer DNA. Basically, this organism goes and infects a plant. It goes and infects a plant, and when it does, it is going to transfer this tDNA inside the plant. And when it does, the plant will be developing what? Tumor. Now, suppose I'm using the mechanics over here for removing the tDNA part and basically inserting my gene of interest. What is my gene of interest over here? Any gene that I want to insert. Then I make the agrobacterium tumefaciens go infect. Again, my gene of interest is going to be transferred out there. That is why it is a natural vector that is being used in the case of plants. Again, if you want a vector-free technique for plants, you have biolistic. If at all you want a vector for animals, it is retrovirus. Again, if you want a vector-free technique, that is a microinjection. Please know about that. Okay. So agrobacterium tumefaciens is a soil bacterium that naturally causes tumors, that is crown galls in plants. It carries TI plasmid that transfers a segment called tDNA into the plant cell, transfer DNA into the plant cell. In genetic engineering, the TI plasmid is modified to remove the tumor-causing genes and used to transfer the desirable genes into the plants. Hence, it acts as a natural genetic engineer or natural vector for the gene transfer in the case of plants. Done. That's all. Cool. Now, write a labeled diagrammatic representation of recombinant DNA technology. You have to explain everything. So what do you do first of all, my dear children? You have the foreign DNA and you have the vector DNA. It can be a plasmid. It can be anything, but a plasmid, suppose. So always remember, okay, I have discussed this so many times. What do? With a restriction enzyme, you cut a gene segment. Similarly, with the same restriction enzyme, you're going to make a cut over here also. Done. Now, with the help of DNA ligases, you're going to join this. You have got the recombinant DNA molecule. Now, what do? Recombinant DNA molecule is made to put inside the host. This is known as transformation, right? Now basically the host which is there, it is keeping on dividing, and it has the recombinant DNA. That is the whole thing. That's the whole thing all about. So if you are asked about this, make the diagrammatic representation of this. If you are asked to explain the steps of each and everything in detail, you have to explain each and every step out there. First one is isolation of DNA. This is something that we have not discussed. So how do you isolate the DNA? Suppose you are isolating the DNA from a fungi. A fungi will be having, so there are three cases. Suppose it's a fungi. Fungi has a cell wall, and the cell wall is made up of chitin, which is why you will need what? Chitinase. You will need chitinase. Suppose this is a plant. A plant has cell wall made up of cellulose. So what do you need to break it down? Cellulase. Okay, suppose it is of a bacteria. A bacteria has the cell wall made up of peptidoglycan. Peptidoglycan. So what do you need? You need lysozyme, and lysozyme is an enzyme that is there in our tears also. Okay, so please remember. So you need enzymes like this specifically for specific things. If at all you are asked about fungi, please know you would need what? Chitinase. If you are asked about bacteria, you need lysozyme. If you are asked about plants, you would need cellulase. So please know all about that. There's one question that I saw, I think I have not included over here. I saw it somewhere in the previous year, where they asked you, they gave you number of things which are used in restriction engineering, genetic engineering technique, and they asked you, if at all you want to insert something into a plant, what do you need? What are the things that you would need? So please know all this thing. In a plant, you would need cellulase. Apart from that, after getting inside the plant, you have the cell nucleus which has RNA and protein also. So you have to remove the RNA with the help of RNase. You have to remove the proteins with the help of proteinase. You have to remove the fat with the help of lipase. And finally, you have the DNA that you have to take it up, right? So please know about that. Okay. So after isolating, you cut the DNA using restriction enzymes. And after that, you separate it with the gel electrophoresis. How do you view it? You use ethidium bromide to view it under the UV radiation, or basically autoradiography machine. Please remember, it gives a orangish red color, bright orange red color, ethidium bromide. Okay, and it will be only visible under UV radiation. So please know about that also. Now, insertion of the desired DNA into the vector. First of all, you separated the fragments. Now you took the fragments out there and you put it inside the vector. Now what do? You introduce the vector DNA into the host cell. So you put it inside the vector, right? Now this vector has to be put inside the host cell. So for that, what do? You use microinjection, biolistic, whatever method is there. Now after that, you do selection of recombinants, like with the help of antibiotic resistant gene or blue-white colonies, whatever is there. Finally, you check for the expression. And lastly, you do downstream processing. Downstream processing has two things: separation and purification. Okay, it is having separation and purification. Separation and purification, as in basically, if at all you want proteins, it's not like just proteins are going to be made. Lot of other things are also going to be made. You have to make sure that it's separated from everything. Apart from that, you have to purify it, and later you would also have to preserve it. Okay, so separation and purification are the two major things that comes under downstream processing. Later, you would have to do the other things as well. For example, adding preservatives, do the quality checking and everything. All of them comes under downstream processing. So these are the few steps. Please know about that. As I told, give yourself a chance that if at all I want to insert a gene inside a plant cell, and I'm taking this gene from a bacterial cell, what are the things that I will be needing over here in this entire process? You should know about what are the basic things that you will require. Okay? Yes. Done. Now, biotechnology and its application, again, you get a 5-marker questions coming off from this chapter. This is going to be the second last chapter. After that, we have organisms and population, and then we are going to end up. So let's see biotechnology and its application. The first one is genetically modified plants have been developed through core techniques of biotechnology. How are these genetically modified plants useful to the humans? So basically, they're asking you the importance of GMO plants, that is genetically modified organisms or genetically modified plants. So first thing, enhanced crop yield and efficient mineral usage. So genetically modified plants, my dear children, they can sustain with limited amount of minerals also. Okay, they do not need much amount of, because they're okay with this mineral usage out there. That is one very important thing, which is why farmers will not be need to spend too much on fertilizers out there. With the minimum nutrients out there, they can easily survive. Now, resistance to pests and diseases. So you might have seen the example of Bt cotton as well as RNA interference. Okay, this makes the plant resistant to pest. So that is also a good thing. Now, reduced use of chemical pesticides. Again, over here, since you are creating the plant itself, resistant against pest, you will not require chemical pesticides, and obviously when you're not requiring chemical pesticides, the soil fertility is also going to be maintained, because all those pesticides are going to mix up with the soil and going to cause trouble up there, right? That is also done. Tolerance to abiotic stresses. Plants can survive in conditions where there is drought, salinity, heat, cold, everything. It is going to sustain all of that. Now next thing is improved nutritional quality. Okay, the plants are going to produce something that is more richer in nutrient. For example, you might have heard about golden rice. Have you heard about golden rice? Golden rice is a rice which looks golden color. Why? Because it is vitamin A enriched. Vitamin A enriched. So in places such as Africa, where there was too much malnutrition going on at that place, golden rice was being supplied at some point such that they could at least have vitamin A in their food. Okay, so that is one thing. Next thing, reduced post-harvest losses. So after harvesting also there will be so many losses, because the plants and seeds which are there, they could be damaged and things like that could be happening. But with the help of recombinant DNA technology, with the help of genetically engineered plants, they could survive all of it a lot better. Cool. So these are the points that you have to write. Now, briefly explain the use of transgenic animals. So transgenic animals basically are the ones where I am going to put my gene into an organism. So this organism has a recombinant type of gene out there. We call this as recombinant, or basically a transgenic animal. Why do we need transgenic animals? The first one is to study of the normal physiology and development. For example, there is a factor that is known as insulin-like growth factor. Now this is a very important thing in our body. Now, I want to know, what does it actually go and do inside our body? What are the key aspects where it goes and shows its effect? What is its effect on the growth and development? So how will I get to know that? Basically, I put the gene inside an organism. Mostly 95% of the times, my dear children, the transgenic animal which is used is a mice. Always remember, it's a mice, not sheep, not goat, nothing. It's usually a mice. So when I put this growth factor inside the mice, what is going to happen? It is going to show inside the mice there will be some changes, effects on the growth, and with that changes I will be getting to understand that, okay, while inserting this, all this thing happened, so probably these are the functions of insulin-like growth factor. So for understanding the normal physiology and development, we use transgenic animals. So helps understand gene regulation and effects on growth and development and body function. Example is insulin-like growth factor research. Second, disease study and human models. For example, I want to see someone who has cancer, what are the areas which is being affected, such that I can create drug in response to that, right? So similarly, organisms have been made models for diseases such as cancers, cystic fibrosis, arthritis, Alzheimer's. So C square and A square, please remember it. Cancer and cystic fibrosis, arthritis and Alzheimer's. These are the diseases which have been studying using animals out there. So we make some changes out there in the body of the organism and see what does it have the effect in the, or basically for finding drugs also. Okay, for finding drugs against these diseases, we can try it on the animals. Obviously, this is a very bad thing to do, but this is how it works. Now, biological products. Now, this point is very, very important for production of biological products. There are two things which are given in your NCERT. One is alpha-1 antitrypsin. Alpha-1 antitrypsin is something that is produced in our body. It's a protein. It's a protein that is not a hormone. It's a protein that is produced in our body. And this protein helps us protect against emphysema, which is a respiratory disorder. Okay. Now, I cannot tell my body to produce it more. So what do? I take the gene and I make an organism produce it for me. Okay, so isn't it production of biological products using a transgenic animal? Yes. So don't forget alpha-1 antitrypsin. And why do we use it for? For treatment of emphysema, that is a respiratory disorder. The second thing which is there is alpha-lactalbumin. It's not alpha-1-lactalbumin, it's alpha-lactalbumin. This is again a protein. This protein is found in human milk. Now, I made a cow. A cow produced this particular protein in their milk, and it was the transgenic cow whose name was Rosie in the year 1997. So it produced a nutrient-rich human-like milk that contained alpha-lactalbumin, and again, it is super beneficial for organisms. Okay, so these two products, biological products, are super important. So first is study normal physiology and development. Second is disease study and human models. The third one is biological products. Now the fourth one is vaccine safety testing. So before vaccine were tested on us, it was tested on animals. Okay, just to see whether it is actually working or does it have any other issues being caused, side effects being caused. So polio vaccine, everything was being tested just like this. The next thing is chemical and toxicity testing. So basically what do you do that? You make an organism very sensitive to even little bit amount of toxins out there. And then when you insert the toxin, we'll see if an organism is this sensitive, what is the reaction that is going to happen? Because in humans also, there are organisms which are very sensitive, there are organisms which are not so sensitive. So we have to look for the people who are very much sensitive. What are the effects that are going to be shown? If it is too much effects, then probably the toxicity level is going to be less. Get it? So that is the thing. So genetically modified animals used for quick and accurate toxicity test for drugs and chemical testing. Over here, always remember, you need the approval of GEAC. That is Genetic Engineering Approval Committee for doing all this thing. Okay, Genetic Engineering Approval Committee for doing all this thing. Always remember that. Now here itself, go study about biopiracy and go study about patency, if at all you do not remember. I think I have taught you in 2 marker, 3 marker. No, I have not. In 1 marker, I think I have done. But please make sure biopiracy and patency is clear to you. Okay, done. Now, explain the application of biotechnology in the field of medicine. In the field of medicine, how does biotechnology play a very important role? Any idea? Any idea, my dear children? So just give me a second, okay? Let me see what all is left. Okay. First one is in the production of insulin. So, insulin which was there, it was produced when first of all, what is insulin? Insulin is a hormone. It's a hormone, okay? It's a hormone produced by the pancreas of human body, and particularly this particular hormone is super important for one very important thing, that is for trapping all those glucose and converting into glycogen. What is the use of trapping glucose into glycogen? Basically, it makes sure about one very important thing. That is what? That is the blood glucose level is being maintained. It is being maintained. That is the role of it. So insulin makes sure that our blood sugar level is not too high. Suppose some person has some problem in their pancreas such that insulin production is not happening. What do we have to do? We have to give them insulin. So usually insulin was taken from cattle and pigs and cows and things like that, but then people started developing what? Allergies in response to that, which is why a company came into the picture, and that was Eli Lilly. Okay, this Eli Lilly company faced a problem, that is, in the case of insulin, there is A chain, there is B chain, and in between this A and B chain, there is one more chain which is known as C chain. Okay, and over here you have the disulfide bonds. This is how it goes. This is what a pro-hormone, as in, this is not the mature hormone, this is a pro-hormone which is produced in our body. In our body, we have some system that is going to cut this particular portion from here such that we just have A and the B chain, and in between that we have the disulfide bonds. Now this is going to be the actual hormone, I'll call this as the mature hormone. So this machinery is there in our body in the case of eukaryotes, right? And you need something similar happening in some organisms' body also. So that is very, very, very difficult. Pro-hormone to mature hormone formation. Listen, this is a pro-hormone. Do not go and write pro-enzyme and come. It's a pro-hormone, and this is a mature form of hormone. Now, so Eli Lilly came into the picture and said, okay, let's take a prokaryote. We definitely understand that it cannot make a cut over here for this particular C chain because eukaryotes and prokaryotes are different. And when I take a E. coli, that is a bacteria, it's a prokaryote and it will not have the same machinery like me. One thing that can be done is that in one E. coli, I can put the gene for A chain. For A chain. In another E. coli, I can put the gene for B chain. For production of B chain, what do I get at the end? I get A from here, I get B from here. In the laboratory condition, can't we just make the disulfide bonds over here? Yes, and that is how we got the insulin, the human-like insulin, which is known as humulin, humulin, not millen, it is humulin. Please remember this. Okay, so what did we do? We took the A gene and the B gene, A polypeptide chain gene and B polypeptide chain gene. We put it in two separate E. coli. Both of them produced their own polypeptides, and now we joined it. That is humulin. It was produced by Eli Lilly company. Got it? So over here, what is the answer for this question? Biotechnology helps produce safe and large quantities of proteins using microbes. Example, human insulin, that is humulin. Earlier insulin was extracted from cattle, pig, pancreas, causing what? Allergies. Human insulin consists of A and B polypeptide chains linked by disulfide bonds out there. Eli Lilly created separate DNA sequence for A and B chains and inserted them into what? E. coli plasmid. Bacteria produced both the chains, which is purified and chemically combined to become the functional insulin. This allowed the large scale production. So please know, this is one application of biotechnology in the field of medicine, and this is super important. Okay, so pro-insulin to insulin, I hope you know, or pro-hormone to mature hormone, I hope you know the transition. Now the next one is gene therapy method. So gene therapy method is where basically this was used on a 4-year-old child who had a deficiency of one particular enzyme, that was adenosine deaminase. Adenosine deaminase, you call it ADA. So in her body, the cells did not have the gene. Okay, there was a deletion of the gene which was forming ADA, adenosine deaminase. Now, when there is a deletion of this gene, do you think adenosine deaminase is going to be produced? No. What is the problem if at all this gene is not produced? There will be toxin buildup. Okay, there will be buildup of some very not so important toxin which can create problems. Now, this is a very big difficult situation for certain cells which you call as lymphocytes, because in lymphocytes you cannot afford to have toxins being built up there. So that means we need this gene for survival. That is very important. So that's when gene therapy was being introduced, okay, into a 4-year-old child. What was being done is that the steps of this gene therapy is super important, so you should know this. Okay, first thing that was done, that lymphocytes were taken from here. So the first thing that was done is lymphocytes were extracted from the patient. First step. Second step, which we usually miss, we culture it. Okay, we culture the lymphocytes. Okay, we culture the lymphocytes outside the body. Now what do? We transfer the gene for ADA using a vector. Now you tell me, what is a vector? Over here it's an animal cell. For animal cell, if at all you are needing a vector, I'm not talking about vector free method, I'm talking about using a vector. What is a vector? Retrovirus. Disarmed retrovirus is going to be my vector over here. I transfer the gene. So basically in my lymphocytes, now I have this gene. Now what do? I basically put it back into the body. What is going to happen? After a few days, again the lymphocytes number are going to be less, because it's not immortal. The number of lymphocytes are going to be less. So what do you need again? You will need to do transfusion again. So ma'am, gene therapy, which is a method, it's not completely useful. It is useful, but it will be completely curative if at all gene therapy is done in embryological stage itself. When an organism is in the form of embryo, at that particular point, if at all it has been done, that's when it will be a permanent cure. Otherwise, it is not going to be a cure. Why not cure? Because over here the cells are not immortal. Similarly, there are other methods also. Bone marrow transplantation. Sometimes it does not work. There is another method where you give this enzyme repeatedly. Again, you need repeated transfusion. So always remember, gene therapy in adult stage, or bone marrow transplantation, or basically the enzyme replacement therapy where you give the enzyme again and again, all of them is not a permanently curative method. The only permanent cure that you have for this is doing gene therapy in the embryological stage itself. Okay, because those cells, if at all one cell has it, it will distribute it to all the other cells out there. Got it? Okay. So that is a general thing that we should know. So yes, lymphocytes withdrawn from the patient and then cultured. This step has been missed, so I'm writing cultured outside the body. And basically, what do you transfer? I told you transfer the gene, but you call this as ADA-cDNA. Adenosine deaminase complementary DNA is what you take. Okay, complementary DNA. So please remember it as such. If at all you want an idea, I can give you, but I was thinking about the time. So listen, DNA is there. DNA transcribes into an RNA. You know, basically exons, introns, both of them are there. Here also exons, introns will be there in HNRNA. Now what do? You remove the introns, you will just have the exons only, which codes for things. That is known as an mRNA. Now, when you just have the protein and you just want this particular protein, what do? You make a complementary DNA. You make a complementary DNA out of it. So from an RNA, you're doing reverse transcription to make a DNA. This is known as complementary DNA, because we do not want the introns and things like that. That is why we did this. And then we put it inside the body. That is why we call it complementary DNA. Just know about it. Okay? Works, ma'am. Perfect. Now, ELISA is one of the methods of molecular diagnosis. What is the principle of this technique? I told you what is the principle. There is antibody antigen linkage interaction. A specific antigen, virus, bacteria, protein, or specific antibody is attached to a plate. Either it can be antigen or it can be antibody against HIV virus, right? The patient sample is being added. If the patient has matching antibody or antigen, they will bind together. Over here we have taken antigen, which is going to bind. A coloring agent, chromogen, is also mixed. The enzyme reacts with the chemical to produce a color. If a color change is there, that means antigen antibody is being protected. So no color change means disease is not being present. So again, how do you do it with antigen? Basically, you know HIV viruses that causes things. So you put it over here. Now, the patient's blood is taken. In the patient, if at all that person is having the disease, obviously they produce antibodies against it. So the antibody will come and attach. Okay, so you get it? So this is the same logic that is working. But in general case, we just talk about antibody, antigen, antibody. So you can just also talk about that also. Now, disease is what? HIV, AIDS. Now, always remember in your NCERT there is one question. PCR, polymerase chain reaction, is also used for diagnosing diseases. Is the answer yes? Yes. Whenever the question comes that a method which is used for finding the trace amounts of organism, very little amount of thing will be there, still you can find the disease using which method? It is PCR, because PCR you do amplification, right? And PCR can also be used for detecting HIV. Know that. Can be used for detecting HIV. Please know that. Now, traditional breeding techniques have failed to keep pace with the demand and to provide sufficiently fast and efficient systems for crop improvement. In this context, explain how the tissue culture technique has contributed to crop improvement programs by mentioning any 5 of its application. What do you do in tissue culture? First of all, you need to know that. Suppose this is a plant. You take the tip of the plant. Okay, why tip? Because over here you have meristematic cells. What do you call the cells as? Apical meristem. Okay, or probably you can take from the bud also, because this bud will be giving rise to what? A new branch and things like that. You call this as axillary meristem. So usually you take from apical and axillary. This is super important. Why? Because if at all you want to produce virus-free plants, you have to take from here. These parts will, suppose this plant has virus attack being onto it, okay? But these parts will be free of the virus. Apical and axillary will be free of the virus. That is why you take those particular portions. Now you take that, you now call this part as explant. What do you call it? Explant. Now explant you put in a culture medium. The cells will grow and it will fall something like this, which is known as callus. A group of cells which is known as callus. Now the callus is transformed into what? A nutrient medium containing rooting hormone and shooting hormone. A new plant is there. Suppose you make multiple plants like this, thousands of them, you will call this as micropropagation. You will call this as micropropagation. Now what is the use of this? First of all, regeneration of the whole plants from the explant due to property of totipotency. First thing, what is the use of tissue culture? Is that I can regenerate a whole plant from the explants. Why? Because there is totipotency. Totipotency means a cell has the potential to become anything it wants. It has maximum amount of potential, totipotency. So regeneration of the whole plant is possible from tissue culture? Yes, ma'am. Rapid production of large number of plants in a short time through micropropagation. When I say tissue culture is being used to produce thousands of them, I call this as micropropagation. So can I produce thousands of plants? Yes, ma'am. Production of genetically identical plants. Can I produce genetically identical plants? Suppose I have a plant which is super good and I want multiple copies. Can I produce with that? Yes. So genetically identical plants I can produce. Next thing, commercial production of crops like tomato, banana, apple. If at all I want to in a large scale, can I do it? Yes, ma'am. Recovery of virus-free plants. Can I produce virus-free plants also from this method? Yes. But for that, what do you need? Axillary and apical meristem. Axillary and apical meristem. Always remember this. Okay, and the plants that you get from tissue culture is known as soma clones. Soma clones. Why? Because they are all from the same cells, though it will be like clones. Somatic hybrid is different. Soma clones are different. Somatic hybrid is when you take two plants, two separate plants. For example, tomato, potato. You take two cells, remove the cell wall of both of the cells. What is left behind is known as protoplast. What do you call it? Protoplast. Protoplasts are fused. This is known as somatic hybrids. Now we will call this as pomato. What do you call it? Pomato, potato and tomato both. Done, ma'am. Now what is RNA interference? How does RNA interference help to develop resistance to tobacco plant from nematode infection? Then that is the next question for which you should know the answer. So listen to me carefully. There is a nematode, an Aschelminthes, which is known as Meloidigoin incognita. This is that particular organism, right? Now what does it do is that it affects the root of the tobacco plant. And how does it do? It goes inside, forms a cyst and be there. It's not this big, okay? It's very, very small thing. It goes inside and basically have its DNA, make the RNA, that is the mRNA. And this mRNA will undergo what? Translation to form the proteins, and thus proteins are going to cause trouble in this particular plant out there. Okay, so what is the method in which I can prevent it? There is only one way I can prevent it, and that is RNA interference method. RNA interference method. What am I going to do over here? How am I going to interfere in between this? Basically, I take some genes from here, from the same organism, and I put this gene inside what? Inside a vector, because for transferring something to the plant, I need a vector. Which is a vector? Agrobacterium tumefaciens. So here I put the gene of my interest instead of transfer DNA place. This goes and affect the cell in the plant. Now in the plant, my dear children, what is going to happen? In the plant, this particular gene that I have inserted over here is going to produce double-stranded RNA. The gene that I have taken from here and put it inside the agrobacterium tumefaciens is now inside the plant, and this gene is going to produce what? Double-stranded RNA. dsRNA is what I call it. It is like this. Okay, ma'am, what is the use of this? This double-stranded RNA is going to now target the mRNA. What? So listen to me. mRNA is actually single-stranded structure. Only when it is single-stranded, then only it can undergo translation to form the proteins. Now suppose this double-stranded mRNA came into this particular picture. This double-stranded RNA came into the picture over here. So what? Will it make triple-stranded RNA now? No. This double-stranded RNA, oh my god, just a minute. Okay. I need patience, yes. So this double-stranded RNA was there, and your mRNA was right over here, which was going to undergo translation to produce the protein, right? Now what happens is that this double-stranded is broken down. One strand is free over here. That one strand comes and makes a bond over here. Now do you think mRNA is single anymore? No, Baba, it has become mingled. So it becomes double-stranded. Do you think double-stranded mRNA can undergo translation? Do you think it can undergo translation? The answer is no. Do you think proteins is going to be made? No. Do you think plant will be affected by myeloid incognita anymore? No, Papa. That is the logic. So we are interfering in the RNA, and for that you need double-stranded RNA itself. Out of that, one strand comes and binds, but always remember it's a double-stranded RNA that you are requiring over here. Very, very, very important. So how do you write this answer? Again, so identifying the nematode gene. Scientists first identify the genes in the nematode that are essential for its survival or ability to infect the plant, using Agrobacterium tumefaciens to insert the gene. So to deliver the silencing gene into the tobacco plant, scientists use Agrobacterium. So I took the gene, I used Agrobacterium tumefaciens for that, and basically now the Agrobacterium tumefaciens which is there, it is going to infect the plant cell. Now in the plant, it will produce double-stranded RNA. Right? Now double-stranded RNA triggers RNA interference. It goes and does the RNA interference, silencing the targeted genes in the nematode body. What is going to happen? Basically, the plant is not going to produce anything anymore. Got it? This is RNA interference. For this you need double-stranded RNA. Very, very important thing. Now there is one more application of biotechnology in agriculture, and that is BT, cotton and things like that. What do you do over here, and what is the logic being used over here? This is also super important. So let's see. Over here, the first thing that you require is Bacillus thuringiensis, which you call as BT. Remember, it has a gene which is known as cry gene. This cry gene is responsible for production of inactive toxin. I call this as protoxin. Why is it inactive over here? That is a question. Why is it inactive? Why it does not affect the BT, that is Bacillus thuringiensis? Because for it to be active, you need some ambient pH, and that is not being given over here in BT. Okay, fine. Suppose an insect somehow took the BT inside. Suppose this was a butterfly caterpillar, and now inside the butterfly caterpillar you have this. Okay, so here you have the Bacillus thuringiensis, and this is suppose the mid-gut region. What is going to happen? The protoxin over here, the inactive protoxin over here becomes active, my dear children. Why it becomes active inside the midgut? Because here you are providing an alkaline pH. Because of this alkaline pH, what is going to happen? The protoxin becomes active form of toxin, which is exactly why there will be perforations found over here. There will be perforations and later the organism will be killed. First of all, it targets the epithelial cells of the midgut region. Later leads to perforation leading to the death. So, ma'am, the protoxin which is produced is inactive because there was no alkaline pH in Bt cotton in the Bt organism. Exactly, that is the reason. And when you got the alkaline pH, it becomes active. This is super important for you to know. Now what do I do is, I take this gene and I put it inside the plants. The plants will also produce this protoxins which will become inside any organism that will try to eat the plant. Any organism as it is very specific, it has to be an insect. So this cry genes are insect group specific. Always remember that. So there are multiple type of cry genes in your NCERT. It has been written like this. So there is cry 1AC and there is cry 2AB. Okay, sorry, AB. Cry 1AC, the first letter of the first word is going to be starting with C. So I am writing cotton. C. Second word starts with B. Second word is bollworm. And does it start with B? Yes. So you have two genes, cry 1AC and cry 2AB for killing cotton bollworms. Similarly, you have cry 1AB. Cry 1AB for corn borers, for killing corn borers. So how do I remember it? There is only one, that is here you had two of them, here you just have one, and basically the first letter is C and the last letter is B. Okay, I know it's confusing, but you need to remember it. It's been usually asked in MCQs in every question out there. Cry 1AC, first letter, first word starts with C, second word starts with B. So both of them are like CB only, but here it is pretty long, that is why you have two of them. Here you just have one, and basically C and B is starting and the end. Okay, so why do I ask you to remember it like this? That sometimes you might go write 2AC over here, 2AC or 1AB. So always remember only one is there and it's B at the end. So that is a corn borer. Done. Now, the cry gene, cry 1AC and cry 2AB from thuringiensis coding for the toxins are transformed to the cotton plants. The genes have been transformed. Proteins encoded by these genes control the cotton bollworms. Bt cotton plants express the Bt toxin by producing toxin that provides insect resistance. Bt toxin exists as inactive protoxins. Insect injects the inactive toxins, convert to active in the alkaline pH, okay, which solubilizes the crystal. In this alkaline pH, it will be able to solubilize the crystal and the toxin will be there. Toxin binds to the surface of the midgut epithelium, creating the pores out there. Always remember, that is very important, and they kill insects such as lepidopterans, coleopterans and dipterans. Lepidopterans, coleopterans and dipterans. Girls, this is very important. Cool. Now let's go ahead with the last chapter, that is organisms and population. With this, we will end it up. So it goes like this: write an equation to calculate the population density at time t. So what is the population density at time t? How do you represent it? Nt is equals to n naught. This is the initial population plus b plus I. B is what? The birth rate. I is what? Immigration rate. Organisms getting inside the population, that is immigration. Minus death plus emigration, the organisms going out. Okay, B plus I minus death, D plus E. Okay, this is very important. So write an equation to calculate the population density at time t. This is the equation, minus has to come, this has to be put inside bracket also. Now population density fluctuates due to changes in the four basic process. Explain the four basic process. Natality means what? The birth rate. Okay, the number of individuals reproduced in a place at particular time. Mortality is the number of individuals lost from a population per unit time. Immigration is the next thing, where the number of individuals which are being added up. Emigration, number of individuals which are being living in the population. Out of all of them, these two are the most important thing. We consider immigration and emigration only when there is colonization, as in some organisms are coming from outside to inside and things like that. Okay, I'm going a little fast because yes, So what is mutualism? Explain any 4 examples of mutualism. Or there is a different question that I saw. That is, what is mutualism? Give any 2 examples. Plant-animal interaction often involves co-evolution of mutualist. Justify your answer with an example. This was also given. So what is mutualism? Is where both the interacting organisms are getting the benefit. Example is lichen, example is mycorrhiza, and example is fig tree and wasp, and also rhizobium bacteria and leguminous plants. Listen to me very carefully. Lichen is a combination of algae and the fungi. Mycorrhiza is a combination of plant and the fungi. What is common over here? Fungi is common over here. Algae produces what? Algae can produce its own food, and this food can be given to the fungi. Plant can also produce its food and give it to the fungi. So in both of the places, the producers help in that case. In the case of lichen, the fungus help in providing protection and also absorbing water and nutrients. Listen, the fungi has the ability to go inside and take in everything which is there. So basically, fungus will be helping in absorbing water and the minerals, as well as it helps in the protection in the case of lichen. In the case of mycorrhiza, the fungi will go deep into the soil and get phosphorus. This is very important. You have studied it in microbes in human welfare. Apart from that, growth and development, overall growth and development also it helps. Resistance, to provide resistance again other pathogens also it helps. On the plant other hand, plant provides the food to the fungus. So both of them are getting benefited. Now rhizobium bacteria and leguminous plants. Here the rhizobium bacteria fixes nitrogen and gives to the plant. What does the plant do? Plant again makes food and gives it to the rhizobium bacteria, because rhizobium bacteria cannot prepare its own food. Got it? Again, both of them has a relation like that. Now fig tree and wasp is there. Wasp helps in pollination of the fig tree. Right, and fig provides what? Food and also space for the egg to be laid. So what is mutualism? It's an interaction where both the species are getting benefit. Right, plant-animal interaction often involves co-evolution of mutualist. Why? Because over here, my dear children, if one organism is getting any changes out there, if the other organism do not evolve, do you think both of them will be able to sustain without each other? No, because if fig tree is not evolving, you will not get any space to lay the eggs. If wasp is not evolving, pollination is not going to happen. That is why co-evolution has to be taking place. So justify your answer. This is how you have to justify, by telling the examples of it. So 4 examples of mutualism is being mentioned over here. Please know different type of questions can also be framed. Next, name the type of interaction seen in each of the following. Clownfish living among the tentacles of sea anemone. So you might have seen so many tentacles that is there. In between that you have a fish. What type of relation does it show? It is showing commensalism. It is showing commensalism. Now this is super important. Commensalism is super important. What is exactly happening in the case of commensalism? One organism is getting the benefit. What is that one organism? Clownfish. And tentacles of sea anemone. Anemone is not getting benefited, not getting harmed. That is why it is a +0 relationship out there. Koil and cuckoo bird during the breeding season. Koil goes and lays Koil and cuckoo bird, it's the same thing. Koil and crow is being asked, okay? Koil and crow. So koil goes and lays the eggs in the crow's nest. What does it do? Basically, the koil has evolved. It's a parasite, okay? It has evolved in a way that its egg looks like the crow's egg. Now the crow papa guy will go provide nourishment to all of them and basically gives its energy to growing a koil's egg out there as well. So this is parasitism, where one organism is getting benefited, that is the koil obviously, and the crow which is there, it is getting negatively impacted. And this specifically, if we say, it's brood parasitism. This is what you call as brood parasitism. Always remember, host is the crow and parasite is the koil. The koil evolves to look like crow. It's not like host will evolve. It's the koil who is going to evolve. The barnacles growing on the back of the whale. Now barnacles which are there on the whale's body, my dear children, they are there for transportation. Barnacles, does it cause any harm to the whale? No. Whale, does it provide any good thing to the barnacle? Yes, it allows in transportation. So this is a +0 relationship. No harm, no benefit. So again, it is commensalism. Now we have marine fish and copypods. Just like how you have lice and humans, how you have ticks and dogs, similarly you have marine fish and copypods. Copypods, my dear children, they are parasites which are going to feed on the marine fish. So marine fish is negatively impacted, copypod is positive in that. This is parasitism. Now sexual disease in Mediterranean orchid, that is ophrys. So over there, the bee that comes to the flower, that is ophrys flower and orchid, it undergoes pseudocopulation. Okay, it undergoes pseudocopulation, thinking that's a female flower. So the female bee out there, right? On the other hand, so it gets the benefit of doing pseudocopulation out there. And it can also take some pollen grains also. That is one benefit. Next thing, the plant gets benefit because pollination is definitely going to happen. So this is mutualism, and this is also an example of sexual deceit, which has already been mentioned out there, which is why you just say it is mutualism. Actually, we cannot consider it like that, but yes. Now, explain the fascinating forms. I have already explained, just go through it. I'm not going to re-explain it because that will waste a lot of time. It is exactly what I have told you in previously. So yes. Now, mention the population interaction exists among the following: abundant tortoise and the goats. So abundant tortoise were there on one particular area. They were feeding on the grass. When goats were introduced, what happened was the abundant tortoise become extinct. It's because of competition, my dear children. They were both competing for what? The grass out there. So competition led to competitive exclusion of abundant tortoise. This is competition. Tiger and deer, my dear children. Are you? Tiger and deer. Tiger will go eat the deer. So there is prey and predator relationship. This is predation. Sea anemone and clownfish, we just saw it is commensalism. Wasp laying egg in the fig fruit, it's basically mutualism. Cuscuta growing on the eggplant, it will be parasitism. Why? Cuscuta does not have chlorophyll to make its own food. That is why it depends on the plants for food. Okay, so that is parasitism. Plus minus, because the plant is getting negatively impacted. Okay. Now, what is commensalism? Explained with two examples. Commensalism is an interaction where one organism benefits, other is neither harmed nor benefited. Example, orchid growing as an epiphyte on the mango branch. On the mango branch, you might have seen orchid. Orchid is not there for harming the mango plant. Orchid just needs sunlight. Mango plant, does it have any benefit or negative effect? No. But is orchid getting the benefit? Yes. This guy is not having any benefit nor harmed. So plus zero relationship. Clownfish and sea anemone, we just told. Barnacle on the back of the whale, again, it is just for transportation. It does not cause any harm to the whale. Whale does not get any benefit also. Cattle egret and grazing cattle. So whenever you have seen cows moving, you might have seen some birds. That is known as cattle egret. Why they are just around the cow? Because whenever the cow moves, basically it flushes off the insects. Now the insects are being fed by the cattle egret. Okay, so it is getting the benefit. Here it is neither benefit nor loss. Okay, now state Gauss's competitive exclusion principle. Two closely related species, okay, that survives on the same resources cannot coexist indefinitely. Two closely related species that feed on the same resources cannot stay together indefinitely. One will definitely be eliminated over time. That is known as Gauss's competitive exclusion principle. Example is abundant tortoise as well as this thing. Good. Okay, now what are ectoparasites and endoparasites? List any three parasitic adaptation. Parasites are the one that feeds on the ectoparasites are the one that feeds on the external surface of the host, and parasites that are living on the internal body, it's known as endoparasites. Okay, so ectoparasite is this that lives on the external surface, and endoparasite is this which lives inside our body. Now, which lives inside our body, these parasites will be having some adaptations. For example, they will not have any unnecessary sense organs such as ear, things like that, because they want to move very easily and they want to make their bodies literally lightweighted. So this is first thing. Presence of adhesive organs and suckers, because they do not have hand to cling on to places, so they will be having adhesive organs or suckers. Third one, loss of digestive system, because for example, tapeworm stays in our body. In our body, digestion happens, the things are broken down to very small components. These things have to be just absorbed by the tapeworm. They do not have to break anything down, which is why again they do not have any digestive system. Now they will be having high reproductive capacity to increase their number, and mostly they will be having a very complex life cycle. Example is given in your NCERT and you should not be missing out on it. That is human liver fluke. Human liver fluke, my dear children, it has two intermediate hosts. The last host is human beings. It has two intermediate hosts. One is snail and one is fish. It has to get inside the snail, it has to get inside the fish, then it will come into our body. So we are the ultimate definite host, but here these two are the intermediate host out there. Okay, this is for human liver fluke. Very, very important. Snail and fish. Okay, snail and fish. Very, very important. Cool. Now, predators are prudent and conduits in nature. Explain and support your answer for giving reasons. So, why do we consider them as predators? Prudent means they are clever enough to understand that if I eat all of the prey, tomorrow I will not have food. So, they avoid over-exploitation. That is why we say that they are prudent. So, predators play an important essential role in the ecological balance. They are considered prudent because overexploitation of prey would lead to their decline due to food scarcity, which is why they will not do overexploitation. Now, why do you call them conduits? Conduits as in they are channels. Channels of what? Energy transfer. So from the lower organism, they transfer the energy to the upper organism. How? By acting like a chain. So predators are conduits in the energy flow of an ecosystem, transferring energy from prey to the other organisms which are there. So lion, hunting, okay, that's all. So that is what you have to write for prudent and conduits. Now there's one more thing. They are asking about the defense mechanism that have been adopted by the prey population. And here you have defense mechanism in the case of animals and plants separately. In the case of animals, the basic example that you have is cryptic coloration or basically camouflaging. So if at all I put a green frog over here, will you be able to see it? No. Why? Because it has been camouflaged. So this is a defense mechanism that you have, camouflaging. The second one is some are poisonous and therefore avoided by predators. Some organism, for example, monarch butterfly. You might have seen the butterfly, right? Monarch butterfly is blue and orange in color. This butterfly produces highly distasteful substance such that the predator will not come and eat it again. Okay, so that is why. So some can produce poisonous substances or basically highly distasteful substances such that the organism will not eat it again. And monarch butterfly is an example for that. So in the case of animals, you can write camouflaging and also the example of monarch butterfly. Now in the case of plants, you can give the examples like this. First one is, you need to understand that most of the insects are going to eat the plant. Almost 25% of eats are phytophagous. That means the plant needs protection. First one is morphological protection in the terms of production of thorns. Now thorns are there in the case of acacia and cactus. And basically these thorns will be preventing any organism to come and eat it because they will have thorn attack being done. That is morphological defense. Then there is chemical defense, where what is going to happen? Some plants are going to produce some toxic substances, for example, cardiac glycosides. This is very important term. Cardiac glycosides. These are being produced. When these are being produced, what is going to happen? Basically, the organism might even die. It can even die. This is seen in the case of calotropis. You might have seen this. This is a wild plant that is being grown. You call this as what? Calotropis. So in calotropis, what do we have? We have cardiac glycosides that is there for protection against herbivory. Please know about this. Okay. Apart from that, there are certain plant defense chemicals that are being produced which we use for our use. That is nicotine, strychnine. Okay, nicotine, strychnine and quinine and this thing, what do you call it? Caffeine. All of these things are something that we utilize for our purpose, but actually it's plant defense mechanism. So do not forget calotropis and cardiac glycosides, thorn in acacia and this, monarch butterfly and also camouflage. These are the prey defenses. Okay. Now what do we have? Write any 4 factors that influences population density. Okay. What are the 4 factors that influences population density? I told you: natality, mortality, immigration and emigration. Draw a graph showing exponential growth. Exponential growth, Baba, how does it look? It looks like this. Over here you have population density and over here you have time. Exponential graph will always grow up. It will always grow up. And it produces what? A J-shaped curve. So, ma'am, do you think it's realistically possible? No. Because in every environment, my dear children, it has a carrying capacity. We all fight for resources. At the end, we will all fight for resources. Why? Because resources are limited. Now, this is a condition where resources are unlimited. That's when the organism will grow exponentially and will produce a J-shaped curve. J-shaped curve over here. In nature, we have another curve which is known as logistic growth, which goes like this. It grows and then it comes like, so this is an S-shaped curve. It was initially J, but it becomes S-shaped curve. Why? Because there is a carrying capacity being involved over here. There's a limitation out there. Always remember that. So here there is not. So how do you have different formulas for both of them? Yes. In exponential growth, the formula is dn by dt is equals to Rn. R stands for intrinsic rate of natural increase. That means it stands for birth minus death. Okay. So in a population where the birth rate is more, obviously population density is going to be more. If death rate is more, population density is going to be less. R is intrinsic rate of natural increase. I can also say that according to Darwin, it is said as reproductive fitness. So high R value means basically high, more reproduction. B is more, right? B is more, birth is more, means more reproduction. So you can also consume like that also. Now over here, how do you reframe the formula? The formula is exactly similar up to some point. dN by dt is equals to Rn, but here you have to talk about K minus N divided by K. What is K? Carrying capacity. As I told, beyond carrying capacity, nothing can go ahead. So K minus N divided by K, that is super important for here. This is a formula. Please make sure you write it, you practice it. This is a population density. This is time. Okay, this is a realistic thing that is usually found. Now I think I have a question which was there in the question bank and I needed you to solve it. So please make sure this is not 50th question. This is going to be the 10th question. So I had made it solved in the 50 most expected question, which is why I kept it. So refer to the graphs given below showing the growth pattern in organism and answer the question that follows. Curve A represents which type of growth? So curve A is given right over here. And which type of growth does it represent? It is exponential growth. dN by dt is equals to Rn. Curve B, my dear children, it represents what? Logistic growth. You also call this as Worlds-Pearls logistic growth. Okay. Now, which growth model is considered more realistic one in nature? Which is considered more realistic one? This one. Why? Because we definitely have limiting resources. That is why it is considered more realistic. Fine. Now, when bacteria are cultured under controlled optimum environmental condition, which curve is obtained? When bacteria are cultured under controlled optimum, listen, here they are asking you one very tricky question. Controlled optimum environmental condition. That means you have, optimum means you have everything which is needed for you to grow. And it is controlled also. Controlled as in you have to think in a way that I am making sure whatever extra is there, I am removing it. Whatever thing I have to provide, I am providing it also. So it is optimum condition, definitely optimum condition, and I am controlling it in a way, in a good way only. That is, I am removing the waste products that can create toxicity and everything. So this is a good thing. If this is a condition, then organism will be growing in an, which condition will it grow? It will grow in exponential phase only. Since optimum is given and since controlled is also given, if at all they had mentioned you only controlled environmental conditions, that means that basically here not everything is free. Okay, in that condition it would have been logistic growth, but here they have mentioned controlled optimum environment. That means everything which is necessary is given. It is controlled also. That means super good. It can easily grow. This type of questions can be confusing for you, to listen to me very carefully. Sometimes they give you ideal environment. If only ideal is given, then exponential growth. But if at all they are saying ideal environment with limiting resources, ideal environment is there, but sooner or later everything is going to get limited. Limited term comes into the picture. That means it's talking about the S-shaped curve. Okay, if just ideal, then this. If limiting term is coming, then this. Similarly, over here, controlled optimum environment means it's all good only. Only controlled, if it's there, it's bad. Then it will be this. Okay, if controlled optimum conditions, then it is this. Cool. So it is, remember, it is exponential thing that you are going to get. Next thing, the interactions such as predation, competition, parasitism can be represented by which curve? Whenever we say predation is there, parasitism is there, competition is there, which curve is there? You have too much competition out there, right? So obviously the curve would be this one. There is limiting factors. You do not have food. Why? Because there are predators and things. So I'll say in that case it will be logistic growth. Done? Done. Very, very, very important. And I think so these chapters do not have any 5 marker, and I think with this we complete all the 5 marker questions. Well, I really hope you watch the entire video, because it is definitely going to be useful for you. And if you have any doubts, do let me know. If you think that this video had helped you, definitely do let me know about that as well in the comment section below. Wishing you all the best. Please take care of yourself. Lots of love. Bye-bye.